Good morning, everybody. So, ठीक है जी. I think I'm on. You can hear me, na? Oh. So let's start with the. It's supposed to be at 8:30. We are already 5 minutes late. Today we are going to start with the first speaker. Here is Professor Lija Rajshekhar. She is what is not in a she is born and brought up in Kolkata, ex-alumni of Calcutta Medical College. She was from, got a MD from PGI Chandigarh. She is and currently working in the Nizam Hospital, Hyderabad, and she's a giant in adult rheumatology and a very known person in pediatric rheumatology and immunodeficiency diseases. I request Professor Lija Rajshekhar to deliver his talk. Dr. Lija, please.
Thank you, Dr. Tapas, for the kind introduction. And it's really a privilege to be here on the platform of ISPID. And thanks to Dr. Priyankar for giving me this topic as an opening batsman. And what I will do in this talk would be to take you through the pathophysiology as well as the phenotypes of our experience with monogenic lupus. If you look at this slide, you will see that I have listed a few slides, a few genes, and I have tried to categorize them into pathway so that it is easy for us to understand. This slide also highlights the fact that while the pathways may be different, since I'm speaking on monogenic lupus, the phenotypes converge. And also that the genes listed here are incomplete. As you read every day, you will see more and more genes described which will explain the entire phenotype of lupus as we know in adult sporadic lupus. So starting with the most important one, the most celebrated molecule associated with monogenic lupus, the opening batsman of the complement proteins, that is the C1q, which is basically a polypeptide which has 3 types of peptides in its globular head, that is the A, B, and C, and it behaves like an immunoglobulin molecule with a tail which resembles the Fc portion of the immunoglobulin. So as we all know, that is the globular part which recognizes the immune complexes bound on bacterial or viral or microbial or any other surfaces. And once we have this C1q attached, then C1rs complex is formed, and ultimately it is this C1q-RS C3 complex which initiates the classical complement pathway and goes on to the receptors which are present on many cells of the innate and adaptive immune system, and then triggers the further immune response to pathogens. Putting a dampener on these complements are the regulatory proteins, and I'm just describing one of them. There are so many, but the one which has been described with the phenotype of lupus is the complement factor I, which basically which causes the degradation of C3b and C4b. And when there is a mutation, patients who cannot stop this degradation present with hypercomplementation activation and a phenotype of monogenic lupus. A very important molecule which is directly in the path of clearing DNA is the DNase 1 ligase 3, which is one of the 4 or 5 DNA endonucleases. What is important about this enzyme is that it is the only enzyme which can cleave complex DNAs. What do you mean by complex DNAs? Which are complexed with proteins and lipids, and they are found in apoptotic microparticles. They are found in the chromatin, which is extruded from cells, and in the neutrophil extracellular traps, where the DNA is complex to many antimicrobial peptides also. Now, when there are defects in DNA ligase, there is accumulated cell-free DNA, and this accumulated cell-free DNA is engulfed into the PDC, where it triggers a huge TLR79 pathway MD88 mediated production of interferons. Now we all know that the endosomal TLRs are important in the pathophysiology of lupus. It is very well known that TLR 7, 8, 9 and including 3, they are the endosomal and they recognize single-stranded, double-stranded RNAs and DNA, especially the TLR 9 which recognizes the CpG DNA, which is present on many microbes, viruses, bacteria, and even the mitochondria. And through this pathway, by working through the downstream molecules of MD88, we have activation of the interferon pathways. We have descriptions of monogenic lupus associated with variants in UNC93B1, and this is very recent descriptions. But what this protein does is basically it helps in the trafficking of the endosomal TLRs. So when these endosomal TLRs, when there is variants in UNC93B1, then there is hyper secretion of TLR9, and normally they are downregulated when this is a normal wild type variant, but in the presence of mutations, you have a cascading TLR9 pathway here. Similarly, you could have gain of function mutations in TLR7, again, which downstream results in high interferon levels and a lupus-like phenotype. Now, once you have a very high interferon levels in the body, then these interferons and the inflammatory cytokine milieu, they push the B cells away from the normal pathway of the germinal center, where they would have undergone selection of autoreactive clones, into an extrafollicular pathway, where even autoreactive B cells are released into the bloodstream as those producing autoantibodies, as well as those which go into short-lived plasmablasts and even memory cells. Another gene which has been associated with the phenotype of lupus, though better known for its role in monogenic Behçet's as a haploinsufficiency, is this one, A20. Basically what it does is it switches off the NFK beta activation. Once it is mutant and this NFK beta activation is not switched off, then an inflammatory milieu develops, which again pushes the adaptive immunity into the B cells, becoming autoreactive, remaining circulating in the stream and causing disease. And this is how it happens. Another protein which is important in monogenic lupus is the trap protein. Basically, in trap protein also, it is related to the endosome lysosome, and endosomes and lysosomes of important cells like the PDCs, the macrophages, and the osteoclasts. And why this is important is because in this particular gene mutation, you get a double phenotype of spondyloepiphyseal dysplasia, endochondral dysplasia, as well as lupus. And why this happens is because this protein is important. Dephosphorylation of osteopontin in macrophages, in osteoclasts, is important for the function of both. And when it is impaired, then the osteoclasts don't migrate, they remain in one place. And this is just a representation of the truncated protein. The part in red on your left, on your right, is the part of the protein which is lost when the mutant happens. So when the dephosphorylation of osteopontin does not happen, then again TLR signaling is amplified. Moving to a slightly different pathway and impinging on to interferonopathies is mutations in IFIH1, which is basically a cytoplasmic viral receptor, which works through the MVAS adapter molecule and again activates interferon signaling. Mutations in this have been described not as only with severe viral infections, because it is basically a viral sensor, but also with the phenotypes of interferonopathy as well as autoimmunity like lupus. So among the adult rheumatologists, we don't think about single genes because we have a large number of lupus and their phenotype is so classic that in the absence of unusual features, we are not thinking of monogenic lupus. We think about it when we see a young child. We also know that in each of these sporadic lupus, we may find many variants in many genes, and they could probably be interacting with each other and with environmental trigger to produce the phenotype of lupus. So, and So let me just take you quickly through the phenotypes that we have seen in our clinic. So this is a child with C1qa mutation, the classic lupus, this is what as adults we see day in and day out. And in this child, she was born of consanguineous marriage, and there was a classic course, and the classic course included a myelitis, a nephritis, a remission, and then failure of therapy followed by relapse, and so it goes. This is captured at a time when she was doing well, but again, what is important here is that though she started her illness with a thumb abscess, her course is not of repeated major infections, it's more of lupus. We have another child with the C1qa mutation at a different site, again a classic lupus. Another child with C1qb mutation, but born of non-consanguineous parentage with almost complete lupus, though there is no lupus nephritis. Even adults, 50% don't have lupus nephritis. And there is no hypocomplementemia in this patient, but there is a homozygous mutation. So there is previous literature which describes up till date almost 70 patients with these mutations. We have people in the audience who have described their own cases. The cases that we have described today, I have described today, are not yet published. So I'm sure these 70 must be many more. And it's just a matter of time that we describe much larger populations, and then another matter of time before we decide whether these patients need to be treated differently or not. The CFI, which I discussed, which is a dampener of the complement system, a large deletion as well as a novel mutation led to a phenotype of hypocomplementemic urticarial vasculitis. You may note that the complements were rock bottom. However, there was no phenotype of classic lupus in this patient. Other phenotypes which has been described in literature include recurrent infections, include viral infections, as well as prominent vasculitis. We have had probably three children with DNase 1 mutation. The first child we saw presented with only diffuse alveolar hemorrhage associated with constitutional features. We thought she would have turned out to be the Copa syndrome, but even in the absence of autoantibodies, the diffuse alveolar hemorrhage, we'll probably call her an incomplete lupus or a non-autoimmune manifestation of a protein which is classically described with monogenic lupus. The next two cases are more like textbook pictures. The index case is 6 years old, with born of second degree consanguinity, symptomatic for the last 2 years, with fever, arthritis, dyspnea, severe MR, biopsy proven class 4 glomerulonephritis, lupus, and with classic ANA, dsDNA positive low complements, very strongly positive lupus anticoagulant and IgM ACL antibodies, which could explain the severe MR because it is a non-criteria manifestation of antiphospholipid syndrome. This family is being worked up with the help of Dr. Dipyaman at Ashoka University, and he told us that the sister is also homozygous for the same mutation. The parents and unaffected siblings are heterozygous for this mutation. There are 40 other cases described. What is important in the review of this literature is that these patients often have prominent urticaria, and they have not only the usual antibodies of lupus, but also the anti-neutrophilic cytoplasmic antibodies. So if by any path you see a child with urticaria and ANCA antibodies, and you're getting ANA, that's also another red flag. Now, we had a 12-year-old male patient who basically presented to us with severe autoimmune cytopenia. And at that point, we realized that there were syndromic features, there was short stature, there was a barrel-shaped chest, and he had a deletion in the ACP gene. The mother and father were heterozygous for the same mutation, they were consanguineous, the child is doing well on therapy. This was classic radiographs of the patient, so there was no doubt that this was, but the mutation was novel. And till date, published 29 patients of SPENST, but I'm sure there are many more in the wings. This mutation, as you all know, associates with Behçet's, but we saw it in a patient with classic lupus and refractory classic lupus, which means that this patient is not improving despite many immunosuppressives that we have given the patient. Now, do these mutations present as lupus? Yes, there are other reports, and I'm sure again many more will come. We had a child who came to us with basically ILD and had a severe hemolytic anemia, low complements, and ANA which was cytoplasmic. And so this was the CT scan of the patient. The patient was heterozygous for this mutation. We tried to do the interferon levels to see if this correlates with the interferonopathy which was described. We didn't find the elevated interferon levels. At that point, we were not doing the interferon genetic scores. And so we are not sure, but this was such a unique phenotype that we believe that if we complete the functional studies in this patient, we will prove its pathogenicity. I would like to basically bring home this point that in a group of patients where the interferon gene signature was recorded in this paper by Dr. Bello, if you can see that those genetic defects which are strongly associated with interneuropathy obviously have a very high genetic score, and then to your right, those which are associated with classic monogenic lupus have much lower interferon scores, and some of them even have normal scores. So basically, I think this makes us think of what prevents there are certain groups of genes which will directly cause interferonopathy with a classic phenotype, which I'm sure Dr. Amit will take you through, and then linked there is monogenic lupus. Those monogenic lupus which present non-classically will probably be somewhere in between these in their interferon scores. So I would like to end here because I'm sure there's a lot of interferon biology which is going to be discussed, and I'll also be listening eagerly to that. But monogenic lupus should be suspected in familial lupus of consanguineous parentage, especially if early onset. Even in adults, if there is a severe lupus with unusual manifestations like panniculitis, we had a patient with SOX1 where we haven't completed the functional studies, but if you have cytopenias, urticaria, incomplete lupus, help with a genetic diagnosis, try to bring it into the list of things that you would wish to do for the patient and get it done whenever possible, because going forward, as we go deeper into the understanding of these biologies, we will be able to probably provide them targeted therapy. But again, once the process starts, it goes up to a point when the pathways overlap, then targeted therapy may not help, and that is why if we have red flags in the clinic, we should do these things early, because these are chronic diseases and you have to take the patients through a lifetime of therapy. I would like to acknowledge Dr. Kirti and Dr. Kavita also in the audience, and a lot of other residents who have worked up these cases diligently and recorded that so that we can produce this information to all of you, as well as the journey with the Foundation of Primary Immunodeficiency Disorders, without which we would not even have thought of these in 2014, that we could do this even in an adult rheumatology clinic. This boy here in the front of the picture has been receiving support with FPID with monthly intravenous immunoglobulin injections for a diagnosis of XLA. Thank you very much.
Thank you, Professor Lisa, for wonderful description from the basics to the clinical presentation. If you have any question from the audience.
Anyone having any question? Ma'am, I have a question. Being an adult rheumatologist, so lupus is more of a phenotypic diagnosis with a positivity. So in recent times, there are upcoming reports of lupus-like phenotypic presentation in a case of VEXAS syndrome. So would you like to call VEXAS as a acquired monogenic lupus?
So, I wouldn't be the authority to speak on yes or no, but what I know where your question is coming from, and I've had the same doubts, because when you read publications which call themselves monogenic lupus, you will realize that even they are unsure of what they are calling as monogenic lupus. Because as an adult, we are trained to say that you will classify a patient as lupus if they fulfill all that. But short of that, even at the bedside, you will say, okay, this is lupus, even if I don't have the serology. So this kind of confusion in what we are terming as monogenic lupus is what you're also asking. So
Yeah,
I'll not say vexus is monogenic lupus, but I'll say that you will find lupus-like phenotype in many monogenic disorders, some of which could be somatic also.
So like even for the what all described for the monogenic lupus, as you also mentioned that many of the mutation doesn't satisfy the classical clinical criteria of lupus, and so do holds for the vexus syndrome also. So that was my thought whether it could be in future, because one patient reported from PGI only. We used to deal that patients. He had a NA positivity and cytopenia. So initially you had a diagnosis of lupus, then eventually it turned out to be vexus syndrome. There are upcoming reports, one more case reports in Oxford Rheumatology. So in that way, can it be an part of the extended spectrum of the adult part
Absolutely.
of the?
I mean, tomorrow you'll find another report of the full functional analysis of another gene, because so many are involved in even maintaining tolerance.
Dr. Sagar, please.
Thank you, ma'am. Thank you for the wonderful talk. I think the whole purpose of getting to a monogenic diagnosis sometimes is to offer a targeted therapy, and that is how the outcomes can be different, as you rightly mentioned. I would like to know your experience on plasma infusions, especially in C1q deficiency. We have had siblings who have been put on plasma infusions monthly, the fresh frozen plasma, because it kind of replenishes C1q, and their phenotype seems to get better. So, any comments on that, ma'am?
So the experience which we had with the long-term treatment of the C1QA patient, some of these patients are lost to follow-up. The one where we had experience, we kept on pushing with standard therapy for lupus, which seemed to work for most periods of time. And so we didn't feel the need to give her FFP separately. But if you don't have the full-blown lupus and you have recurrent infections, maybe, but I have no personal experience.
So, any, Devas, you want to make comment?
Good morning, ma'am. Thank you for the nice talk. Ma'am, your experience on the use of JAK inhibitors in your cohort, ma'am.
So actually I didn't show the patient because of two reasons. We have say a 25-year-old lady who has a lot of paniculitis in her lupus, and then she's losing weight and becoming very, very low BMI. At that point, and because we had got confidence in monogenic lupus ability, so we did a workup and we found that she had a SOX1 mutation, but it was not present in her parents. And so we're not sure that that is causing everything. But because it was there, we gave her JAK inhibitors, but it didn't help. And so we wondered whether if we had done, or if we were able to do that workup 15 years or 20 years ago when she started her disease, at that point if we had offered her JAK, would it have helped? We don't know. Because once these pathways are activated, then the entire system gets activated, and then you may not be able to break the cycle. That's my understanding.
Thank you, Dr.
Thank
for
you.
wonderful talk. And we go on to the next session.
Thank you, ma'am. Thank you, ma'am. Up next, I would. Thank you, ma'am.
Professor Amit Rawat is known to the audience, already being introduced yesterday. Dr. Rawat is from PGI Chandigarh. He is the backbone of the PGI immunological lab, because he is behind the lab and he's giving service to all over the India to this on immunological service in pediatric, and he is the founder member of this society also. I request Professor Amit Rawat to deliver his talk on. So, Dr. Professor.
Thank you,
enteropathies.
Dr. Tapas, for your kind introduction. And the topic I would be delving on is type 1 interferonopathies experienced from India. So where does the term interferon comes from? So I want to take you back into history. And the first description of something which was causing virus interference or interferon was described as early as 1957 by Isaacs and Lindemann. This was published in the Royal Proceedings of the London in Proceedings of Biological Sciences, and what they found was that if you put heat inactivated influenza on a chorioallantoic membrane, then and incubate it overnight, there was a secretion of a substance which, when you transferred it to a fresh chorioallantoic membrane and then you add a live virus, there was no replication. So there was a substance which was secreted which was causing interference in the viral replication, and there's the term interferon because it interferes with viral replication. So interferons, there are mainly three types. What the two main types described here are the type 1 and type 2, and what we are more concerned today are the type 1 interferons, which is a large family of genes. The type 1 alpha has got many members, almost 13 members. Then we have interferon beta, epsilon, kappa, and omega. These are all type 1 interferons. And then we have got type 3 interferons, which are mainly the interferon lambdas. And they have got their receptors. The IFN alpha has got IFN AR1, AR2, and the IFN type 3 interferon. They have got the receptor for that is the IFN LR1 and the IL-10 receptor beta 1. So what is the the main importance of these interferons is that they've got this potent antiviral properties. So when a cell is infected by virions, the virus uses the replication machinery of the host to produce new virulent particles. However, it also induces the production of these type 1 interferons, which then bind to other cells, and then they signal the nucleus to produce these antiviral proteins, which then curtail the further replication of these viruses in other cells. However, why is this type 1 interferonopathies? Because it's something where this antiviral immunity has gone berserk, and these are rare monogenic immune disorders where the main pathogenic mechanism is a persistent type 1 interferon activation, and they have been identified as a unified disease entity, although they affect multiple pathways in the last decades, and they can result from multiple defects like nucleic acid sensing and metabolism, interferon regulatory pathways, and others. So there's aberrant sensing of self-nucleic acids, activation of innate immune sensors, leading to a unbridled type 1 interferon activation, mainly interferon alpha, but also, and this then leads to systemic inflammation. It can lead to neuroinflammation and vasculopathy. So why are these interferonopathies important? They are usually present very early in life with a multi-system disease. They have got diverse etiologies converging, involving multiple pathways and converging on one common pathogenic mechanism, which is the type 1 interferon. And as has been highlighted by the previous speaker, if they can be identified, there are targeted therapies which are available for these patients. The clinical challenge, although remains that there are diverse phenotypes, but they have a common shared biology that is of interferon activation. So this cartoon here depicts the different pathways which are involved, and as you can see, most of the genes, they are involved in the nucleic acid metabolism, where you've got genes like RNase H2A, H2B, H2C, and then you've got other genes like TREX1, which is classically associated with Aicardi-Gouttiere syndrome. You've got SAMHD1 and others, DNase2. There are some genes which are also involved in DNA repair, like your BLIM, ATM, DCLRE1C. And then you've got some others which are involved in nucleic acids signaling, like the STING1 and the COPA. And then some genes associated with mitochondrial integrity. Other genes involved in post-interferon signaling, like the gain of function in STAT1 and JAK1, or the loss of function in ISG15 or STAT2. And then you've got others like which involves the proteasome machinery. So this figure highlights the different pathways. So there is accumulation of self-DNA or RNA because of the defects in nucleic acid metabolism. There is a aberrant nucleic acid signaling. mainly through TLR7 and leading to activation and then persistent type 1 interferon signaling leading to diverse clinical manifestations like your chilblain lupus, CNS involvement, lupus nephritis, or immune complex deposition. So these are diverse defects in nucleic acid metabolism and sensing which converge on type 1 interferon signaling, and some of them can present with an early lupus-like phenotype. So clinically, when you suspect or suspect type 1 interferonopathy, you've got recurrent fever, systemic inflammation. There can be CNS manifestation mainly in the form of basal ganglia calcification, leukoencephalopathy. Skin lesions are very common, like your villaceous skin lesion, chilblain lesions. Some of these can present with interstitial lung disease. You've got joint contractures. And what you can do clinically is that you can measure the type 1 interferons. Conventional ELISA is not a very good method to measure these interferons. This is of no use. You have to use a single molecule ELISA or a digital ELISA to measure them, or you can do an indirect measurement of interferon signaling either by RNA quantification or flow cytometry, and followed by that you can do a genetic sequencing. But however, you should be mindful that some of these variants in these genes could be somatic and which could be missed. In terms of treatment, I'll just be briefly alluding to, there are a lot of drugs now available. Most of us are familiar with the JAK inhibitors, which are widely used, but then there are others. There are some reports of the use of reverse transcriptase inhibitors in these patients. You could directly block the type 1 interferon with cefalumab or rontelizumab, or you could block the IFN interferon alpha receptor 1 with anifrolumab, or in some patients even hematopoietic stem cell transplantations would be useful. So we are very familiar with using JAK inhibitors, but then there would be other conditions where a STING inhibitor or even a TLR7 inhibitor or inhibitors of ER stress might be useful. And so apart from JAK inhibitors, there are other drugs which could be useful in a given situation. So this cartoon again depicts the approaches. So you could either use RNase FC for removing the RNA containing immune complexes which drive this type 1 interferon pathway. You could use this binding protein. You could eliminate the long-lived plasma cells. And the heart of this is the plasmacytoid dendritic cells, which is the main producer of type 1 interferon. You could block that with anti-BDCA2, which is a specific marker for plasmacytoid dendritic cells. You could use a TLR7 inhibitor or IREC4 inhibitors, or you could block the interferon signaling either by blocking the type 1 interferon alpha receptor 1 or JAK or TYK2 inhibitors. And this cartoon depicts the same thing, so I think I will not delve more into that. In terms of the assays which are available for diagnosing type 1 interferons, so as I've said that ELISAs are available, although the most useful test would be a SIMOA or the single molecule assay which can detect it, or you could do an indirect, what you look at is the interferon stimulated genes or ISGs, and this you can look by different genetic tests, like you could look at by microarray, you could do it by a real-time PCR, a nanostring, you can use nanostring to do that, or you can use RNA-seq. Flow cytometry would be perhaps the most simple tool to detect an elevated type 1 interferon signature, and what you can do is look at the CD169, which is Siglec-1 molecule on monocytes, which is a very easy readout, or you could also do functional assays like cell-based reporter assays. Most cell lines are the THP1 cells, where you incubate the cell line with the patient's serum or plasma, and then you look at the different genes which are upregulated in that cell line. You could use a cytopathic effect assay or a plaque reduction assay to do that as well. So these are some of the different assays available. So most labs would either use flow cytometry, which is readily available. You'd need only two markers, a CD14, which is for monocytes, and 169, which is Siglec-1 for a type 1 interferon, or you could do a gene expression analysis. And again, to highlight that these interferons are three types: type 1, type 2, type 3, And although they have got separate receptor, type 2 or the interferon gamma is the prototypic type 2 interferon, and this is involved more in Mendelian susceptibility to mycobacterial diseases. But they converge on the very similar STAT molecules and JAK molecules. So if you're trying to look at the type 1 interferon pathway by something which is downstream of this, you should be mindful that what you would be estimating might not be truly type 1 interferon, it could be type 2 and type 3 as well. So in our lab, what we are doing is we are looking at the type 1 interferon signature by these 6 genes, which are the interferon stimulatory genes, the interferon stimulated genes, and 2 housekeeping genes. So this was done in collaboration with the Indo-Italian project where I was a PI, and the gene sequence of all these genes was cloned into a plasmid vector, and then the plasmid was diluted to obtain a standard curve, and then real-time PCR was done to calculate the IFN scores using the CT values. So this was the construct. It contained the 6 ISD genes, 2 housekeeping genes. It was cloned into a plasmid, and then the plasmid was serially diluted to get a standard curve. So this is the standard curve you get for the type 1 interferon signature using the 6 genes and the housekeeping genes. And over last maybe 5-6 years, we have been doing this type 1 interferon signatures in a lot of patients with suspected type 1 interferonopathies or monogenic lupus, where the clinicians have a high suspicion of a monogenic lupus. The total number of patients was 357, and we found an elevated type 1 interferon signature in 150 of them, and almost half of them had a genetic defect, making to a quarter of them had a genetic defects. So these are the different pathways where we found defects in these patients. So most of them were in the nucleic acid metabolism and clearance pathway, where the number was 31, and the different genes which were involved were TREX1, RNase 2B, 2C, DNase 2, DNase 1, L3, as Dr. Lisa had highlighted in one of her cases. STING1 we found in a family in 3 patients, IHI1, one patient with TLR3, then a lot of patients with complement deficiency. So early complement deficiency in C1q, C1qa, B, and C also results in an elevated interferon score, and the exact mechanisms of which is not clear. And then we've got other genes involving immune dysregulation or DNA damage and other pathways. So this was the breakup of the different genes involved in the type 1 interferonopathies in our experience. So when we looked at the interferon score in terms of the different pathways that are involved, we can see that the nucleic acid metabolism pathway, the score was the highest, followed by the complement pathway, and then different other pathways. And what we also did was that in the 79 patients, there were some patients where VUSs were reported in these different genes. And when we looked at the type 1 interferon scores, we were able to upgrade some of these VUSs into likely pathogenic. So a significant proportion could be upgraded to a likely pathogenic, and others which when we performed the type 1 interferon score, which was not elevated in these patients, they either remained VUSs or we could downgrade them to likely benign. So I'll just quickly move through some case vignettes. So case 1, this was an 8.5-year-old girl who was referred to us from Kashmir. She had oral ulcers, hematuria. She was initially admitted in hematology with the diagnosis of Ivan syndrome, with anemia and thrombocytopenia. The ANA was 3+ homogeneous, C3 and C4 were normal. Because of the low platelet, she had ecchymosis, epistaxis, upper GI bleed. In addition, she also had failure to thrive, short stature, global developmental delay, spastic diplegia, and she had cytopenias as well. This is the family tree. She was born of a consanguineous marriage, and when we looked at the family history, the 2-year-old younger brother also had failure to thrive, global developmental delay, spasticity, although he did not have the autoimmune hemolytic anemia or the thrombocytopenia. And when we did first we performed the NGS, and there was a large deletion in the ACP, homozygous deletion in the ACP5 gene, and and this is a figure, the X-ray of the spine to show the platyspondyli, and the type 1 interferon score was again elevated in this patient. Case 2 was a young girl who presented with contractures of the knee, the ankle, the wrist, with a claw hand. This is the picture of the father, and this is the child herself who is showing full-blown interstitial lung disease. Laboratory investigations, she had anemia, neutrophilic leukocytosis, ESR was elevated, so was the CRP, rheumatoid factor was also elevated, ANA was 4 plus diffuse, the complements were normal, and when we looked at the type 1 interferon score in the child and the father, they were both elevated. And this girl had SAVI, I'm sorry, this was SAVI, and the defect was in the STING1 gene. It is a heterozygous variant, and the brother of the index patient was also affected. So this is the index patient, this is the brother, this is the father. So the 3 members in the family were affected with this defect. Case 3 is a 6-year-old boy who presented with fever, erythematous maculopapular rash, pain and swelling of bilateral ankle and knees and wrist joints, delayed development of milestones in infancy. On examination, he had pallor, clubbing, arthritis of left knee, bilateral knee joints, and tarsophalangeal joints. This was the X-ray. This was the MRI which showed intensities in frontoparietal and temporal regions, and there was also some attenuation in the vertebral arteries. And this child had a mutation, homozygous mutation in DNase 2, which is involved in DNA metabolism. This was a homozygous variant. And again, in this child, the type 1 interferon score was highly elevated compared to was elevated in the patient compared to control. Case 4 is a recent case that we saw. This is a 1.5-year-old girl who was referred to us from pediatric neurology. She had global developmental delay, no social smile, no eye contact or verbal communication, partial neck holding, and she could not sit with support at 1.5 years of age. She had seizures on first day of life, macrocephaly, hypertonia. She also had dystonia. On MRI, I don't have the images for this child. There was diffuse cerebral atrophy with paucity of white matter. There were multiple areas of calcification in the basal ganglia. She was born of a consanguineous marriage. 2 elder brothers were normal. And the differential diagnosis that they kept was the TOS sequelae or Aicardi-Goutieres syndrome. And since the phenotype was so clear, we went ahead with a targeted interferonopathy, which we do in-house. And this girl had a mutation in the RNase H2C gene, which this variant has already been reported, and so this was a likely pathogenic variant in this girl. Just one more case of COPA syndrome, where there is mutation in the COPA1 gene. This is also an autosomal dominant disorder. So the genetic etiology of monogenic lupus in our cohort, most of them are defects in the early complement pathway, most of them C1q, C1qa, some with C1qb, some with C1qc. We also have a couple of patients with C1r, and then there are defects in type 1 interferonopathies with DNase L13, DNase 2, ACP5, STING1, and a few patients with TREX1 and ADR gene as well. And when we look at the pathways, the complement deficiency is by far the most commonest, comprising around 60%, and then we have type 1 interferonopathies in another third, and then there are other diverse mechanisms which can lead to monogenic lupus. So this is to show that you could do a Siglec-1 easily by flow cytometry in a patient where you have a suspected phenotype of a type 1 interferonopathy. So you need CD19, CD14 for monocytes and CD169. So I'll just skip through this. So when we tried to look at the clinical phenotype across different scores, as the score was higher, the number of manifestations and the severity of manifestations was increasing. So this type 1 interferon score could be a useful functional marker, especially where you have VUSs and you are not sure, or the phenotype is not very clear. And again, just to summarize, when the clinical picture is very characteristic, like you've got intracranial calcification, neuromotor impairment, a paniculitis, or a Chilblain lupus, like phenotype of interstitial lung disease, arthritis or monocytes, then you can look at the family history. You look at the type 1 interferon signature. The type 1 interferon signature is useful, but you have to be mindful that the type 1 interferon signature can be elevated in other diseases as well. So JDM, juvenile dermatomyositis, is one disease where there's a elevated type 1 interferon signature. In otherwise, lupus also, around 80% of lupus also have a type 1 interferon, elevated type 1 interferon signature. So it is sensitive but not specific, because it could be a high interferon signature may be seen in other forms of lupus which are not monogenic, JDM, and also viral infections. And you can then proceed to a whole exome sequencing or a whole genome sequencing for a genetic based diagnosis. So with that, I would like to acknowledge the international collaborators, Dr. Marco Gattano, Dr. Angelo Ravelli, and Stefano Volpi for the support during the Indo-Italian project, grant from DST and DST SERB, national collaborators, patients and their families, and Dr. Madhubala Sharma, my PhD student. She did this work on the type 1 interferon gene signature as a part of a thesis. Thank you very much.
Thank you, Dr. Amit, for a wonderful and extensive talk on internopathies. This shortage of time, if there is any question, then.
Any one question? Sir.
It doesn't get elevated in lupus, for example, in type 1 interferonopathy, but a lot of these interferonopathies that you showed, sir, have got high CRP. Any explanation for that? That's number one. And just a brief comment, CD169 really helps. We have also set it up in Bangalore. It is an easy test to set up if you have a flow cytometry facility. Yes.
So conventionally, type 1 interferons, they will inhibit the production of, because they will inhibit the transcription of the genes which are involved in the in the liver for CRP synthesis. And usually in lupus, CRP is only elevated where you've got polyserositis or you've got infections. So, I mean, when you want to differentiate between activity and whether it is an infection, you go for a CRP. That it's usually true, but I'm not sure why in some of the other disorders the CRP is not muted as in SLE. I'm not sure about that.
Dr. Suparna.
I can't hear you properly.
Suppose I have a patient early onset lupus and I'm suspecting a problem in the interferon signature and my whole exome sequencing comes out to be normal. What should I do next? Like, she has all this difficult arthritis, she has lung manifestations, but the WS has turned out to be normal.
But you still could do a type 1 interferon signature, and if it is elevated, probably you could try some newer drugs.
It is elevated. It is elevated.
No, elevated mean how much elevated? I mean, like, you have to look at the magnitude of
Dr.
the elevation.
Suparna,
because
you make
even
it
usual
short,
lupus.
but the next talk is online talk.
So even in usual lupus, the type 1 interferon signature might be elevated. So you have to look at the magnitude of the, and if she is not responding to your conventional, you could probably think of
Thank you,
changing
Dr. Rawat,
therapies.
for the comment, and
Thank
thank
you.
you for the wonderful talk, because we don't have time to take it more. The next talk is online talk. Thank you both the speakers for wonderful and extensive talk, and thank to the audience for patience hearing. Thank you all.
Thank you, sir. Next, we move on to Professor Sudhir Gupta oration on anti-cytokine autoantibodies, new roles and recognition. The speaker will be Dr. Stephen Holland, who will join us online. And I would like to request Dr. Sudhir Gupta, Dr. Amit Rawat, and Dr. Vignesh to chair the session. And Dr. Sudhir Gupta will also be joining us online. Thank you. I would request Dr. Amit Rawat to say a few words on Sudhir Gupta. Thank you.
So, good morning, everyone, and warm welcome to all members and delegates for the fourth Professor Sudhir Gupta oration. And I have the honor and privilege to read Professor Sudhir Gupta's CV. So it runs into pages, and I've tried to be brief, but even then I'll try to be brief. So Dr. Sudhir Gupta graduated from the King George's Medical College, Lucknow in 1971. And then he moved on to the US in 1971 and served as a teaching faculty at the Tufts University School of Medicine, Boston, and a visiting fellow in the medicine at the College of Physicians and Surgeons, Columbia University, New York. He then joined the Memorial Sloan Kettering Cancer Center and Cornell University Medical College, New York, and soon rose to the rank of an assistant professor of medicine and immunology. In 1982, he moved to the West Coast and established a division of basic and clinical immunology at the University of California, Irvine, and in the process became the youngest professor of medicine and clinical immunology, one of the youngest professors of medicine and clinical immunology in the US. Dr. Gupta is board certified in clinical immunology by the Royal College of Physicians and Surgeons of Canada, both in allergy and immunology and diagnostic laboratory immunology by the American Boards of Allergy and Immunology. He is a fellow of the Royal College of Physicians and Surgeons of Canada, Royal Society of Medicine, London, American Academy of Allergy, Asthma and Immunology, the American College of Allergy, Asthma and Immunology, and he also happens to be the founding fellow of the American College of Rheumatology. His awards include Honorary Doctor of Science from his alma mater, KJMU, Lucknow, Arthur Mansell Award from the College of Physicians and Surgeons, Columbia University, a Lifetime Achievement Award from the Jeffrey Modell Foundation, New York, Most Distinguished Physician and Researcher and Teacher Award from the American Association of Physicians from India. The Board of Regents of American College of Physicians conferred upon Dr. Gupta its highest honor, the Mastership of the American College of Physicians. In 1986, Dr. Gupta, along with his wife, Professor Abha Gupta, were presented to Her Majesty the Queen Elizabeth and Prince Philip at a special ceremony of the Royal Society of Medicine, London. He also served as a scientific advisor to late Prime Minister Indira Gandhi. Dr. Gupta has served on several important and national international committees, including the committees of the Institutes of Health, FDA, and the Advisory Board of the WHO. And his name has adorned editorial and advisory boards of more than 35 scientific journals. And he was the founding editor of the Journal of Clinical Immunology and served as its editor-in-chief for 3 decades. He has made several fundamental contributions to clinical immunology. These include the ontogeny of human surface immunoglobulin, receptors on human eosinophils, potassium channels on human T cells and their roles in autoimmune diseases, gene mutations in CVID and hyper IgM syndrome, and he has published in high-impact journals including Nature, Science, PNS, and the NEGM. In recognition of his scientific contributions to immunology, the University of California, Irvine has established an endowed chair in its name, Professor Sudhir Gupta Endowed Chair in Molecular Immunology. This is the first endowed chair in the department at the University of Irvine. His association with India was in 2012 when he and his wife founded a not-for-profit and charitable foundation, the Foundation for Primary Immunodeficiency, the FPID, with a mission to support patients with early diagnosis, treatment, and research for primary immunodeficiency both in the US and in India. And the scientific advisory board of this foundation consists of Nobel laureates, 4 members of the National Academy of Sciences US and the Academy of Science. This foundation has been supporting early diagnosis for PID at multiple institutions in India. The foundation has been organizing and supporting the national and international conferences of primary immunodeficiency, which have held in India regularly. Dr. Gupta has also been working closely with the DG ICMR and the Secretary of Health for establishing for establishing 2 centers of excellence in PIDs, one at Chandigarh and at Mumbai. We are very grateful for him for having been mentoring us over 15 years, for holding our hands through this time, and for providing academic opportunities that would not be envisaged without his pioneering vision. Thank you, sir. Now I request Dr. Vignesh to read the oration of the citation of Professor Stephen Holland.
Thank you, Dr. Amit Rawat, and good morning, one and all. So it is indeed my honor and privilege to introduce Professor Stephen Holland to this esteemed gathering. So I also had the honor of observing Dr. Stephen Holland at National Institutes of Health doing clinical rounds as an amazing clinician scientist. So Dr. Holland is currently the Director of Division of Intramural Research at National Institute of Allergy and Infectious Diseases. He's the Chief of the Immunopathogenesis Section within the LCIM, the Laboratory of Clinical Immunology and Microbiology at NIH. Dr. Holland provides overall executive direction of the division's basic and clinical research activities, which cover infectious diseases, immunology, and allergy. He discovered the STAT3 mutation for hyper IgE syndrome and GATA2 deficiency for the monomac deficiency syndrome. He is the author of more than 800 publications and has been named as an NIH Distinguished Investigator. He is a member of the National Academy of Medicine and has received the American College of Physicians Award for the Science, the Boyle Scientific Achievement Award of the Immune Deficiency Foundation, the American Society for Microbiology Abbott Award, the Irwin Netter Award of the Association of Medical Laboratory Immunologists, the NIH Distinguished Clinical Teacher Award, and the Walter E. Stam Mentoring Award of the Infectious Disease Society of America, among other awards. So, Professor Holland, now the stage is yours. Thank you so much.
Since Dr. Stephen Holland could not make it today, so we would like to request Dr. Surjit Singh, sir, to please accept the customary shawl and the oration plaque. After the, okay, sir. Okay, so we would like to request Dr. Stephen to start the oration. Thank you.
Thank you very much. Can you hear me? Yes. All right. I'm genuinely sorry not to be there with you. It was my hope to be able to join, and I'm hoping that we can get the slides set up properly here. They worked so well yesterday. There we go. Is that in the right format? Very good. So I'm going to talk today about anti-cytokine antibodies. But I want to say that I had hoped to be there in person, hope to be there with you and with Sudhir, because it is really a great honor to be asked to give the Sudhir say after decades of spending
Dr. Steve, you are on the presenters mode actually. It's now we're good. Yeah.
Now it's okay.
Yeah, thank you.
So being able to work with Sudhir has been an extraordinary opportunity and gift. And as you all know and have heard, he's an extraordinary physician, academic, humanitarian, philanthropist, and it is an honor to be able to call him a friend. So I want to talk today a little bit about, of course, primary immune deficiencies are the essence of the meeting. But I want to talk about some of those that often don't get considered in the primary immune deficiencies. I think this is oversight. So in the United States, the current rate of primary immune deficiency is thought to be between 5 and 10 per 100,000. In contrast, in the United States, the rate of tuberculosis is around 3 per 100,000. So I tell Americans, if you are ever considering tuberculosis in a differential, which is pretty common, as a differential point, you really need to be considering a primary immune deficiency as well. In India, of course, the situation is very different. No one believes that the rate of primary immune deficiency in India is really as low as less than 0.5 per 100,000. That is simply an artifact to make the diagnosis, and it's clear from all of the work coming out of this symposium, all of the work that Sudhir has sponsored, that the rates are going to be quite similar, if not a bit higher in India than in the United States. And in contrast, the rate of tuberculosis is still very high. We know that rates of tuberculosis will come down. That is a problem that can be solved. And making sure that we get our rates of primary immune deficiency up where they belong, so that we are making the right diagnoses and the right treatments is essential. Now, the rates of primary immune deficiency are increasing around the world because, frankly, the recognition of primary immune deficiencies is increasing around the world. And this is largely driven by the cost of sequencing, which, as you can see here, has gone down dramatically over the last several decades. And the cost of sequencing is getting into the range where it will be easily affordable by almost everyone very soon. Getting the analysis done, that's still going to be a little bit of a trial, but I'm sure we'll get there soon. But one of the things that has driven the increase in the number of primary immune deficiencies is the recognition that there are both somatic mutations, as was mentioned just a moment ago, as well as autoantibodies. And I want to talk just briefly about a couple of autoantibodies that I think are important. Now, I'll be honest, I'm talking about these antibodies because these are the ones I know the most about, but they're also ones that I think are relatively common and are probably worth thinking about. So, anti-cytokine autoantibodies mimic Mendelian defects. We're going to talk a little about anti-interferon gamma, which is widely thought to be a mimic for the MSMD phenotypes, about the anti-IL-23 antibodies, which are somewhat similar and very common in the setting of thymoma, and then about anti-GM-CSF autoantibodies that are really sort of interesting and I think not widely appreciated by most internists. Anti-interferon alpha autoantibodies have gotten lots of discussion in the context of COVID and several other conditions like influenza and yellow fever vaccine, and I'm not going to discuss those today. So, despite the fact that Mycobacterium tuberculosis was discovered in 1882, or at least was announced in 1882 in the kitchen of Robert Koch, shown here, after having had a particularly disagreeable meal, it was not recognized, we've not understood what really causes it. And over the 140 years since that time, many other mycobacteria have been recognized, most of which cause disease infrequently and require more immune compromise than is typically required for people to become affected by tuberculosis. And I'm going to refer collectively to these mycobacteria in this group here as the non-tuberculous mycobacteria. Oh, my apology. And we're going to talk about these in the aggregate as the non-tuberculous mycobacteria. Now, non-tuberculous mycobacteria typically come in three different varieties. There is isolated skin infection, which is mostly about exposure to water. There is isolated lung disease, which is associated with cystic fibrosis and ciliary defects. And then the least common form of mycobacterial infection is disseminated disease, which tends to be severe and in young people. And you can see in this poor boy, he had really profound hepatosplenomegaly due to an IL-12 receptor defect. And this has led to the recognition that there's really one major pathway for mycobacterial control. There are multiple genes, more than are shown here, but mostly they are around the synthesis and response to interferon gamma. And you see interferon gamma activating its receptor, leading to the killing of mycobacteria, as well as the production of IL-12 in a very effective positive feedback loop. These diseases are typically referred to as Mendelian susceptibility to mycobacterial disease,
which is a useful way to think about them, but very important to keep in mind that there are more infections in this than mycobacteria. And while all of the mycobacteria that you've heard of and many that you haven't are associated with defects in this pathway, so are gram-negative rods like Salmonella and Burkholderia, gram-positive rods like Listeria, fungi like Talaromyces, which I'll come back to, Histoplasma and others, and viruses. So this pathway does not discriminate in terms of being susceptibility for mycobacteria alone. And I think gets missed in some of the cases where other organisms are the presentation. The other thing to keep in mind is that because these are genetic disorders, this MSMD pathway, they typically, not always, show up in childhood. And they certainly are exacerbated by the administration of BCG vaccine. But I'm an internist. And so I start out seeing adults, and this woman came to us quite a while ago. She had grown up in Vietnam during the war. She had left there and come out through the refugee camps and came to the United States in her 20s, and she was a very healthy person. She had a job, she had children, no problems, and then at age 60 developed this problem here that you see as osteomyelitis, that is breaking through her chest and discharging pus that is full of M. avium complex. And her problem was that she had anti-interferon gamma autoantibodies. And as you see here, these antibodies fully block interferon gamma, prevent it from signaling at all, and therefore completely phenocopy mutations in the interferon gamma receptor. Well, at the time we were seeing her, it was recognized that there were other people like this, and this was in the early 2000s. And here is a case report from Germany in 2004, reporting a woman who had disseminated M. colonnae and Burkholderia gladioli sepsis that was fatal. But she was a Thai woman identified in Germany. Here, a 57-year-old Filipino man living in England who had tuberculosis, M. colonnae, and autoimmunity. And then 6 Southeast Asian women, the one I showed you was an example of that, all of whom lived in the United States, all of whom had disseminated and severe disease, all of whom were from Southeast Asia. So the question then was, if this is what happens to Southeast Asians outside of Southeast Asia, who have IgG antibodies against interferon gamma, what was happening in Southeast Asia itself? What was going on there that might be relevant to understanding the disease of people outside of Southeast Asia? And a gifted Thai infectious disease doctor, Plongchan Chatsoksat, had been studying patients that did not have HIV who did have disseminated opportunistic infections, Salmonella, Cryptococcus, Talaromyces, Histo, and so on. And she identified these patients in Thailand, confirmed that they didn't have HIV, and she wondered what was going on. And we were lucky to be able to collaborate with her to identify that indeed these people also had these anti-interferon gamma autoantibodies. And here were examples of what they looked like. This man had disseminated Talaromyces and Mycobacterium fortuitum. This woman had disseminated Salmonella involving her lymph nodes. And these autoantibodies really fully mimicked the MSMD phenotype. But, you know, again, just like in the mutations, the disease that's caused by these antibodies is not just about mycobacteria. And here I've simply highlighted in all of the red asterisks, each of these is a fungal infection attributed to these autoantibodies. And you can see there are quite a few of them. And in particular, I want to draw your attention to Talaromyces marneffii. Now, if you are in southern China or Vietnam or Laos, then the overwhelming likelihood of a Talaromyces marneffii disseminated infection is that it's due to these anti-interferon gamma autoantibodies. But not only are these Tellermyces marneffii infections common in Southeast Asia and southern China? They're also common in this part of India, in northeastern India, around Manipur. And although I've seen case reports of HIV-associated infections with Tellermyces marneffii in Manipur, I've not seen from India any reports of anti-interferon gamma autoantibodies. And I would love to hear more about that. We're certainly happy to collaborate and help in it, or just to enjoy learning more about it. But I would be thinking that Manipur would be a good place to start looking for causes of disseminated Tellermyces marneffii. All right, so why are these things happening? Well, we think that it is HLA related, and that makes a lot of sense. If it's regional and ethnic, then HLA is one of those things that is regional and ethnic. And there are two HLA alleles, DRB1 1602 and DQB1 0502, that are associated with disseminated disease at a fairly high level. And you can see the p-values here, which are good, not diagnostic, but pretty persuasive. And it turns out that these HLA alleles encode binding pockets that recognize the terminal 10 amino acids of the interferon gamma molecule, as you see here. Interestingly enough, those same 10 terminal amino acids are reproduced in certain proteins in Aspergillus, Aspergillus terreus, flavus, nigilans. And those amino acid stretches are able from Aspergillus to bind to these HLA pockets and lead to the production of antibodies. And so the hypothesis, and I think a very, very compelling hypothesis, is that people can be exposed to Aspergillus. They inhale that or ingest it. They then produce antibodies against the protein in Aspergillus called NOC2. Those antibodies then cross-react with interferon gamma and lead to antibodies that are fully neutralizing to interferon gamma and lead to disseminated disease. And the authors of the paper describing this then went to delete the epitope from interferon gamma and were able to restore function again, suggesting that this epitope was really important and that it was the cross-reactivity with Aspergillus that might have been the genesis of it. I think this is a strong hypothesis. It's not the only possibility, but one certainly to know about. Well, okay, once you know that the antibodies are the problem, well, we're pretty good at how to handle antibodies these days. Anti-CD20 therapy or rituximab or any of the other drugs that do essentially the same thing can reduce the titer of interferon gamma autoantibodies and lead to remission of disease. As you see here, the titers go down, people remit. This woman, we stopped her therapy. She relapsed. Her antibody levels came up. We treated her again, and she went right back into remission. So we're pretty confident that these antibodies are, in fact, the effectors and that getting rid of the antibodies helps. But like most things, it doesn't work in every setting. And when we've had people who did not respond to the B-cell therapy, we went after plasma cells. And we've used bortezomib, which has been effective. You can see this woman's abscess going from this to this over several months. But there are some people who either don't tolerate bortezomib or did not respond. And in those cases, we've used daratumumab, which is an anti-plasma cell agent targeting CD138. And so these are approaches that can be used when rituximab is unsuccessful. In addition, there are excellent experiences using cyclophosphamide, which is certainly highly effective, much less expensive, and there are decades of experience using it for antibody therapies. And I keep on thinking, though I do not have any experience, that efgartigimod, the FcRn blocker, should be interesting to use in this disease in terms of shedding pathologic antibodies. So these antibodies, they're bad. You'd like not to have them. Treating the antibodies will make the disease get better faster in people who are affected. But how do you make the diagnosis? And I don't know exactly what the technique is that's being used in India now to identify tuberculosis exposure. But we've moved now in the United States and Taiwan to the use of the quantiferon assay. It's a 4-tube bioassay in which blood is collected and stimulated, and then you look at the production of interferon gamma in the supernatant. And let me just explain the assay for a minute because it comes in handy. When you have these 4 tubes, the first tube has no agonist, the second tube has an antigen, the next tube has a different TB antigen, and then the fourth tube has a mitogen. And you need the mitogen tube to be sure that the cells were alive when they were collected and shipped, and that the cells were able to produce interferon gamma in response to a mitogen so that you can interpret their lack of response or their positive response to an antigen. Well, in the setting where you don't have tuberculosis, the antigen responses are negative and the mitogen response is positive. When you do have tuberculosis, the antigen response is positive and the mitogen response is positive. But when you can't detect the mitogen response and you can't detect the antigen response, that's called an indeterminate quantiferon. And what's happening there is that the anti-interferon gamma autoantibodies are binding to the interferon gamma itself, preventing them from being detected in the ELISA and giving a negative reaction in the mitogen tube and every other tube and leading it to be called indeterminate. Well, if you have a patient who has autoantibodies to interferon gamma, they almost always have a quantiferon assay that's indeterminate. But what about in the opposite direction? If you take people who have indeterminate quantiferon assays, how often do they have blocking antibodies? Because there are a lot of ways to get an indeterminate quantiferon. The cells can be freezed, they can be baked, they can be on steroids, they can be on other immunomodulators. How do you know what an indeterminate means? Well, we teamed up with a large testing company here in the United States and collected 850 samples that were blinded to us. So I have no idea from whom they came. I have no idea whether they're male, female, Asian, Caucasian. But when we took an unselected population of quantiferon assays and we took only those that were indeterminate, 1.2% of the indeterminates had fully blocking antibodies to interferon gamma. So I just mention this to remind you that if you get an indeterminate quantiferon assay and you're wondering why would that happen, anti-interferon gamma autoantibodies are one important reason to consider. So I'm going to stop talking about this now. It's a fascinating disease, high titer neutralizing antibodies, they're IgG. There is a strong gender predisposition. That is, in Southeast Asia, the male to female ratio is about equal at 6-4. But once people leave Southeast Asia, the ratio changes, and the female to male ratio is about 9-1. There are multiple opportunistic infections here, not just mycobacteria, strong HLA association. And when people get outside of Southeast Asia, it's almost exclusively women who get sick. And there are ways to treat it. All right, I want to move on in the same pathway and just mention this case that started out with a woman from Cambodia who had 4 years of Burkholderia gladioli bacteremia. Burkholderia gladioli is only seen in cystic fibrosis and chronic granulomatous disease, so we were very intrigued that she had these disease. And this is a lymph node excised after 4 years of intravenous meropenem therapy. And that is just frank pus. And you can see here in this picture that there were these branching filamentous forms inside her macrophages, and that is the Burkholderia. She was not killing this at all. And we identified that she had an anti-IL-12 P70 autoantibody. But it wasn't until a few years later that Aristine Chang, a gifted investigator in Taiwan, came into the lab and said, no, 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 I don't think that's it. And she persuaded me that, in fact, what was going on was that her antibody was to the P40 subunit, and that she was neutralizing IL-12, yes, but the really important point was that she was neutralizing IL-23. And it was important because IL-23 neutralization was leading to this syndrome. And the reason that the IL-23 is so important is that IL-23 is required for the development of mate cells, the mucosal associated invariant T cells. And in the absence of these cells, people are really at high risk for a variety of infectious complications. And over a series of really elegant experiments, Aristine showed that these patients got a variety of infections very similar to what we saw with the anti-interferon gamma antibodies. And in particular, we found a lot of it in thymoma. And in fact, if you looked at patients with thymoma, if they had severe infections, over 80% had autoantibodies to IL-23. And you can see here, this is our group with thymoma. These are their titers of anti-IL-23 autoantibodies. So if you have patients with thymoma and if they have infections, please think about this autoantibody. They're really quite interesting, and they are involved in a whole variety of infections. And they can be associated with other autoantibodies, excuse me, and are very common in thymoma. The last antibody I want to talk about is the anti-GM-CSF autoantibody. And this was one of the very first anti-cytokine autoantibodies described in the 1980s because it was associated with pulmonary alveolar proteinosis. And PAP, which you see here, is associated with this crazy paving pattern on the chest CT. You can see these little septations here that are characteristic of pulmonary alveolar proteinosis. And you see how there are affected areas and unaffected areas. And on biopsy, you typically see these cholesterol clefts where the alveolar space has been crowded out by macrophages that are chock full of surfactant and lipids that they've ingested. And then the lipids get centralized, and in the process of fixation, they get washed out, and you get those cholesterol clefts. That's pulmonary alveolar proteinosis. But GM-CSF is a funny cytokine. It's made by a vast number of cells, as shown here, and it acts on a large number of cells. That is, unlike interferon gamma or IL-23, which are pretty well circumscribed in the immune system, GM-CSF has many effects in many systems outside of the immune system, as you see here, endothelium, mast cells, and epithelial cells. Well, in the process of exploring cases in Thailand, we identified patients who had disseminated cryptococcal disease who had these anti-GM-CSF autoantibodies. And when we saw that, we came back to the NIH and we went to Jack Bennett, one of the founding authors of the Mandel, Dolan and Bennett textbook in infectious diseases. And we said, Jack, do you have any spinal fluids from people with cryptococcal meningitis? And working with Jun Kwan Chung, shown here on the left, we pulled out a variety of CSFs from the freezer. And lo and behold, about 10% of those CSFs had anti-GM-CSF autoantibodies. Fascinating, though, almost all of them were in the cases that had Cryptococcus gattii. And for those of you who are too wise to have gone into infectious disease, you want to know that there are two forms of Cryptococcus, gattii and neoformans. Neoformans is what you see in HIV. Gattii is what you see in the non-HIV host, the, quote, immunocompetent people. And those were the ones associated with GM-CSF. And then shortly after that, we encountered a very healthy, active man who had had a seizure while driving in the countryside. And he went into the hospital where they saw this lesion in his brain. They thought it was cancer, but when they biopsied it, it was pus, and it grew nocardia. And that got us interested in the possibility that nocardia infections could be part of this syndrome because he also had anti-GM-CSF autoantibodies. Now, so I've told you that these antibodies are associated with alveolar proteinosis, cryptococcal meningitis, and severe nocardia. Is there anything different about these antibodies between these different sets of patients? And the simple answer is no, not really. We can't see a big change between the antibodies in these different groups. And in fact, over time, people who show up with infections can develop PAP, and people that have the PAP can go on to develop infections. And just show you two examples here. This man, the one that had the brain abscess, had been in the military, and so we were able to collect his plasma going back decades. And what you see here is that it was 6 years between, 13 years before he developed the antibody, and that antibody went on for 6 years before he developed the CNS nocardia, and then it was 6 years after that, then he finally developed the pulmonary alveolar proteinosis. In addition, although the titer, that is the binding ability of his antibody, went up and down over time, its ability to neutralize stayed roughly the same. That is, the blocking activity of the antibody was not adequately reflected in its binding activity. So these anti-GM-CSF autoantibodies are associated with both pulmonary alveolar proteinosis and bad infections, in particular cryptococcus and nocardia. The infections can present without the proteinosis, but it often develops later. There are also GM-CSF autoantibodies present in Crohn's disease, but not typically associated with PAP and not typically associated with infections. These may be responsive to GM-CSF treatment, and treatment to try and eliminate the antibodies, say with rituximab or daratumumab, tend not to be as effective as in patients with anti-interferon gamma disease. All right, well, I'm almost done here. And I'm just going to point out that there are lots of ways that antibodies happen. And that's still a bit of a mystery. What makes them come on? Why would they begin if they were not there in the first place? And so infection itself, that is the cataclysmic stimulation of the immune system, can be part of the trigger to develop autoantibodies. And so in really, I think, a seminal paper from P.J. Utz's group in California, early in COVID, they looked at people who were sick with COVID-19 and were hospitalized. And what you see here is that in patients who had COVID disease, they had many more autoantibodies than healthy controls or patients with other autoimmune diseases. And in particular, what was the group that these patients had high titer of antibodies to? Interleukins and other cytokines. Well, that was interesting and certainly goes along with the observation that anti-interferon alpha autoantibodies in COVID were associated with severe disease. But then the question really ought to come up, well, is that because it's COVID or because it's just bad infection? And so he went on several years later to ask the same question to people who did not have COVID but did have severe enough respiratory infections to get them into the intensive care unit. And once again, you see here, these are the patients in the intensive care unit, people who were in the ICU and didn't have infections, and then healthy controls. And you see here that interferons and interleukins are much more highly represented in those who have severe disease from other respiratory viruses than COVID, and more here than in patients who had ICU hospitalization without infections. So these suggest that stimulation of the immune response by severe infection may trigger the development of autoantibodies. and may contribute to some of the things that we see. Now, not all development of autoantibodies has to be bad. We don't know whether some of these might have ameliorative effects and others have detrimental effects. I can also say that most of these antibodies are not neutralizing. So exactly what they mean from a functional standpoint is not so clear. Okay, well, you have survived. I wish I were there to survive with you and to be able to spend time talking about some of the exciting science that I know you're hearing and that I wish I could share. I've tried to talk about anti-interferon gamma and anti-IL-23 autoantibodies that are associated with multiple opportunistic infections, as well as anti-GM-CSF autoantibodies that cause pulmonary alveolar proteinosis, cryptococcal disease, as well as nocardia. There are many other autoantibodies. I mentioned the anti-interferon alpha, but there are others directed against many other cytokines as well. And those will be discussed in more detail in the years ahead as we develop the cohorts and figure out exactly what are the immune consequences of these autoantibodies. So I just conclude by saying that these are acquired innate and adaptive immune defects. They are dynamic. They can have on-target and off-target effects, and they can be treated. And I am quite confident that they affect more diseases than we think. And I really want to know what's going on in Manipur, and I hope you will tell me about it. Many other antibodies yet to come, many that have been described already: anti-erythropoietin, anti-GCSF, osteoprotegrin, IL-17, lots and lots and lots going on. And these are certainly having effects on clinical manifestations in patients that we have not yet described. So I want to just acknowledge people who have done this work, in particular in my own group, Lindsay Rosen, Krista Zerby, and Sarah Brown. We could not have done this without them. Our collaborators here in the Dental Institute, Nikki Moutsopoulos, and in the Arthritis Institute, particularly Sarthak Gupta, and collaborators at University of Athens, Harry Moutsopoulos, at Konkan University, Plonchon Choksad, and Paul Bastar and Jean-Laurent Casanova at the Necker and the Rockefeller. So finally, I just want to say again how grateful I am for the chance to speak in a symposium honoring Dr. Sudhir Gupta, who really has been an inspiration and a guide in so many of these endeavors. Thank you very much.
Thank you, sir.
That was great, Steve. As usual, that was just a beautiful presentation. And I think they have
Happy to take
something
questions.
else to add.
If there are any questions for...
Thank you, sir. That was an eye-opening presentation. Sir, we have quite a few patients of thymoma who, some of them are, because of pure red cell aplasia, they are on immunosuppressive agents. Some of them are also on IVIG because of hypogammaglobulinemia. So we have quite a few of them who present with bacterial, viral, as well as fungal infections. So would you also consider rituximab in those patients?
Yeah, I think I would. If you have B cells, we're certainly happy to test for the anti-IL-23 autoantibodies. But thymoma, like APOSED or APS1 deficiency and like RAG deficiency, thymoma has real troubles with thymic regulation of T cells that seem to be very interested in telling B cells to do bad deeds. So yeah, if you've got evidence for autoantibodies in thymoma, I would certainly think about it. The pure red cell aplasia, you really want to see, is that anti-erythropoietin antibodies, or is that T cell driven or B cell driven? And I think in these settings, in good syndrome, that is, people who really have no B cells and no immunoglobulin, we do not see much in the way of anti-cytokine autoantibodies. But in thymoma patients with preserved B cells, there certainly can be T cell dysregulation and B cell dysregulation even after resection of the thymoma. So yeah, I would look, and we're more than happy to help to look with that if you're interested.
Sure, sir. Thank you, sir.
Fantastic talk. It was always a pleasure to listen to you. Now, idiopathic CD4 lymphopenia is also, some of them are autoimmune in origin. They have autoantibodies to CD4. Could they also have infection pattern because of neutralizing antibodies to various cytokines?
Yeah, I gotta say, I don't really understand very much about the autoantibodies to CD4. They're not complement fixing. They're not lytic. I hear that those antibodies are there. I still don't really understand what they do. But, if the question is, are there autoantibodies in people who have those anti-CD4? Are there other autoantibodies, maybe against cytokines, in people with idiopathic CD4 lymphocytopenia? In some of them, yes. And I think, as is true for most things, you're going to have to check. I will just say that for those of you who might be interested in sending blood to us here, we've now worked out a system to do it on Guthrie cards, on blotter paper, so that all you need is a finger stick. Can be done in somebody's home. Just put a few dots on a piece of paper, stick it in an envelope, and mail it, and we can elute the antibodies from it, and the assays are just as good as if we did whole blood.
Because we had a patient of CD4 lymphopenia and had nocardia infection in the brain, recurrent nocardia infection. So looking at what you, I was wondering whether he also had an autoantibody.
I would be thrilled to help you find out.
Sure. Thank you so much.
Sorry, excuse me. We'll not have more questions because, it's already midnight and Stevie was...
No, that's fine. This is when the party starts over here, man. Come on.
Okay.
Just because you guys have morning doesn't mean that I'm not looking for a good time. The bars are open.
We can have
here.
a couple of more. Are you okay with that?
Excellent
I'm
talk,
fine.
sir. There is a patient who has both cryptococcuses as well as disseminated aspergillosis. And after listening to your talk, I think we need to do, so once his treatment is over, we have completed the 3 months of treatment, and once it is better off, then probably we should send a sample for autoantibodies, probably.
Well, I think that, this is not going to answer every question. It's like doing whole genome sequencing. We feel like we're doing a great job when we get 40% success rates. This will account for some patients, not all patients. But it's just like any other testing you do. If every test you run is positive, something's wrong. And so this is just one more element that we should have in our armamentarium. So we'd be happy to do that. Finding people, if you find somebody who's susceptible to both Cryptococcus and Aspergillus, that's funny. And if they've got disseminated disease with both of those, we have seen disseminated aspergillosis with anti-GM-CSF autoantibodies. And it certainly can be associated with cryptococcosis. So that would be a very reasonable thing to do.
Thank you, sir.
We can have one last question from Rudrapan. Yeah.
So great talk, Professor Holland. And so I just wanted to let you know, thanks to you and Lindsay, we were able to set up the PSTAT1 neutralization assay at Lucknow. And
great.
In our lupus cohort, we now have approximately 23% patients positive for anti-interferon antibodies. And in addition to COVID that you showed us, we also have a lot of TB and roughly 3.5 times the risk of TB and 3 times the risk of herpes zoster in these patients. So a lot.
Wow, that's really nifty finding.
So, thank you.
Have you published that yet? You should do that.
No, I was just about to send it over to you. Maybe in the next couple of days. Yeah.
I have nothing to do with it, but you need to get it published. That's a great observation.
Okay, I think we'll close the questions now.
All right. Well, I'm going to hit the bars. So thank you very much. It's really an honor
thank
to
you
be
very
part
much.
of
Thank you very much. So the award, the scroll and the honor would be received on your behalf by Professor Sujit Singh, and it will be handed over to you in NIH by Dr. Deepthi, who would be traveling next month. Is that okay?
I'm looking forward to all of those things. Yes.
Okay.
That sounds wonderful. Thank you.
So now I request Professor Sujit Singh to receive the award in Professor Stephen Holland's behalf.
Thank you very much.
Thank you, sir,
Thank you.
for your insightful oration. Up next, we have two oral paper presentations. The first one will be by Dr. Nidhi Desai from ICMR NIH Mumbai. And I would like to request Dr. Sujit Singh and Dr. Lisa Rajashekhar to kindly judge the presentations. Thank you.
Okay, good morning everyone. I'm Nidhi Desai from ICMR NIH in Mumbai, and today I'll be presenting part of our work on autoinflammatory disorders that we've done in the last 3 years. So, to go from here. Okay, so as we all know that inflammation comes in multiple different flavors, and that is very evident clinically that in autoinflammatory disorders there's a wide range of clinical manifestations, and this is because of multiple genetic causes to it. So there are around 60 genes that are known to cause autoinflammatory disorders, and knowing the exact genetic mechanism is very crucial because this guides us, this tells us about what pathways are dysregulated, and therefore what treatments can be given to these patients. So from our experience of around 60 cases of autoinflammatory disorders, where we have done whole exome sequencing to find the monogenic cause, we have found pathogenic or likely pathogenic mutations in only 50% of the cases. So approximately 50% of these remain unresolved. So we wanted to see what is causing this big group of unresolved. Out of them, we have at least 2/3 who have a variant of uncertain significance in a candidate gene that might be linked to autoinflammatory disorders. However, many of these are missense variants. Further, many of these come from disorders that are autosomally dominantly inherited, but we still have almost 3/4 of the variants that are coming from asymptomatic carrier parents. So it makes the classification as per ACMG guidelines a little difficult for us. So with that, I would like to say that with the heterogeneity in the clinical phenotype and lack of definitive functional tests and known incomplete penetrance in many of these disorders, it makes the resolution of variants of uncertain significance very difficult. So for this, we decided to go with a transcriptomic workflow. So we took the RNA from the peripheral blood and did a total RNA sequencing a total RNA sequencing library after globin and rRNA depletion, and then sequenced in a 150/2 platform with around 40 million reads per sample. And then we subjected them to multiple different bioinformatic pipelines, and I think most of this goes on a trial and error basis on these monogenic cases, because we don't know what exactly will help us in every situation. So we did multiple pipelines, but I will just go by some cases and what was most useful for us. So the first case that I wanted to describe is actually two families who had a very similar presentation of recurrent fevers, arthralgia, and ulcers. And in both of these families, the symptomatic patients were carrying this heterozygous mutation in TREX1, but there was one family member who did not have any symptoms, and she also carried the variant. So we knew from other tests that we had done on this pedigree that the mutation itself could be having a impact on the protein function, and we were also able to see differences in the STAT1 and the DNA damage pathway in this patient. So because of that, we went back to the RNA-seq data from this pedigree, and we saw that most of the, all three of the patients had a very strong expression of the mutant allele, whereas the one family member who was completely asymptomatic had a roughly 50% of both the mutated and the wild type allele. So with this, I can say that maybe there is a chance of monoallelic expression in some of these cases, which could be explaining why only some members of the family were expressing the disorder. The second is a case of a missense variant, where we had two different pedigrees, both with very different manifestations. One who had recurrent stroke-like episodes and vasculitis, and the other had mainly different autoantibodies and arthritis of the large and small joints. So both of these families, we had a missense variant. We had a missense variant in ADA2, and by using a strategy called Outrider, which helps us see what are the most different outlier kind of events in RNA-seq data, we were able to see that both of these patients have some similar set of genes that were outliers. We confirmed by qPCR that the ADA2 expression itself was low in both of these patients, and we were able to see common targets of lysosomal organization, type 1 interferon pathway, and NF-kappa B pathway that are dysregulated in both of these patients. The next strategy is like, what do we do with all of the cases where we don't have a variant of interest, or where we don't know what to put them under. So for this, we decided to first train our data set on known pathogenic variants, where we did a weighted gene correlation analysis, and we narrowed down to modules that were specifically enriched in either the interferon or the pyrin-mediated autoinflammatory disorders. And then we took these modules, and so that is around 1,500 genes, and we tried to look at how they are segregating our query cases. So with this, you can see that there is a lot of query cases that still remain unresolved, and I think the more well-trained our training data set is with more different kind of cases, I think we will be able to solve many more of them. But you can see that many of our query cases do cluster with like the interferon signature pathway here, or the pyrin-related inflammation also. So with this, I would like to conclude that although the DNA does tell us what might happen and does point us towards a mutation, RNA actually tells us what is happening. So this will, to include RNA-seq as a tool in our diagnosis toolbox, helps us in giving us more clues about whether there is a monoallelic expression or a different transcript isoform that is expressed, as well as giving some molecular stratification as to which pathways may be dysregulated, and that can help us in treatment even if we don't know the genetic disorder. So with that, I would like to thank my lab and our collaborators and our patients.
So, Dr. Nidhi, what do you think are the clinical implications of your study?
Yeah, so I think one is where we have a VUS. Sometimes RNA-seq tells us why that, whether that VUS can be more on the pathogenic side or the benign side. And the second is where we don't have a VUS, or we don't know what genetic it could be. We can still probably, give some input as to, yes, maybe a JAK inhibitor would be more useful because this is more likely a interferonopathy, or maybe going for a IL-1 inhibition where we are seeing the similar trend as in other pyrin associated disorders.
So you did it on whole blood, right?
We did it on whole blood, yes.
So I mean, it's possible that the lack of penetration could be due to lineage specific, cell specific expressions. So are you planning to explore that?
Yeah, that's a good question. So there are two things. So we did the whole exome also on blood, right? So there could be somatic variants that we are missing anyway because we might not have had the depth in that. The second thing is about the RNA-seq data. So we are currently trying to do a little bit more analysis into, there are bioinformatic pipelines to actually see which cells are there in your RNA-seq data, and then doing variant call on them. Let you know.
Good work.
Very nice presentation, and you went, ahead just not only with the sequencing, but went ahead with the transcriptomic data. So my question is that in terms of the heterozygous mutation, that you are able to explain the incomplete penetrance just because of the monoallelic expression, why the asymptomatic carrier might not develop the symptoms because of the wild type expression is more compared to the mutant allele. But my question might be now, how when there is a homozygous mutation, do we also find incomplete penetrance in those cases or not? And can that be explained by the monoallelic expression or not?
Okay, sorry. So if I understand right, you're asking if there is incomplete penetrance in homozygous mutations.
Yes.
Okay. So I have not personally seen any of those cases so far, but I think one of the things that could explain something like that is additional variants that might exist in maybe the non-coding regulatory regions of genes, and maybe that's affecting half of your genetic genotype, right? So maybe instead of going from a homozygous, now you're actually at the expression level of a heterozygous individual. So
thank you.
Is that it? Okay, thank you.
The next presentation is by Dr. Abhishek from SGPGI, Lucknow.
So, good morning to all of you. Good morning to all of you. Today we will be discussing 3 cases of mutation in inflammasome components. So the first patient is a 9-year-old boy who was initially presented with fever and rash, which was cold-induced, and it was terminated by fenofexidine. It has subsequent similar presentations, and his inflammatory markers were elevated. So after these 3 episodes, his inflammatory markers became normal and he became asymptomatic. So whole exome sequencing reveals heterozygous mutation in the NLRP1 inflammasome, where serine at 482 position was replaced by phenylalanine. So he has recurrence of fever and weak, and which was subsequently at every medication was terminated by fixofenadine. So on functional analysis, it was found that this mutation leads to disruption of the NAC domain of NLRP1. So due to this disruption, NLRP1 did not bind to thioredoxin 1, which negatively regulates NLRP1. So our second patient was a 8-year-old female who has a 1-year history of swelling and pain in joints, who was initially enrolled in medical genetics for evaluation of short gesture, webbed neck, white space nipples, and Turner's was suspected, but her karyotype came normal. On initial evaluation, camptodactyly and knee arthritis was found. And also her inflammatory markers were elevated, and on ultrasound, extensive extensor tenosynovitis in the wrist and synovitis in the knee was found. On whole exome sequencing, mutation in NLRP12 was found, where base pair deletion at C-terminal leads to a truncated NLRP12. Our third patient was a 23-month-old male who presented with large volume recurrent diarrhea and urticarial rash at 1 week of life. He has multiple episodes of intermittent diarrhea, and at age of 9 months, he has additional symptom of self-limiting jaundice, and at age 18 months, he has additionally ascites. On endoscopy, snowflake appearance of duodenum was observed. So, and also his inflammatory markers were raised, and cold-induced urticaria was there at the age of 23 months. Whole exome sequencing reveals that it has duplication of the NLRP3 gene. So, similar to these 3 patients, we have total of 8 patients, including 3 patients. So, total 3 NLRP3, 3 NLRP12, and 1 each with NLRP1 and NLRC4. As you can see, they have varied manifestations like fever, urticaria, and GI involvement. But 1 patient with NLRP12 mutation has lupus. And so, next, what we did, we look for the serum and blood and urine levels of inflammasome products, that are IL-1 beta and IL-18. And what we found, that as compared to healthy control, there was elevated levels of IL-1 beta and IL-18 in serum and urine. Next, we did the ex vivo experiments, and we isolated their PBMCs and activated inflammasome with LPS priming and benzyl ATP activation. And what we found, that upon activating the inflammasome, there was very high levels of IL-1 beta and IL-18 in the supernatant. This was interesting, but it was also interesting that on LPS priming also there was more elevated levels of IL-1 beta and IL-18 as compared to the healthy controls, which are the bottom black lines. So this suggests that inflammasome pathway is highly activated and most sensitive in these mutants, in these patients with mutation. So NLRP1 is basically present on the barrier surfaces of the skin and is pro-inflammatory inflammasome. So reported diseases have presentation of keratosis and palmoplantar carcinoma, and our patient has novel presentation of cold-induced urticaria and fever. NLRP3 has similar, our patients with NLRP3 mutation has similar manifestation as reported earlier, and NLRP12 is also expressed in the myeloid cells. It has an anti-inflammatory role. It acts as a brakes for NF-kappa B activation and NLRP3 inflammasome. So our patient represents a novel presentation of juvenile lupus and tenosynovitis. So to end, NLRP1 is critical mediator of inflammation in barrier cells, and chronic inflammatory state leads to NLRP12 activation. NLRP12 mediates anti-inflammatory effects by inhibiting an NF-kappa B activity and NLRP3 inflammasome. And in case of lupus, interferon-mediated RUNX1 dependent NLRP12 suppression lead to increase in autoantibodies production, IgG deposition, and progression of lupus nephritis. And loss of mutation in NLRP12 leads to exaggerated NLRP3 activation and may shows phenotypes similar to gain of function of NLRP3 mutation in NLRP3. So these are the references, and thank you.
Abhishek, how did you treat these patients?
Sir, actually I'm a non-medical PhD student, so I am presenting in behalf of my clinical colleague. She is not well, so I may not be able to, but the first patient was treated with fenofexidine.
You mentioned a patient who had a lupus-like disease or lupus disease.
She.
So in that patient, what was the value of the IL-1 beta and the stimulated IL-18? And was it on the lower end? Or
in patient which has lupus, she has maximum levels of IL-1 beta and IL-18 in both serum and as well as in urine.
as high as the NLR? As high as the highest one? There was one
highest one for NLRP12, which has lupus.
Okay,
then
fine.
NLRP1 and NLRP.
And what do you think is the reason this patient is getting lupus? It's not a described phenotype.
No, yes ma'am. But there was a paper in 2024 which proves that in lupus mouse model and in cell line that when NLRP12 is inhibited or truncated, then the mouse model has more lupus-like phenotype, which is interferon mediated. Like interferon acts as a negative regulator of NLRP12 by RUNX1 dependent. So NLRP12 is involved in controlling interferon mechanism of action. So
Thank you.
Any other questions? Okay, thank you. Thank you very much.
We move on to the next session. I would like to request Dr. Mahesh Janarthan and Dr. Suma Balan to chair the next session on exploring the landscape of type 1 interferonopathies from nucleic acid biology to targeted therapies. The speaker of the session will be Dr. Alexander Bello. He will be joining us online. Thank you.
Good morning, pleasure to be here. A very salient topic, and Dr. Alexander Bello is Professor of Pediatrics and the head of National Reference Center for Rare Rheumatic and Autoimmune Diseases in Lyon, France. And his main areas of interest are pediatric rheumatological inflammatory diseases, systemic autoimmune disorders, and interferonopathies. It's a very approachable and a very friendly researcher, and very amiable to discussions anytime you want. The floor is yours, Dr. Bello. Welcome you to start the talk.
I don't know if you see me or if the connection is correct. Is that okay, your side?
Yes, I think it's okay.
Yes.
Okay, fantastic. So, namaste, everyone. It's my immense privilege to be with you on Zoom. I'm sorry for not being on site. I was not allowed to travel because of the Dubai transition. But any case,
There's an
I'm
echo,
very...
Dr. Bello. Is it?
Yeah,
are
I
you
see
on
that.
two separate devices at the same time?
I have only one device.
Only one. I think carry on again. Let's see.
So I think there is no more echoes now. So
Yeah,
it
it's
sounds
fine
better.
now. Thank you.
Okay, so good, thanks. So first I'd like to thank the organizer for giving me the opportunity to be with you, and Sudhir especially, we know for a couple of years now. So yeah, I'd like to speak about this fascinating field of type 1 interferonopathies. I saw that in the previous short talk you have already great interest and skills in this field.
But back to basic, interferon, what that means exactly. So this is the key antiviral cytokines, as you know, and it was discovered almost 70 years ago by Jean Lindenmann and Alec Isaacs. What they did at that time, they infect, let's say, chicken eggs. It was a chorioallantoic membrane, but we'd say some cells with the flu, with influenza virus. And they took the supernatant of the experiment and they applied this supernatant to another cell line, the other chicken eggs. And they realized when you infect the cells in the second situation and when you have transferred the supernatant, there is a sort of resistance of the cells relative to the infection, meaning that there is some interfering factors that act there that is secreted, and that's the name of interferon that came up at that time. Now we know a bit more about this type 1 interferon production, and globally, so it is picked from Rafaela's talk and paper, very nice review. You see on the left that essentially here it is the way our cells can detect RNA, DNA with intracellular sensors that will, at the end, produce large amount of type 1 interferon. Another situation is to detect extracellular material that comes into the endosomes, and here it's essentially the TLR that will enable the detection of nucleic acids and activate the production of type 1 interferon, which globally, these type 1 interferon are quite difficult to analyze in the serum. So they then go through the specific heterodimer, if not one, if not two, and signal via JAK-STAT signaling to induce the so-called interferon-stimulated genes that are also the interferon signature. You mentioned this in the previous talk, so I know you're familiar with this. Aicardi-Goutieres syndrome, so it's an entity first described in 1984, so a couple of years now. It's two neuropediatricians, Jean Aicardi and Françoise Goutieres. They were neurogeneticists and neuropediatricians in Neckers, and they found in single families recurrence of situation of kids and neonates with encephalopathy plus microcephaly plus brain calcification. It really looks like a Torch syndrome, but they screened for infections and congenital infection. They didn't find anything, and in the sibling, it recurred. So they said there is something familiar there. There might be some genetics here, and then it was called the Aicardi-Goutieres syndrome as a neurological disease, essentially, at the beginning. But 4 years after, they found a sort of marker that can be picked up in the CSF, which was interferon alpha. So they said, well, we have not the gene, but we have at least a biomarker, which is the interferon alpha. And now, I mean, 20 years after, the gene was discovered and make this entity clearer. So it's made of encephalopathy, leukodystrophy, microcephaly, cerebral calcification, and something interesting for the immunologist, childblain in some cases. And Yannick Crowe, in a couple of years from now, but discovered the gene TREX1. So as you mentioned this in the previous talk, so you know you're familiar with TREX1. That's not a dinosaur. That's a gene responsible for this specific entity. And other people working on childblain, for example, said, well, we are interested to check if TREX1 could be seen in patients or family with the childblain, and that was the case. And even in adult onset SLE, 0.5 to 1% of lupus were carrying pathogenic variant in TREX1. So it was a start for the immunologist to think about this gene. And Yannick Crowe, a few years after, introduced the concept of type 1 interferonopathy, saying that when you overproduce, because of genetic defect, this large amount of type 1 interferon, and we consider that most of the manifestations are linked to type 1 interferon, that could be called the type 1 interferonopathies. So now the field and scope is broader. And there are some very severe situations, starting even before birth, with brain calcification, microcephaly, but there are some milder phenotype. Here in the blue, I mentioned the neurological phenotype. You can have just paraparesia. You can have just late onset paraparesia. You can have just brain calcification, or possibly nothing else that a positive interference signature, which make this entity quite difficult to analyze. And on the other side, you can have also so here it's a paraparesia. I hope the video works, but you see this boy was diagnosed with an FEH1 mutation. It's a dominant mutation, and he has a paraparesia. And but he is totally fine in terms of intellectual development, but he has this paraparesia. His father has the same, slightly less severe. The grandfather has just brain calcification with nothing else. There are some patients with childblain or even full-blown lupus, and in a single family, you can have either neurological involvement, autoimmune phenotype, or both, and across the different generation, it can be totally different, even if it's the same gene. So just something special to remind. So let's start with this specific first part, which is probably the most clear type 1 interferonopathies, when you have some anomalies inside the intracytosolic detection of RNA or DNA. So globally, this is a picture of all your cells. We are lucky to have sensor to detect DNA, RNA. It is a way to fight against viruses, as you know. So it's very powerful to produce interferon to protect us from viruses. And we have a signature that we can run this type 1 interferon signature. It's the transcriptomic signature that reflects the presence of type 1 interferon, and it's always positive in type 1 interferonopathies. So always is not the perfect term in medicine, because sometimes there are some exceptions. Here for RNase H2B, which is a specific genotype where sometimes type 1 interferon signature can be normal, but globally remind that it's essentially always positive and very discriminant compared to controls. We really like this signature because it can be informative even in non-genetic disease to say that there is a high interferon contribution in lupus, as you know, but in dermatomyositis, tetrasomy 9p, it's just because the cluster of interferon genes is located on that site. But that explains why this patient develop also autoimmunity. In GIA, do you see that some essentially is negative, but some patients have positive interferon signature, which is still intriguing and might be meaningful for the treatment. So we have set this analysis on routine for a couple of years, and we run about 2,000 interferon signature a year. Now we increased to 4,000. So I think it's definitely useful, especially because patients can have normal CRP and sky-high interferon signature. So my opinion is that we should increase the number of interferon signature if possible. So here you see the cells I mentioned, and actually there are plenty of enzyme. This is the first part of type 1 interferonopathies. Plenty of enzyme that will digest these nucleic acids or convert these nucleic acids into some specific pathways. And actually this is really potent and act permanently within the cytosol. And we can imagine that there is not a lot of nucleic acid in the cytoplasm, but actually that's the case, even without any outside infection, because we have mRNA for the translation of protein, we have mitochondrial DNA also, but something very important, we have also within the nucleus or retroviruses, endogenous retroviruses. Initially, we thought it's just junk DNA and it doesn't, it's totally useless. It's just the architecture of the chromosome. But probably these retroviruses play a role by replicating within the cytosol. But on a steady state basis, there is all this enzyme that digest the excess of nucleic acid. So there is a balance between the production of nucleic acid in the cytoplasm, enzyme that will digest or metabolize this specific nucleic acid. And when you have a defect in these various enzymes, you will accumulate the nucleic acid that will be detected by the innate sensor. The innate sensor, they don't know if it's an outside viral infection or if it's your own material that is accumulating in the cell. So you produce large amount of interferon, and that's an explanation why it looks like Torch syndrome, because actually you produce the message that there is something wrong in your cells when you have this mutation. So here it's just an illustration of a patient with a SAMHD1 B allelic mutation. He developed early in life cerebral palsy and childblain. So childblain is a good red flag to think about type 1 interferonopathies. Interferon was positive any case. And I just wanted to mention that as we feel that it's this sort of enzyme are critical to get rid of the retroviruses that replicate in the cytoplasm, Yannick Cruz tested purely just on type 1 interferon signature. Look at this specific treatment, reverse transcriptase inhibitor, thinking that if retroviruses, endogenous retroviruses, replicate in the cytoplasm and share the machinery with HIV, possibly RTI could be useful to treat this patient. That's what he saw, where the patient was included in the 8 patient treated over 12 months. He showed that we can decrease the type 1 interferon signature over time, and at the end of the protocol, it came up again and rise a bit, meaning that in this genetic disease, we can target the interferon signature with an antiviral treatment, which is quite fascinating because maybe in the future, even in non-monogenic lupus, and we have a trial that's just starting here in Lyon to look if abacavir lamivudine can reduce the type 1 interferon in lupus patients. So the other part of type 1 interferonopathies are linked to an excess of innate sensors that have gain-of-function mutation and will induce, without any nucleic acid, an increase of the signal and then of interferon production. So here you have the innate sensor or adapter that can be linked to this overexpression of type 1 interferon. And I just want to say a few words about the SAVI, which is a fascinating disease, and I'm sure you're all aware of this, and Raphael made the first description. You see the skin lesions with the vasculitis and tissue loss. It's really impressive. Some of the patients can even present with a lupus phenotype. And associated to this is often an interstitial lung disease, which is very difficult to treat, evolving into fibrosis. We treat this patient with the JAK inhibitors. Here, in all this gain-of-function mutation, there is no reason to introduce reverse transcriptase inhibitors. As you can imagine, it's purely something linked to overactivation of the sensors. And it seems to be powerful, at least in skin. This is one of my patients who was let's say, correctly treated in terms of skin lesions. For the lung, we felt that it was almost stabilized, if not no more progressive or slightly less in terms of kinetics. And but unfortunately, many of these patients still have this lung disease, very severe, that will require possibly lung transplantation. And on our French experience, 3 of the patients who underwent a lung transplantation under JAK inhibitors actually died. I didn't make the follow-up, but it's now 3 out of 3. So meaning that even if you make this lung transplantation under JAK inhibitors, it's still fatal in a few months or years after the initiation of the treatment. So that's a very, very challenging situation. So possibly looking at the bone marrow and made some intervention there, we have few reports saying that bone marrow transplantation could cure the disease. And together with Matthew Porteus, Sébastien Vial, we thought that maybe we could try to first knock out the gain of function allele and then edit the genome and re-express the wild type alleles. That could be an option in this very severe disease, but it's still ongoing and totally in vitro right now. Another cousin of SAVI is the COPA syndrome. Due to mutation in COPA, it can affect more maybe the kidney with some lupus nephritis presentation, but also the lung, with lung hemorrhage, while SAVI it's more interstitial lung disease. And these two situations are well connected because actually COPA is critical for the recirculation of the protein from the Golgi to the ER. And within this complex, the COPA complex, it can help the sting one molecule and to recirculate into the ER. And when it's stuck to the Golgi, it will serve as a gain of function sting mutation, exactly the same phenotype. So interestingly, in most of the disease, there are some expression difference, penetrance difference, which is intriguing. It might be monoallelic expression, as you mentioned earlier. It could be also that there are a second gene, very important. And here, recently, colleagues have found that this haplotype, HAQ, in sting may explain why some COPA carrier do not develop the disease, because this is a protective allele that will decrease the production of type 1 interferon. So here, this is a beautiful expression of the different genetic tree. You can see here the patient's carriers with no symptoms, and globally, all of them had this specific protective haplotype. So that's something interesting to say that maybe here for COPA syndrome, we should maybe look at this specific haplotype as well. It is informative. In vitro, they showed that indeed, when you have the haplotype, you decrease massively the interferon expression. And on our side, we have a couple of families as well. We looked, this girl was initially, so the index case here, initially described with the lupus nephritis a couple of years ago, saying that yes, COPA can be a lupus cause, the monogenic cause of lupus, sorry. And the father developed at the age of 50 years a membranous nephritis, and actually he is carrying this protective allele, so it's probably not a fully protective allele, HAQ, but maybe it delayed the symptoms during the life, possibly. And still on her data, and Marie-Louise Frémont made a fantastic work to pull up all the data, additional 5 patients without HAQ allele have no symptoms, so meaning that there is still some work to do to learn about this expressivity and penetrance of the COPA syndrome. I jump to the extracellular detection of nucleic acids that go through this endosome and then been sensed by TLR. I will say a few words on TLR because as you are all from the immune deficiency field, you're very familiar with this hemizygous mutation of TLS7. So this is purely immune deficiency with severe viral infection like COVID-19 or flu and now many others. Viral infection have been linked to TLS7. And just during the COVID, Jean-Laurent Casanova presented this study, even if TLS7 was already known. But what was funny, it's that the same year or the year after, TLS7 was also identified with a gain of function mutation driving a lupus phenotype. And probably in immune deficiency, there are many situations where a defect can be associated to viral infection, and an excess of this same molecule can drive an autoimmune phenotype. So now there are more than 14 patients. A lot of them present a lupus phenotype that have been described, and I just present one we have in Lyon with the early onset lupus, classical lupus plus neuroinflammation and some paraparesia. And the girl to develop a lupus nephritis, so globally all the mutations in the TLR are located in the pocket that interact with the nucleic acids. Initially, we have not the diagnosis. We realized the interferon was positive. We checked when we had the variant that it is an overactivation here on an NF-kappa B luciferase assay. But the girl was treated with a B cell depletion. It was the third or fourth line of treatment, and it works well, actually. Maybe not anticipated. I'm sure I won't start with this, with the genetic diagnosis, but globally she's fine just by targeting the B cells. So it's a way to remind us that TLR signaling is really important in B cells. Globally, this is the picture of all the cases reported so far. And for you, just a reminder that it's highly lupus associated here, TLR7 gain of function mutation, plus neuroinflammatory disease. Another gene, just sort of chaperone protein associated to TLR, is this UNC93B1. So you know this gene from HSV susceptibility, but there is also a mutation that can drive the lupus phenotype by a defect of recycling the TLR at the endosome membrane. And the phenotype in UNC93B1 is more chilblain or lupus nephritis. Then I move to the last part, which is this cascade of type 1 interferon signaling. So when the cytokine is produced, go to the IFNAR heterodimer and then signal via JAK-STAT. So this specific part, there is some natural break. We all produce in all cells some natural JAK inhibitors, let's say, and that will drive a global overexpression of interferon-stimulated genes, and depending on the cytokine, maybe all the pathway can be involved. So it's a broader phenotype, let's say. We started with this boy a couple of years now, with the lupus, and found a variant in SOX1, which is an inhibitor of type 1 interferon and the JAK-STAT cascade. But when you look at this, the first analysis is the segregation, and the segregation teaches us that the brother, totally healthy, and the father, totally healthy, carried the mutation. So we thought, okay, that's not a good idea, we stop there. But another family pop up with one thrombocytopenia, autoimmune, plus another with a lupus phenotype, and again, a variant in SOX1. So it was a different variant, but still predicted pathogenic, not seen in any other situation. So we ran the assay and try to learn more about this. Here you have the segregation. The brother has psoriasis. Would you think it's the same disease? Lupus, cytopenia, psoriasis. The father has just a positive interferon signature, and the aunt, I think she's having nightmare with the physician because they said it's a sarcoidosis, it's a lupus, it's a rheumatoid arthritis. She has many, many different diagnoses. That means that indeed she has an inflammatory disease and actually probably a SOX1 haploinsufficiency. So SOX1 is all natural JAK inhibitors specifically induced by the interferon, not only type 1 but also type 2 as you see here. And knocking at the door of Frédéric Rioloca and Stefan L. in Germany, they had also patients with Evans syndrome. And we pulled this observation together and found that indeed, in vitro, we can show that there is an overactivation following interferon gamma stimulation of PBMC of the cascade, as you see here for phospho-STAT1 in the patient, and that could be normalized using a JAK inhibitors in vitro. So the interest of this is that you can treat the patients. And for the psoriasis, back to this specific phenotype, is it or not linked to SOX1? The patient, it was very difficult to treat the patient with methotrexate, with steroids. But at the time, the dermatologist say, well, maybe, let's say, a JAK inhibitor could be helpful. And it was the only way to treat this refractory psoriasis, meaning that actually, more than the specific phenotype, the genotype is maybe more important to decide a treatment. Another hit in the cascade is PTPN2. This is another natural inhibitor. In that case, it's not induced. SOX1 is induced by the cytokine under the receptor, while PTPN2 is widely expressed on the steady state. We found still Evans syndrome and lupus, and globally I have the feeling that these JAK-STATopathies are more frequently associated to Evans syndrome, cytopenia, or lupus phenotype. So we went to the analysis of the different variants, and this is the same story of SOX1. So for the sake of time, I won't be too in detail of that. But some of the variants were loss of expression, others were loss of function. And we see that these patients have an hyperproliferation of T cells under IL-2 stimulation that can be rescued by a JAK inhibitor. And the last observation is this girl who was diagnosed by Dr. Asunta Ho in Hong Kong. She had early onset lupus with the typical rash, but some anomalies like clubbing, myositis, high double-strand DNA antibodies. And she discovered now a STAT4 mutation, which was located in an SH2 domain, exactly the same place of the other patients described with the STAT4 gain of function. We developed, I don't know if you have seen the picture, but a very severe skin lesions and diffuse scleroderma. It was a New England Journal paper. And so we tested in vitro the variant that showed an increase on NF-kappa B activity and interferon production. And it's not actually STAT4. If you think about interferon, you'll think about STAT1 or STAT2, but there are some indication that there is alternative signaling of using STAT4 that can overactivate the type 2 interferon after, and it's a sort of amplification loop. And also there are some variant in GWAS detected in STAT4 for lupus. So we treat the patients with the JAK inhibitors, so my colleague in Hong Kong, and it was really powerful to stop the lung disease and the skin lesions. So to finish, how to treat these patients, how to go directly and for the good treatment. Globally, the JAK inhibitors, there are many reports on that. It seems to be quite tissue specific. It works well on the fever, on arthritis, on the skin. For lung disease, what we learned from Savi, it's not so easy to treat the lung disease, and maybe it protects the evolution in some extent. We have not seen any efficacy of JAK inhibitors in CNS and neurological involvement, unfortunately, which remains a situation very challenging, possibly also developmental lesions that cannot be treated by any immunosuppressant. And there are some exciting data coming up from some published cohorts, so one from the NIH group and others from Peter Broden in Sweden, showing that anifrolumab could be an option. There are also some case reports. So here you target directly the IFNEO1 receptor. So collectively in France, we have around 26 more than that were put in the study, but 26 were already analyzed, showing, so this is the description of the patients treated off-label by anifrolumab. This is the phenotype of the patients, so a lot of them with neurological involvement, lung disease for the COPA-SAVI patients, etc. And so this is the global characteristics of the patients, the dose of anifrolumab that was used, and the concomitant treatment. And as you may see, a lot of them are also treated with JAK inhibitors. It's a second-line treatment plus JAK inhibitors, but it seems to work on skin quite well. Here, this is a patient with refractory childbearing, even under JAK inhibitor, who was cured by anifrolumab. We have also the situation where, for the first time, we can normalize the type 1 interferon signature, which is not always the case for JAK inhibitors, especially in SAVI. It's not possible to normalize interferon signature with only JAK inhibitors, while it is possible with anifrolumab. And then, just as a conclusion of this large overview, I think personally, I always think about type 1 interferonopathies, even when someone knocks at the door, no, it's my medical student, but I always think about interferonopathies. But think about this: if you have childbearing, if you have neurological features, paraparesia, cerebral calcification, even if in a multiplex families with some autoimmunity, some with neurological symptoms, it might be all of this situation the same gene. the expressivity, penetrance, we have loved to learn about this. Type 1 interferon is key, and I know together with Amit Rawat, we thought about this specific exam to be set. I think you have access for some research project to the signature. And the treatment remained very challenging. JAK inhibitors is a good option, but not for all this situation, and probably with a tissue-specific activity. Anifrolumab is quite promising, I should say. And for the reverse transcriptase inhibitors, it's still on the research part of the project. So I just present at the end all the collaborators in France, abroad, especially the ones who took the work on anifrolumab that just presented now. And I'm done and happy to take questions.
Thank you for your wonderful talk, Dr. Bello. Before we go to questions, I think Dr. Jigna wanted to make an announcement.
Yeah, after this question answers are done, the slot 1 and slot 2 poster judging sessions will be starting. We'll have Dr. Amit Rawat, Dr. Vignesh and Dr. Yukurin and Dr. Sarath for the judging of the slot 1 and slot 2. We can take the questions now.
Any questions from the floor?
Thank you for the wonderful talk. It was the JAK inhibitors that you mentioned. Do you think starting JAK inhibitors early, even before neurological involvement, would prevent a neurological involvement in these patients? That's question number 1. And does anifrolumab work in neurological manifestations?
This is key. This question is really important. But I'm sorry to say we have no response or partial response. Let's say for the JAK inhibitors, we know that in terms of dosage, it doesn't go through the CSF easily, and the concentration of the drug is about 10 times less in the CSF compared to blood, so meaning that it's very challenging to block something even with the JAK inhibitor, so it's quite disappointing. And then I think for Aicardi-Goutieres syndrome, the neurological involvement seems to be also partially developmental, maybe linked to the exposure to the type 1 interferon during the pregnancy and possibly not reversible. Right now, we are not convinced that the JAK inhibitors and maybe not the anifrolumab, but we have not enough data, can do something on CNS involvement or the progressive neurological symptoms like paraparesia. And it's very difficult also to catch to start a treatment before the neurological features appears, because it's often part of the onset of the disease. But it's some missing information here, and we definitely need something for this CNS involvement.
And if I may add one more question, how long have you used JAK inhibitors? Does it mean you will have to continue it lifelong?
Actually, when we start JAK inhibitors, we never stop, or I should say on our experience in my center, for those who started the JAK inhibitors 5 or 6 years ago, they're still under JAK inhibitors. There are some specificities for people with a severe childblain with tissue loss, while it is essentially associated to the winter season. In some patients, we can, and when it's only skin lesions, only during the winter, we decrease and stop the dose during the summer, and we take again the JAK inhibitors during the winter season. So there could be some alternative treatment like this. But in most of the patients, they are treated for years with the JAK inhibitors. And we have some few things to check, especially BK virus infection, which is not conventional. Usually we see that in very immunocompromised patients or following organ transplantation, but in patients with JAK inhibitors, we also see some features of BK virus infection and possibly infection and lesion and kidney injury due to BK virus on the which is not very frequent, possibly because also we use very high dose of JAK inhibitors.
Sure. Thank you.
Dr. Alexander, for your experience with anifrolumab, 30 patients which you showed, how were these patients chosen? Were they because of refractory disease or because of the interferon score?
It's also about history and access to treatment. So initially, most of the patients diagnosed were treated by JAK inhibitors because it was available. And at the time anifrolumab came up to the market for adult lupus, we had access to anifrolumab and started to treat some patients that were usually refractory to JAK inhibitors. So our experience is essentially using anifrolumab in patients refractory to JAK inhibitors or with side effects like severe infection. I mentioned the Zika virus. Our feeling is that indeed anifrolumab is restricted to the type 1 interferon pathway, probably more potent to block the signaling more than JAK inhibitors, with less side effect because the JAK inhibitors, you also block many other pathways. So possibly in the future we will decide to treat the patient depending on the genetics. I mentioned this SOX1 PTPN2 deficiency. Here it's quite logical to introduce a JAK inhibitors because you want to restore the missing break on the JAK-STAT signaling pathway. But for purely type 1 interferonopathies linked to the first part I mentioned, so nucleic acid metabolism or gain of function of innate sensor, possibly anifrolumab could be the best first option for the treatment if it's available.
Any age cutoff to start the JAK inhibitors?
Yes, we start. It's not easy because it's make of pills, so we need to make some specific preparation. But we have patients that started JAK inhibitors at the age of 3 months. So yes, it can be used early.
Sir, your experience with JAK inhibitor and BK virus nephropathy, do you see them frequently? And so for monitoring, do you monitor both the urine PCR and the blood PCR or just the blood PCR?
We used to do blood PCR every 3 to 6 months in any patient with JAK inhibitors when they have a large, when they have the treatment. We also associate with area under the curve. We used to do some kinetics to check that we are not too high in the blood. We don't do any urine analysis of BK. And we had two patients who developed BK-associated nephritis, which required the decrease of JAK inhibitors in one and the changes for anifrolumab in the other one. And the patients recovered after this modification.
What is your experience with hematopoietic stem cell transplantation in type 1 interferonopathy? So patient doing well years together with JAK inhibitors, would you still go for hematopoietic stem cell transplantation, or how to decide the group of patients, particularly hematopoietic stem cell transplantation will work?
It's a big debate. Globally, to my knowledge, there are very few reports, essentially for SAVI patients. And in SAVI patients, I'm aware of 3 cases, only 2 published, I think. So the thing is, so my colleague in Neckers, Paris, went to a SAVI patient who developed severe lymphopenia before the molecular diagnosis was possible. And they found a SCID phenotype and said, well, we don't know the gene, but we will perform the bone marrow transplantation. So he was transplanted, and he never developed any lung disease. He still has some skin lesions and chilblain, which is intriguing, meaning that there are also some tissue-driven effect of sting for the inflammation. And this is one report. Another report from a Chinese group said that a patient with a STING1 mutation was treated by bone marrow transplantation and is totally fine right now. And the other from the Gosh, London, get a bone marrow transplantation and a few weeks after died from complication, but he was already severely injured for the lung. We have experience in lung transplantation in these patients, but we know it's also very challenging, and even with the lung transplant with JAK inhibitors, there is still rejection and inflammation ongoing there. So to my opinion, it's still a case-by-case and global discussion, and we have no clear answer. Possibly there are some space for bone marrow transplantation before the lung is too severely impacted for SAVI. For the others, type 1 interferonopathies, I have no clues, no information about this treatment.
Sir? Last question.
I see a colleague would say, we probably need to stop.
Hello, sir. So recently we have seen a case of a 2-month-old child with seizures, CNS calcification, and aspiration pneumonia. We found a novel variant in STAT2 mutation. So can you tell us your experience on STAT2 mutation leading to the type 1 interferon signature? AGS.
I have no, so I will, I'm sorry, I have no experience in the STAT2 associated type 1 interferonopathies, so I cannot comment on that. I know there are some, as for STAT4 or STAT1, there are some excess of STAT1 signaling or defect of STAT1. So here I suppose it's the regulatory part of STAT2 that is mutated. But no, it's totally possible. They have a strong CNS involvement in the report of the literature. So usually neuroinflammatory disease. But I personally have never seen this patient.
Okay, we'll wind up this session now. Thank you, Dr. Belot.
Thank you. Thank you very much. Thank you.
Thank you all for actively participating in the sessions. We won't wait further. The poster sessions have also started. We request the judges to start the presentation, and we also request all the delegates, the posters have been put up, you are requested to go and view them too. We will meet at 12 sharp to restart the next set of sessions.
Basically, topic is?
I'll help, sir. IEI registries in India. Yeah. So thank you so much, sir, for the kind introduction, and good afternoon, everybody. As sir has already said, I'm Dr. Ritika. I work as Scientist D with the ICMR National Institute of Immunohematology, Mumbai. And now I would like to take you through the effort made for the registry for inborn errors of immunity in India. So this is an effort which is, the contributing centers are as I've already depicted on this slide. So the data is from all of those centers from the registry. So as we all know that the III registry's global effort started almost 3 decades ago, with the ECID registry being the first one and has the largest number of cases. And almost simultaneously, the USID network also started the effort on a national registry, initially to start with CGD and then to include all the other IIIs. And Latin America, France, individual countries like UK, France, and all have also made these efforts, and they have quite a number of cases in their registry as of now, which have helped guide clinical trials and also helped answer a lot of research questions for all the researchers and clinicians, which have translated into good clinical outcomes for these patients. So, so it was very pertinent for us to have a national registry for the inborn errors of immunity in India. And this was an initiative which was taken by ICMR, and PID registry is a part of the comprehensive national registry for rare and other inherited disorders, and has a group of overall 6 disorders, including NMDs, metabolic disorders, skeletal dysplasias, etc., and hemoglobinopathies as well. So this was started in 2019 with the centers who expressed their interest in contributing data to the national database. And this is a prospective hospital-based observational study. And of all the centers who expressed interest, ICMR gave the responsibility of nodal centers to ICMR NIH and PGI Chandigarh. And we were given the task to develop the study proforma, and which would be the tool, for developing the online online website and to identify the data variables which would go into the registry proformas. So we actually modified or adapted our study proforma from the USID net proforma, and it very elaborately captures data on the demography, family history, consanguinity, clinical presentations covering the infections, auto-inflammatory, autoimmunity, vaccine complications and malignancies, etc., everything in detail, along with the lab investigations, including the specialized assays done in the labs for proving the diagnosis. Diagnosis as per the IUI's classifications, molecular diagnosis, treatment details and outcomes. So it's an elaborate form which takes about 15 minutes to fill for each patient. And to add, and further, we also developed quality criteria to check the data entered into the registry. So, which was based on the ESET clinical working party definitions for cases which were not molecularly confirmed. So, for phenotypically diagnosed cases, the 20 commonest IIs diagnosed across centers in India, we developed these definitions and shared with the central team. So it took almost a couple of years where this form was adapted, QC criteria was developed, the online portal came into place, it was pilot tested, technical glitches were overcome, then the workflows, SOPs were developed, and the data entry was initiated. So data quality itself has, like at 4 levels in this. So the first level is at the level of the e-form where, there are various checks and validations which are put to ensure that, the correct data goes into the registry. Then, very importantly, the site proforma has to be approved by the site PI, and the site PI has to ensure that only molecularly diagnosed cases, or else those who satisfy the phenotypic definition as per the ESET working definitions, get entered into this registry portal. Further, the QA/QC is carried out by both the nodal centers in turn, and the nodal centers get weekly proformas to check whichever new entries have come into the portal. We validate and we give our comments, and those are then, coordinated by the ICMR central coordinating team with the other centers who are contributing data. And at the last but not the least, ICMR sends reports of number of cases and the quality of data that goes into the registry, and this feedback mechanism goes on, back and forth. So the data which we analyze comes under 4 parameters: the clinical, epidemiological, treatment and outcomes, and policy considerations. For the clinical parameters, it's age and gender distribution, proportion of patients born out of consanguineous marriages, family history of affected siblings, classification as per IUI's classification, and molecular spectrum. For epidemiological parameters, the contributing centers, state-wise disease burden, which we've not really been able to do now because of the limited number of cases as of now in the registry, and comparative statistics with other registries. Then for treatment and outcomes, the proportion of patients who are on IVIG or have undergone HSCT and their outcomes and risk factors. For policy considerations, again, we calculate the diagnostic delay, we can calculate the requirement of IVIG, and number of patients who need to go for hematopoietic stem cell transplantation, because these are all now supported by the National Rare Disease Policy. So as of now, as of December 2025, the cases which were contributed were from PGI Chandigarh, our center, Apollo Chennai, Nizam's Institute, Sir Gangaram Hospital and Jipmer. 708 patients were enrolled, and after exclusion of missing entries, we analyzed patient data for 676 patients. So the median age of diagnosis of patients entered into the registry was about 53 months, with a male to female ratio of 2:1. 23% families reported consanguinity, 14% had a history of sibling death, and 4% had a death of male member in the family. 67% of the patients presented with infections, and a good number, around 40% of the patients had autoimmune or autoinflammatory manifestations, which clearly indicates that we need to increase the awareness among the physicians that recalcitrant autoimmune and autoinflammatory manifestations should, you should suspect an underlying II. Then allergy and vaccine complications were around 6% of the presentations, and malignancy was seen in 1%. Coming on to diagnostic delay, the median age of presentation was about 16 months, and 16 months was also the diagnostic delay, where the median age of diagnosis, I said earlier, was around 53 months. On the right side is a bar chart which shows the IUI's category-wise diagnostic delay, which we can see is maximum for predominantly antibody deficiencies. being about 53 months, where we know that antibody deficiencies, if you have a high index of suspicion, can be diagnosed simply through doing antibody levels and subsets. It's easy tests to get even at the peripheries if you suspect these disorders. So this slide shows a distribution of cases by IUI's category. So the commonest were combined immune deficiencies taken together, 14%, along with another 14% combined immune deficiencies with underlying syndromic features. This was followed by antibody deficiencies and around 22% and phagocytic defects. So on the right-hand side is the spectrum of, is the molecular spectrum which was seen in our patients. And it was only present with 256 patients out of the entire cohort. So around 40% of the patients were reported with a molecular diagnosis. So again, 70% of the patients diagnosed with antibody deficiencies and around 25% with combined immune deficiencies had ever received IVIG. And around 90 patients underwent hematopoietic stem cell transplantation among those who were entered into the registry. And on follow-up, in December 2025, around 17% of these patients had succumbed to infections. On the right side is the bar chart showing the underlying IUI's category of the patients who had succumbed to infections. As expected, most common were combined immune deficiencies, followed by diseases of immune dysregulation, about 25%, and another 25% in phagocytic defects, followed by antibody deficiencies. So this is a survival analysis we did for our patients recently. I mean, around 106 patients we followed up who were registered from ICMR NIH. And if you can see the graph with the survival benefit with IVIG, so patients who were on IVIG treatment, they had a clear, significant difference from those who were not given. So this is only for IUI's categories 1, 2, and 3. We've done the survival analysis with this thing. So as again, 65% survival among patients at 5 years with those on IVIG. For those who didn't receive IVIG, it was only 35% had survived. So there was a survival mean difference of about 25% among these patients. And even with HSCT, the patients who underwent transplant within the first 6 months of diagnosis had survival of almost 90%. While those who underwent transplant, after a year and 1 to 2 years had a survival of about 50%. So from these observations, there are various policy implications. First, the delay in diagnosis emphasizes the need of increasing awareness among the clinicians. So I guess each one of us here, we are already experts. We can train, clinicians around us. And, that would help in having a high index of suspicion of these disorders, where we need to have them. Then a high consanguinity is seen in our population, which also indicates that cord blood screening or newborn screening in at-risk populations might help in early diagnosis. Then mismatch between the demand and supply of IVIG patients needs advocacy for sufficient provision of IVIG by the state governments. And the commonest cause of death being combined immune deficiencies highlights the need for timely HSCT, which we also see with the survival analysis. Okay. So with this, the way forward is that the burden of IIs is clearly underestimated, and we still do not have sufficient registrations into this registry portal. And it would be great if we could, have more and more data estimating the burden of disease, and because data can always, guide policy decisions at the national level. So maybe in the evening during the GBM, we can have a discussion on how we can, increase the registrations into this portal. So that is it from my side. And yes, this data is recently published in IJMR. I think it came online just this morning. And for us, the patients who were referred to us by all the clinicians also have their name on the PID contributors group. And I hope that this contributors group will increase. Thank you.
Thank you, Dr. Yadav.
Thank you.
Any questions? Yes.
Yes, ma'am. Yes, ma'am.
It should be the shortest, and it is actually. the shortest.
Ma'am, logically it should be the shortest, but I have only presented the data which has been given by the center. So this is just, so I don't know the underlying reason as of now.
So you've worked with this registry for 2 to 3 years now. What suggestions?
From the start, ma'am.
From the start. That's how many years now?
2019 onwards, ma'am.
Yeah. So what would you suggest methods to kind of improve the traction of entry into the system?
So ma'am, yeah.
From my center, I know the reason why we have only so few patients, but I'm sure that reason is replicated across the country. So are there any mitigating ways?
Ma'am, there is one. I think initially we only had very few centers who had expressed interest, and we tried to capture data from all the centers we can, but it gets a little difficult considering that everybody is very busy and it's difficult to, it's an added task to enter more patients. But recently also, Government of India has allocated Center of Excellences, I think in 14 states as of now. So if we can, channel the entry of patients into the registry through those COEs, because another thing is that the rare disease policy, it runs through the Center of Excellences. So patients who are registered with these COEs will also get the benefit of the rare disease policy, which says that up to 50 lakhs can be given to each patient for their management. So I think that would be the way to go as of now. Thank you.
So thank you. Any other questions?
Thank you, ma'am. Next, we would request Dr. Joydeep Choudhury and Dr. Shamoranjan Pal to continue to chair the session on mycobacterial infections in IEI, experience from India by Dr. Vignesh P.
Hello. Wonderful session, and the carry home message should be brought forward and carried to home. Next speaker, let me introduce Dr. Vignesh P. He is additional professor of the Allergy Immunology Unit and the Department of Pediatrics, PGI MER Chandigarh, which I think is basically the most, I will say, sacred place of immunology in India. I think so. So from there we have Dr. P. Vignesh, and his topic will be on mycobacterial infections in immune deficient patients. So please proceed.
Thank you, Chairperson, for the kind invitation, and thank the organizers for having me here. So I'd be speaking on this topic, mycobacterial infections and inborn errors of immunity. So this would be specific for our country, that is India, where there is a universal BCG vaccination. So I have no conflicts of interest. So I'm going to talk about mainly these three disorders: the severe combined immune deficiency, chronic granulomatous disease, and the disorder of interferon gamma immunity, or the Mendelian susceptibility to mycobacterial diseases, where we see the mycobacterial infections. So let us talk about SCID, the severe combined immunodeficiency. So this is the multicentric paper published in the Frontiers of Immunology. This is the pan-Indian experience of severe combined immunodeficiency of 277 children with severe combined immunodeficiency. The most common opportunistic infections which we observed in this large cohort of patients is the BCG infection. It's BCG, and the most common pattern of infection is the pneumonia. So you could see BCG right here. I don't know, the laser pointer is not working. Okay. So more than the absent BCG scar, the presence of ulcer at the BCG vaccination site is a more specific marker. So if you encounter any infant with severe infection or recurrent infection, and if you see a BCG site like this, ulcerated scar, so this is a clinical pointer for severe combined immunodeficiency. Of course, they can also disseminate to other organs such as lymph nodes, liver, spleen, and other organs. Unlike the classical MTB spread, which happens from the lungs and then to the other organs, it is the lymph nodal spread which happens with the BCG infection. And usually the BCG infection, we see the presentation at around 1.5 to 2 months of age in children with severe combined immunodeficiency. And sometimes this disseminated BCG can also have a secondary hemophagocytic lymphohistiocytosis, even in patients with severe combined immunodeficiency. So out of the 94 cases with severe combined immunodeficiency, which we had at our institute, 6 had HLH-like manifestations at the early infancy. Most of them were X-linked severe combined immunodeficiency, and the HLH is specifically associated with the disseminated BCG infection. So if you encounter a child with HLH with a dirty BCG scar, it is the lymphocyte subsets which we need to look in these patients. So this is a patient with a RAG1 SCID who underwent a matched related donor transplantation, hematopoietic stem cell transplantation. And he did overall well, except for minor issues such as acute skin graft versus host disease, and he had hypertension related to cyclosporin usage. He fairly engrafted, but what happened over time, this is at the day plus 90, he developed a left axillary node enlargement, followed by a supraclavicular node that is enlarged, and this is the day 130. A white cheesy material is coming out of this, the left axillary lymph node. So what this child has developed is the immune reconstitution inflammatory syndrome of the BCG vaccination following a successful engraftment. So this child required a prolonged antimycobacterial therapy for this to get it cleared, and by day 200, the lesion luckily healed with a scar. So this is another important complication post-HSCT we see in these patients with severe combined immunodeficiency. Moving on to the chronic granulomatous disease, the NADPH oxidase, this is a very important molecule required for the production of the reactive oxygen species within the phagocytes to kill the microorganisms. And the alveolar macrophages are very important for control of mycobacterial infections. So NADPH oxidase is indeed very important for antimycobacterial immunity. So we have 6 different components of the NADPH oxidase complex: the GP91 FOX, the membrane bone complex. If it is deficient, that is the X-linked form of chronic granulomatous disease, whereas the defects in other forms of other components of NADPH oxidase complex, it leads to the autosomal recessive forms of chronic granulomatous disease. Now, before moving on to mycobacterial infections in CGD, you should remember the clinical disease of CGD per se is a close mimicker of tuberculosis. Misdiagnosis of children with CGD as TB is very common in our country because TB is very endemic in our country. The presentation of prolonged fever and pneumonia, if someone did a lung biopsy or a FNAC from the lung tissue, they can have granuloma, lymph node granuloma, and these tend to be mislabeled as a tuberculosis, and they tend to receive months and months of anti-tubercular therapy before a diagnosis of chronic granulomatous disease is established. These patients can also develop vertebral osteomyelitis and gibbous formation. This is the fungal infection which does that, but clinically it mimics a kind of TB spine involvement, and lymphadenitis in CGD can have a prolonged duration of drainage and sinus formation. So as I told you, empirical ATT initiation is very common in these children, and it frequently results in delayed diagnosis. So whenever you clinically suspect a TB but you don't isolate a mycobacterium species either by AFB smear or culture, NBT is the go-ahead test for you, and most of the children you will pick up the diagnosis. So coming on to mycobacterial infections in CGD per se, so this is the article published in 2013. We had 99 patients back then, out of which 22 had evidence of mycobacterial infection, 11 with BCG, and 11 with M. tuberculosis. So if you see the split of BCG infection, most of them had a localized BCG infection, but 5 of them had disseminated forms of BCG infection. And this dissemination, unlike SCID patients, they tend to occur late in these patients, not a very early form of disseminated form of infection or a fulminant BCG infection, which we see here. M. tuberculosis, mostly these are seen in older kids following exposure from one of the family members or nearby contacts. It could be a pulmonary or extrapulmonary form of infections. And it's important to note that a BCG first presentation, that is children who came up with BCG first infection, then diagnosed to have CGD, it is seen in 1 and simultaneous in 5, whereas 5 patients were initially diagnosed with CGD, later on they developed BCG infection. Similarly, for TB, difficult to treat TB, 2 patients were screened and identified to be having underlying chronic granulomatous disease, whereas 6 of them had simultaneous diagnosis of CGD and the diagnosis of MTB infection per se, whereas 3 were followed up as case of chronic granulomatous disease. They were diagnosed early, but over time in the follow-up, they developed Mycobacterium tuberculosis infection. The important point in CGD, the uniqueness of this radiological finding, the calcification of the left axillary node, it is due to the persistent infection and the inflammation that happens in the left axillary node, which results in this form of calcification. We haven't seen noted this kind of calcification in SCED or the MSMD cases so far, but we have seen in many cases with the chronic granulomatous disease, especially the X-linked forms of disease or the NCF2 forms of CGD. And this is a very important radiological pointer. If you encounter any child with pneumonia and if you happen to see the radiological finding of calcification in the left axillary node, CGD is one important differential which you should consider. So overall, the risk of having a BCG-related complication is 10 times higher compared to the healthy control. In healthy individuals, the prevalence of BCG vaccination complication is 0.16%, whereas it is 11% in CGD. And the tuberculosis, the incidence in healthy population is 0.19% in India, whereas in CGD it is 11%. So there is clearly a 10% higher risk of developing BCG as well as tuberculosis infection in chronic granulomatous disease. So whenever you encounter a difficult tuberculosis or a BCG-related complication, you should screen for a chronic granulomatous disease. Okay, so this is a 6-year-old girl with a multiple lytic bone lesions, a punched out bone lesion in the skull, was thought to be having a Langerhans cell histiocytosis. You can see a punched out lesion in the right humerus as well. And this is what the bone biopsy showed. It showed acid-fast bacilli, and on cultures and PCR were subsequently identified to be an M. bovis. So this child's immunoglobulin profile was normal, lymphocyte subsets were normal, and NBT-DHR was normal, not suggestive of chronic granulomatous disease. So what is the disease I'm dealing with here? So this is a Mendelian susceptibility to mycobacterial disease, and this is a classical presentation of a partial dominant form of interferon gamma receptor 1 defect. The flow cytometry is the investigation to confirm this defect or a molecular analysis. You see a very high median fluorescence intensity of the IFN gamma R1 expression. Because there is a problem with the receptor recycling, you get an increased expression rather than a decreased expression, which you see in a recessive form of interferon gamma R1 defect. So this is a partial dominant interferon gamma R1 defect. In India, the first case was reported in 2013. 2 publications, one from Dr. Rashid Merchant from Mumbai and the other one by Dr. Sheila Namputri from Kerala. So the first is the patient with the IL-12 RB1 defect, and the second one is the patient with IFN gamma R1 defect. So I would tend to call them as disorder of interferon gamma immunity, because there occurs a problem with the interferon gamma production, or there is a problem with the interferon gamma response, or you get problems with both interferon gamma production as well as response. So it revolves entirely around the interferon gamma. So the preferred terminology which most likely would come in future is the disorder of interferon gamma immunity, because they don't specifically predispose only to mycobacterial, they also predispose to other infections as well. So why do you identify these disease? Why it is important to identify? Because if it is a disorder of interferon gamma production, you can use interferon gamma as an adjunctive therapy, and several of these disorders can be cured with a hematopoietic stem cell transplantation. And of course, for family screening and avoiding BCG vaccination if you already know that infant is having the underlying defect. So mycobacterial infections in MSMD, they have a selective predisposition to clinical disease caused by a weakly virulent mycobacteria, either a BCG or a non-tuberculous environmental mycobacteria, not so much so with the MTB, the Mycobacterium tuberculosis infection. So this is the largest compilation of the IL-12 RB1 and IL-12 P40, the disorder of interferon gamma production. The most important infections are the BCG and the environmental mycobacteria, and M tuberculosis only in a few of these patients. In interferon gamma R defects and STAT1 defects, again you could see more of environmental mycobacteria and BCG infections which top the list in these disorders. So since ours is endemic for TB and BCG infection is BCG vaccination is universal, we tend to see a lot of unique phenotypes in this disease. The recessive complete deficiencies tend to present early in life, and recessive partial and dominant defects, they present later in childhood or even in adulthood, and the manifestations are diverse. If you come to the specific phenotypes, the complete IFN gamma R1 deficiency, they present with disseminated infections with the bone involvement, which is unique in the complete IFN gamma R1 deficiency. Whereas the partial dominant IFN gamma R1 deficiency, they have unique predisposition only to bones rather than other organs. The IL-12P40 or IL-12RB1 deficiency, they present with disseminated BCG in the infancy with lymphadenopathy or hepatosplenomegaly. If they escape disseminated BCG or if they don't develop a disseminated BCG, they tend to have a very good prognosis. So this is the frequency of bone involvement which you could see. If you see multifocal osteomyelitis in the Mendelian susceptibility to mycobacterial disease, most of the disorders what we see is the autosomal dominant IFN gamma R1 or the STAT1 defects. So this is the experience at PGI. This is one of the PhD thesis. The 59 clinically suspected MSMD patients were screened with a targeted panel and also with in whole exome sequencing in some 27 pathogenic variants, and out of which the IL-12RB1 is the common, followed by the other defects. So the patient cohort, it comprised predominantly of patients from North India, some from the eastern part of India as well. So if we talk about specifically BCG infections, out of 33 patients with MSMD who received BCG vaccine, 70% have evidence of BCG infection. And out of these 23, 13 had disseminated forms. And one patient with IFN gamma R2 and two patients with partial dominant IFN gamma R1 defect had dissemination to bones. And the median age of development of BCG site reaction was 3 months, and subsequent diagnosis of MSMD was 2 years, which is a delay, of course. And also note that if they develop disseminated BCG during infancy, especially with abdominal involvement with the abdominal vessel thrombosis, they have a worse prognosis. M. tuberculosis complex, we do see difficult to control M. tuberculosis also infections also in this cohort. 15 patients, out of which the predominant chunk was in the IL-12RB1 defect. Unlike the pulmonary tuberculosis, we see more of a disseminated form or resistant forms of tuberculosis in these patients. The common sites of dissemination was in the lymph node, followed by abdomen, CNS involvement, and renal involvement, bone involvement in some of them. NTM infections, we are not diagnosing this often, probably because of the lack of availability of the mycobacteriology expertise, because to isolate these organisms and to confirm it by PCR, you need to have a dedicated mycobacteriology lab. We did identify in 7 of them the multifocal bone infection due to Mycobacterium avium complex seen in a child with partial dominant interferon gamma R1 defect, meningitis due to Mycobacterium marinum in a child with ZNFX1, and Mycobacterium fortunatum and Mycobacterium saskatchewansi in a patient with IFN gamma R1 and IL-12B defects, respectively. So I'm going to show just the clinical pictures of how these infections could present in these patients. This is a patient who was thought to be having a chronic recurrent multifocal osteomyelitis, had a multifocal bone infection, and this is actually due to BCG, and he also had a non-typhoidal salmonella infection, and this is a child with a IL-12RB1 defect. This is again IL-12RB1 defect, the localized BCG site inflammation which progressed down to the lymph node. This another patient with MTB, he had a renal tuberculosis at the age of 12 years. 12 years is not the age that you develop renal tuberculosis. He had urine full of AFP. He had abdominal dissemination, he had pulmonary dissemination. He received, I think, 5 courses of anti-tubercular therapy before the diagnosis was made in this patient. The lower panel, it shows the leukocytoclastic vasculitis, which you could see in some of these patients. These vasculitis are usually triggered by salmonella in these patients, but if you have a BCGitis history, and if you have this kind of lesions developing in a childhood, IL-12RB1 defect is the one which you should think of. This is a child with an autosomal recessive interferon gamma receptor 1 defect, autosomal recessive. So this patient presented with the abdominal enlargement, hepatosplenomegaly, had multifocal bone lesion. The bone marrow showed a lot of histiocytic infiltrates, initially thought to be a histiocytic disorder. And it took at least a year to identify what was the microbial trigger in this patient. This patient had a disseminated Mycobacterium fortuitum infection, which is actually doing this, which mimic the clinical presentation of an histiocytic disorder. And this is again one another child who was thought to be having chronic recurrent multifocal osteomyelitis, received methotrexate, I think palmidronate. This is a patient with a partial dominant IFN gamma R1 defect. So this is very important for a rheumatologist. Whenever you see a lytic lesion or if you see a draining sinus, don't never ever think of a chronic recurrent multifocal osteomyelitis. Indeed, think of some infection, an atypical infection, and genetics would give you the answer in these cases. Again, a child with multifocal osteomyelitis in a child with partial dominant IFN gamma R1 defect. And this is a child with MDR tuberculosis who came to us in the early infancy. He had disseminated form of TB. In fact, what he had was not the MTB actually. Yes, I'm onto it. This is the penultimate slide. And to conclude, so what is important in this patient is, in this patient, the defect was identified by a Sanger sequencing. We had to do Sanger of all the exons of IKBKG to identify this variant, because this gene has a pseudogene you cannot pick up in a whole exome sequencing. The clue comes from the examination of the teeth, where you could see a conical incisor that is there in this patient. So to conclude, BCG vaccination site ulceration in an infant is an early sign of SCID. Disseminated BCG in early infancy, it is SCID or recessive or severe forms of MSMD. BCG iris, it is an important complication post hematopoietic stem cell transplantation in SCID. Localized BCG-itis with calcification in the axillary node, you should think of chronic granulomatous disease. An autosomal recessive IL-12RB1 defect is the most common form of MSMD with BCG infection. And disseminated BCG in infancy usually has a poor prognosis. If you have a multifocal bone involvement due to Mycobacterium species, think of IFN gamma R1 or the STAT1 defects. With this, I acknowledge my unit members, the funders, the Department of Health Research and the Indian Council of Medical Research, Government of India, Dr. Jimki Das, who is the PhD student who performed the functional assays for the IL-12RB1 and the IFN gamma R1 defects, and Professor Sudhir Gupta for mentoring, and to the patients, residents, laboratory technicians, and research fellows of our unit. Thank you so much.
Thank you. Thank you, Dr. Vignesh. It's fantastic pictorial presentation. It's really enlightening for all of us.
Thank you. So I think the carry-home message from this talk is basically essentially exhaustive clinical approach with a clinical bias helps to pick up the disease, because unless we pick it up early enough, so we miss the bus. So a good clinical acumen and working as a concerted team and supported by investigations and adequate treatment can only help the alleviation of this problem. So any questions from the house? 2 questions. Yes, madam.
That was a wonderful presentation regarding how important it is to diagnose examples like SCID, CGD, MSMD. How common is an inborn error of immunity in a child, in a young infant who is thriving well and has no constitutional symptoms but only has unilateral BCG adenitis?
Yeah. So thank you, ma'am, for the question. So localized BCG-itis could be an early clue for an inborn error of immunity. So if it is a disseminated BCG, yes, we do evaluate for inborn error of immunity. But if you have a child with localized BCG-itis, I would at least do a CBC to look if there is a lymphopenia, a lymphocyte subsets, and an NBT. At least these 3 tests, so that I'm not missing a severe combined immunodeficiency or a CGD, and have the child on the follow-up. If the child develops disseminated disease or if there is a positive family history, then yes, in future the child would need a more detailed workup.
Thank you. Sir, I would like to stick my neck out and say that in a normal infant, a young infant who is thriving well and has no constitutional symptoms, with just unilateral BCG adenitis, the commonest thing is that the child has no underlying inborn error of immunity.
I would beg to differ. I would beg to differ, having seen all these patients. I agree, most of them are healthy, but this could be an early chance to pick up underlying IEI. We don't have a newborn screen, but a BCGitis is a good screen to pick up an underlying IEI. So I would definitely do at least a baseline evaluation and keep these children on follow-up.
Sir, one question. In our immunization clinics of medical college, it is found that at the end of 5 weeks or 6 weeks, there is some not healing ulcer and there is a inflammation with 1 to 2 lymph node enlargement. Can we wait with antibiotics or search for this presentation like this?
I think the question is similar to what Geeta ma'am has asked. I would definitely screen at least for common IEIs, common and treatable IEIs, that is the severe combined immunodeficiency and CGD. I would rule out if they have a BCG site ulcer and a lymphadenopathy. And also take a detailed family history if there is any family history of PID in them.
But we have seen that this ulcer, after giving antibiotic, is healed.
It's good, they heal, but at least to pick the children early, I think this is the very important early clue for an underlying IEI. Probably if you screen 10, you would identify one.
We can go to Dr. Sumavalan, please.
Yeah.
Related infections and make it a notifiable thing so that when these infants are notified, then the pediatrician can be also requested to do the basic workup in these patients.
Yeah,
that would probably
I
be the way
agree.
to go.
I definitely agree.
Yeah.
Sir, you described quite a few CRMOs who ended up in IEI. So what was different in those CRMOs which made you suspect that it's not a typical CRMO?
So one is the presence of lytic lesions at unusual sites, multiple lytic lesions at unusual sites. Second, no much response to whatever therapy for CRMO that we are giving. So that is another one. So these are the two things which made us think we are dealing with a probable inborn error of immunity due to some unusual infection rather than just CRMO.
Sites as in what? It usually...
It is a similar site. If you apply your clinical criteria for CRMO, they will qualify for CRMO.
Dr. Suparna, and then one last one.
Wonderful please. As usual. So very basic question. So what these patients whom we are labeling as multi-drug resistant tuberculosis, MDR, should we get the genetics of these patients done also?
Definitely, definitely.
And the last one, please.
Ma'am, answering to your question, ma'am. Recently, we have seen a 12-year-old child. Child had a BCG local reaction, absolutely well for 11 years. And now the child developed a disseminated histoplasmosis. When we screened, the child was like having interleukin RB1. And the CRMO, recently we have seen managed as a CRMO. When the child came, when you put them naproxen, the child is not healing well. When we did a whole body MRI, the child was having multifocal osteomyelitis in an unusual area. The child is having a STAT1. So when you image the baby, if your musculoskeletal radiologist is very well enough, they can tell you this is not CRMO.
Yeah.
So
I think that's the key. Having a good imaging person and having a solid radar for knowing when it is not CRMO.
Yeah.
It's equally important.
Thank you so much.
1 second, Umair wants to tell something.
Okay, last question. So in original Dr. Casanova's publication of BCGosis and genetic etiology, there was a large chunk of DGeorge syndrome who had BCGosis. Did you find any DGeorge in your cohort?
No, sir. We haven't
Maybe
seen
we should
DGeorge.
look.
We have seen FOXN1 with disseminated BCG, but not DiGeorge so far. Probably we are missing.
Yeah, this session happens to be very interesting. I think you can address few more questions after you finish.
Thank you. Thank you so much.
Otherwise, we will go on and on. So thank you so much. You can interact, please, later with him. Thank you.
Thank you, sir. Up next, we have 2 oral paper presentations. The first one will be by Dr. Siddharth Jain from AIIMS Delhi. And I would request Dr. Surjit Singh and Dr. Lisa Rajashekhar ma'am to judge the presentations.
Good afternoon, everyone. First of all, thanks to ISPID and the organizers for getting this work onto the oral paper platform. So I'll be shifting flavors a bit to specifically adult PID, and I'll be sharing the experience from our center over the last 2 years. I am an assistant professor at AIIMS Delhi, and I am an adult rheumatologist and immunologist. So at the outset, I'll give you a disclaimer. It's very difficult to really present all of this data in 5 minutes. So what I've done is I've excluded the adult autoinflammatory disease data because we have heard enough about autoinflammatory diseases since the morning, and we'll probably be scratching the surface, but please pardon me for being hurried, but I have a lot to show, but 5 minutes would probably be too less. The background rationale is very clear. We all understand, even if we see the head count of the audience itself, adult PID is sort of an uncharted territory. Most of our PID centers, clinic registries are pediatric centered. There's a lack of awareness, especially in the adult side, with a very high threshold for suspicion. Published data is scarce, not just in India but also globally. Thus, a lot remains unknown about adult PID. So we just started this study out 2 to 3 years back, and the objective was to characterize the clinical immunologic genetic profile and the treatment outcomes of adults diagnosed with PID. This was a single center prospective cohort study. Over the last 2 years, included both adults and adolescents who were diagnosed with PID either immunologically or through genetic testing, meeting the ECID definitions. We captured data pertaining to demographics, clinical presentation, labs, genetic testing, treatment, and the outcomes. So results, so main thing, 98 patients were screened, 47 were confirmed to have PID, 28 were males, 60%. The median age at diagnosis was 24 years. The interquartile range as shown. Significant delay in diagnosis as expected, 4 years being the median, maximum being 12 years. The clinical presentation, because the kind of patients we have chosen at this point in time, was dominated by recurrent infections in 94%, which could be bacterial, mycobacterial, fungal, viral, parasitic, all over the board. There were a lot of autoimmune manifestations as well. We'll come to this when we discuss all of these in detail individually. And some of them had hematological malignancies along with them as well. So we had whole exome data for 87% of our patients, which showed that there was a pathogenic, likely pathogenic variant in about 39% patients. There was a VUS in another 39 patients, of which 44% were really compatible with the phenotype, and the whole exome was negative in the remaining 9 patients. So obviously the treatment was centered around antimicrobials, pathogen guided. IVIG was given in 25% of the cohort. There was immunosuppressive therapy requirement for management of autoimmune manifestations, and of course there was prophylaxis. We had planned HSCT for 2 patients, but in the adult side we did face a lot of challenges because when the infections were coexisting, it was really difficult to get them transplanted, and they often succumbed to the underlying infection. 17% of our patients died, most of them because of infective complications. The others are doing well under follow-up. So this is the division essentially of the data of our 47 patients. So the current understanding firstly is we feel that the adult immunodeficiency is predominantly secondary. Primary is probably not so common. That's probably a myth. Secondly, we tend to believe that the adult immunodeficiency is primarily antibody deficiency. That also I believe. After this data, we would probably be bursting some bubbles. So over the next 5 slides or so, I'll just give you a snapshot of our PID patients across the 5, 6, 7 categories, just to try to emphasize essentially, because I cannot give you details of these patients. Earlier I had presented, I had wanted to convey all the individual clinical data, but that's not really possible. But what I intend to show here is, so even SCID happens in adults. If you see the age of presentation of our patients, 34, 37, 24, this is the age at diagnosis. But the presentation also started for the first time in adulthood rather than it being a delayed diagnosis essentially. So they are starting out late. We are having JAK3 mutations, we are having NHEJ1 mutations, we are having Artemis mutations, essentially going into the domain of hypomorphic SCIDs. Some of them we had multiple VUSs which were not matching the phenotype, so whether that goes into the territory of late onset combined immunodeficiency or something that we are yet to characterize further is something time will say. As far as the category 2 is concerned, that is combined immunodeficiencies with syndromic features, even that was there in adulthood. We had, so I would not be discussing the data about hyper IgE because one of our residents is presenting that data as poster, and I would invite all of you for that. This is in the afternoon session from 3:55 to 4:25, post number 58. But we have STRAT3 variant hyper IgE in adulthood with predominant Aspergillus staphylococcus infections, Aspergillus fumigatus infections, absolutely nothing else, no constitutional delay, no pediatric infections. Some of them did not even have elevated IgE levels, thus really emphasizing that unless we keep a low threshold for suspicion, we might miss them out. There are some syndromic features in the form of STIM1 deficiency or I1 deficiency that also we found out. There was a patient I'd like to mention here, because when it comes to adult PID, there are a lot of times when before testing we think that this is where we are headed, but after testing our impression entirely changes. So this was example case being this patient of a 17-year-old male. He had a periodic fever-like illness. There was some enteropathic symptoms as well. So we had a suspicion of an autoinflammatory disease or an immune regulatory defect which was causing an immune enteropathy. Our immunological workup was normal. So when we did a whole exome, we found out that there was a pathogenic mutation in the ZBTB4, which is associated with ICF2. The patient did have some facial anomalies, but per se did not really meet the phenotype. So that is what we have, whether this is a novel presentation of the gene or this is an incidental variant, difficult to make it incidental because of the clear phenotype match. The patient is planned for segregation analysis and a whole lot of functional workup, which we are not presenting here. Antibody deficiencies, nothing very surprising there. We expect adult PIDs to have CVID, but what is important is even hyper IgMs are lingering around, CD19 deficiencies are lingering around. As far as immune dysregulation is concerned, we have that as well in the adult side. Apart from ALPS, which is pure lymphoproliferative sort of a presentation, we had CTLA4 haploinsufficiency cases as well. They had presented with very difficult to diagnose GI sort of diseases with the conglomeration of infective complications. So there was cryptosporidiasis, there was giardiasis, but even despite treating them, the GI symptoms had not resolved, but that was because of immune enteropathy, for which we had to immunosuppress, and then the GI symptoms got better. So again, an interplay of infectious and the non-infectious complications. CGD, understandably, we have discussed CGD in the previous talk, so I won't spend time there, but these patients can also have HLH, and even after treating the primary infectious cause of the HLH, they may still present, they may still persist, and they might need immunomodulation. We have CARD9 deficiencies, GATA2 deficiencies as well. Apart from the usual mycobacterial infections, the CARD9s notoriously have disseminated cladophorophora infections, either localized to the brain as brain abscesses or disseminated skin infections. Idiopathic CD4 lymphopenia, we cannot complete a discussion on adult PID without talking about that. But as we have discussed since morning as well, it's not a very well described entity. I think a lot of these are phenocopies or other diagnoses which we are not currently aware of. In adults, we also get a mixed bag sort of a presentation where, so for example, this patient was a 23-year-old lady. She had presented with histoplasmosis, disseminated TB. We were suspecting a cell-mediated defect. The NGS showed a JAK2 mutation. The immunological workup showed reduced NK cell function. The patient improved with antimicrobial therapy, but the point being, the NGS will not always give us the desired or the expected sort of a result, and we are keeping this patient under follow-up whether she develops anything new as well. This work would not have been possible without funding from various institutions. I had intramural funds, extramural funds from APLAR, and would not have been possible without that. The inspiration came from my time at PGI, where I was exposed to immunodeficiencies, and I started extrapolating it to adult side because that was fairly unknown. The strengths are, this is a large single-cell cohort focused on adult PID. We have whole exome data for most patients. Limitations, as all of you would understand, there is limited functional testing for VUS. We do not really have the capacity for testing
Dr.
phenocopies,
Siddharth,
which we are. This is the last slide, sir. And being a single center study, there is a potential for selection bias. Thank you. I would conclude here with my take-home messages, which I have somehow conveyed through my slides in the limited time. Sorry for overshooting. Thank you.
One question, Dr. Siddharth. How did you define severe combined immunodeficiency in adults? And which was the unit that referred you the most patients? Gastroenterology, pulmonology, or some other unit?
Sir, so these patients were addressed on consultation basis, sir. So we have a chunk of patients which would come from the gastroenterology side because of early onset IBD sort of presentation. That was one. From the pulmonology side, granulomatous ILDs, non-infective ILDs, we got a referral set system from there as well. So I would say, sir, fairly divided between gastroenterology, pulmonology, and probably hematology, sir. That is what I would say.
How did you define SCID in adults?
Sir, so that was actually a challenge. So from an immunological point of view, we would still go forward with the conventional definitions. Having evidence of combined as in T cell immunodeficiency as well as B cell immunodeficiency, and topping it up with a genetic confirmation is how we have currently defined SCID in adults, sir. As of now, when we haven't gotten a conclusive genetic workup, we feel that there might be an overlap with the entity of late onset combined immunodeficiency. So differentiating late onset combined immunodeficiency from hypomorphic SCIDs is a challenge which we are facing right now, I honestly don't know how to deal with that.
How would you want to take your work forward? It's in a short time, you collected a lot of cases. So what do you think you want to do?
So the plan, ma'am, is to actually go much deeper into these VUSs. So I had put it in my final take-home messages as well. So I think you would agree as well that from the adult point of view, the problem of VUS is even more significant. So unless we actually go down deeper at the functional level, we would not really be able to say whether these are novel phenotypes, novel variants. So essentially going towards that side, because I haven't presented the data of the autoinflammatory side, but in the autoinflammatory cohort, we have a lot of novel variants, novel phenotypes. Whether that exists in this side as well is something which would need functional testing. That is part one, ma'am.
Thank you.
And second is obviously deeper level genetic testing, family level genetic testing. At the government setup like ours, even while offering free genetic testing to relatives of patients with confirmed immunodeficiency, they would still have a reservation getting that done. So that's the plan for the future.
Thank you.
Thank you.
The next presentation is by Dr. Shomodeep Sarkar from PGI Chandigarh.
So I will present my case, the clinical and molecular profile of autosomal recessive CGD. As we all know that CGD is the prototype disorder of phagocytic dysfunction, and for the proper functioning of reactive oxygen species, we need a NADPH oxidase complex, which is formed of multiple subunits. The CYBB will code for the X-linked CGD, the NCF1, NCF2, NCF4 and CYBA will lead to the development of autosomal recessive CGD. The most common form of CGD is the X-linked CGD, whereas the autosomal recessive CGD accounts for 30 to 35% of CGD. But in Asian cohort, it is seen that autosomal recessive CGD is almost as equal as that of the X-linked CGD. And there exists a phenotypic heterogeneity in the patients of autosomal recessive CGD. So we took autosomal recessive CGD for this study. We aim to characterize the clinical and the immunological landscape, to delineate the spectrum of the disease between NCF1 and NCF2, and to perform a genotype-phenotype correlation. So the total sample size in our study was 59 patients. We did all the statistical analysis by SPSS, and the significance was taken as less than 0.05. The diagnostic workflow is as follows. In a patient of suspected CGD, we first did a NBT or DHR. If it was abnormal, then we went for a B558 expression. If it was normal, we went for a gene scan to rule out the pseudogene concept of NCF1. And if it was not present, then we went for an exome analysis. This is the DHR assay in which the neutrophils are stimulated, which will lead to the mean fluorescent index that on the MFI of stimulated divided by the unstimulated will give rise to the stimulation index for which is calculated for the control as well as for the disease. This is the image of the gene scan where we isolated the DNA and is amplified by PCR, and the fragment length is analyzed by the gel electrophoresis. And these are the curves which shows a homozygous variant in the left hand side and the control in the middle, and the heterozygous carrier having two peaks, but one peak is larger than the other. In few patients where gene scan was not done, we had done an ISC staining for P47 in some patients, which shows an absent staining in figure A. So with this, we went to analyze our results. The total number of patients of CGD diagnosed were 120, of which 49% of the patients were autosomal recessive CGD. Among them, most of the patients were NCF1, about 63%, and were NCF1. Among the NCF1, the most common were the C-del7576 GHT deletion in 32 patients, and in the NCF2, the most common genotype identified was the C835-836 del SE in exon 5. In the profile of the NCF1 patients, we divided it into GT deletion versus the non-GT deletion, which showed that there is a significant delay in diagnosis as well as the higher range of disease onset in patients with non-GT deletion of NCF1 patient. And the mortality rate in the NCF1 patient was 20.7%. This is the clinical profile which shows that the most of the infections was pneumonia, and the most common organism that was isolated was the Aspergillus species. And NCF1 also had a significant non-infective manifestation in the form of inflammatory manifestation. It ranged from colitis, uveitis, and lung granuloma. On diagnosis, we started the patient on antimicrobial prophylaxis that led to the significant decrease in the episodes of pneumonia. We tried to predict the mortality as well as the incidence of infection depending on the stimulation index. We did a Yonden index, and accordingly we opted to calculate the optimal SI cutoff, which showed area under the curve of 0.66 to 0.69 respectively, which showed a moderate predictive ability to predict the mortality and incidence of pneumonia in patients with CGD. In the patients of NCF2, we again divided into two spectrum of CDEL, 835, 836 deletion, and the other variant, which were comparable in two groups. The infections were mostly pneumonia, but mostly organism could not be isolated in the NCF2 patients, and the infection rates also significantly decreased. Again, we compared the predictive ability of SI index for mortality and incidence of pneumonia, which showed a very poor predictive ability for the NCF2 patients. Between the NCF1 and NCF2, we did the comparative analysis, which showed a higher median age of onset and a higher diagnostic delay than in the NCF1 patient than the NCF2, and the mortality was significantly higher in the NCF2 patients. So based on that, we did a Kaplan-Meier survival analysis with a log rank p-value of 0.008, which was significantly different in NCF2 versus NCF1 patients. Now we tried to plot all the patients of the autosomal recessive CGD across the globe of India, which showed that in Punjab and Haryana, there is only one mutation of NCF2, that is 835, 836 deletion in exon 5 was present. So that suggested a potential founder effect that is present in autosomal recessive CGD in this region. So in our cohort, mainly autosomal recessive CGD was present in 49%, NCF2 included 20 patients, and the mortality of NCF2 were higher in the NCF1 patients. The future direction in our study is the haplotype analysis and the population-based genetics in this study, and for the cDNA sequencing to identify novel variants in NCF1. Thank you.
Thank you, Samadip. I noticed that 2 of your patients had uveitis.
Yes, sir.
So how did you manage uveitis, and how did you manage liver abscesses in these patients?
Sir, the granulomatous uveitis that is present in CGD is a component of the inflammatory manifestation. It required the immunosuppressive agent in the form of steroids for a prolonged duration, and one of the patient also needed adalimumab also. So the inflammation was persistent. The hypothesis of inflammatory manifestation in such patients is that defective efferocytosis resulting in the defective clearance of the apoptotic cell that lead to the persistent inflammation in such patients. And liver abscess, sir, recently in a patient we had, for the liver abscess, we usually manage with antimicrobials only, according to the culture sensitivity. But recently we had managed, although the genetics is not proven, we had managed a patient of renal abscess in our unit. For that patient, we have used anti-inflammatory agents. Otherwise, for the liver abscesses, we have not used steroids.
You use steroids?
Yes, sir.
Yes, I'm just wondering why you excluded patients with X-linked in the comparison, because you have such a large group. You could have compared all 3.
Ma'am, actually, from our institute, already one publication is there comparing the X-linked CGD and the autosomal recessive CGD. We wanted to show the phenotypic heterogeneity in the patients of autosomal recessive CGD. And in this group also, we wanted to show that NCF1 can present as an older age, can have such varied manifestations. On the other side, the NCF2 will have much earlier onset, and aggressive treatment is needed for NCF2 patients more to prevent the worst outcome.
Do you have a reason in your mind why this, the simulation index was not indicative of mortality? And is there any other way which the sensitivity or it could, the AUC can better?
Ma'am, actually there are multiple confounders also, because severe infection, neutropenia in some patients, decreased neutrophil counts also contributed to the decrease in the stimulation index. Other immunosuppressive agents in some patients also lead to the decrease in the SI index. So predictability of the SI index in X-linked CGD is well described in multiple literature. But on our cohort, the SI index, although it predicted moderately the NCF1, but had no role in NCF2 in predicting the mortality. So maybe other factors that can that can influence the SI index, we can take that and can extrapolate the results. Thank you.
That was a wonderful presentation. If you would allow 30 seconds for observation.
Yes, ma'am.
You have excluded all individuals with a normal NBT and DHR, isn't it?
Yes, ma'am. Yes,
because that's where you started from.
Yes, ma'am.
I just would like to point out that way we would be missing NCF4 variants.
Yes, ma'am.
Because unless we look for the DHR with particulate inducers like Staph aureus E. coli, we could miss NCF4. And we were able to have one case that we recently published in Pediatric Allergy Immunology, where we used particulate inducers and found that the oxidative burst was reduced with them. Thank you.
Thank you.
Thank you. Thank you.
Thank you to all the participants. We'll now move ahead with the inauguration ceremony. For the inauguration ceremony, I would like to call upon the dais the President of ISPID, Dr. Amit Rawat, Secretary, Dr. Vignesh, the Treasurer, Dr. Umer Bagir, Organizing Secretary, Dr. Priyankar Pal, Joint Organizing Secretary, Dr. Suparna Guha. I would also like to call upon Dr. Shanta Datta, who is a scientist and former director of ICMR, National Institute of Research in Bacterial Infection, which was formerly called as NICET. We are joined with us today online by our chief patron, Dr. Sudhir Gupta, who could not be with us today physically, but he has graciously agreed to participate online. And along with him, we have our chief guest, who is Dr. Vishwa Mahun Katoch, who is also joining with us online. He is the NASI ICMR Chair on Public Health and Research at Rajasthan University of Health Science, Jaipur. I would request Dr. Amit Rawat to say a few words, please.
So I welcome you all to this inauguration ceremony of the 8th National Congress of the Indian Society for Primary Immune Deficiency. And I'm reminded of the first conference which was held in Delhi in 2011, which was the first international conference. And we have been doing these conferences annually, one international and one national. The national and international alternate each year, so they are biennials. And it is the first time that this conference has been held in the eastern part of the country, the national conference of the ISPID in Kolkata. And I am heartened to inform you that we had the maximum number of registrations and also maximum number of abstracts which had been submitted for this meeting. So it is indeed very heartening to see that the society has grown, and the interest in these disorders has also been increasing over the years. So with that, I would like to now request Professor Sudhir Gupta, who has been awake for, it must be late in the night in the US, to say a few words.
Vignesh,
Barger, Dr. Goa, and Dr. Datta, and esteemed delegates. It's indeed my privilege to welcome you all to PIDCON 26. I wish I could have been there in person, but because of certain unexpected circumstances, I have to be virtual with you. It has been 15 years since we had the first meeting, and what 15 years has been in India, it has changed the landscape of primary immunodeficiency diseases. And I think all the credit goes right from the beginning when Dr. Katoch and the health minister, Ghulam Azad, we had the meeting, and that was the beginning of establishment of primary immunodeficiency disease centers in India. And the major credit, we are just the catalyst for you. You are the people, the student, the residents, the researchers, the mentors and director of these centers who have really changed the face of primary immunodeficiency diseases, which we can diagnose prenatally and early postnatally and molecularly, And so it is really a pleasure to see how the advancement has been done. And this conference really provides a platform to share your research and foster collaboration among your colleagues, with the clinicians, and the researchers overseas. My gratitude and great sense of gratitude and thanks to overseas speakers. Some of them, like me, got up or stay late to provide you some eloquent talks. Dr. Holland, it was almost a midnight for him. For me, it is 1 o'clock here in the morning, but I wanted to be the part of it. And my special thanks to organizing committee, and especially Dr. Paul for their tireless effort to make this conference successful. My all thanks to all the sponsors as well, and I wish you all a very successful meeting. Jai Hind.
Thank you, sir, for joining us at such a late hour. I'd now like to call upon Dr. Katoch to tell us a few words, please.
Thank you very much. Respected Professor Sudhir Gupta and Indian colleagues, Professor Amit, Dr. Lisa, Dr. Vignesh, and of course Dr. Barbir, and of the organizing team, Dr. Priyankar, Dr. Guha. I think I see that Dr. Shanta Datta has also joined, my former colleague. And I think there will be many which whom would have missed seeing this time. Professor Sudhir also missed, I also missed. And I really join in, saying that what these 15 years have been remarkable. These are perhaps one of the rare success stories I've seen in my lifetime. And of course, all of us, we must reflect, this is driven by a single individual at that time. And he has continued to nurture it. Professor Sudhir Gupta's, your contribution can never be, I think, compared with anything. You have been the substrate, you have been the catalyst, both. But then the teams from Chandigarh, Mumbai, then followed up by Bengaluru, Chennai, Hyderabad, and then now many places. They have woken up and they have really joined, they have put in their heart, they provide the leadership. And when you see those multi-centric papers with this large number of authors dealing with a single problem, they tell the capacity actually. but this capacity has been built over a period of time. Professor Sudhir Gupta not only invested the money and the time, but he brought the people, best people from abroad also, who had the expertise actually. They are present even today. And then I think the local environment became so good that, it has changed. So many people are working on it. But I think I'll just one or two, three messages actually. See the success, how you consolidate it, how you bring it further. I think all of us must reflect one thing: the mutations alone don't decide the disease. It is the epigenetics, it is the other genes which are important, other factors are important. So it is a time to do now a science right at the field level here in India and in different settings. The second part is, that to disseminate anything. I think now because a large number of Indian data has emerged. At that time when Professor Sudhir was talking to you, when I prepared for the minister the talking point, we had a half a dozen papers only from India actually. It was a huge number actually, huge data. So this is the time to come out with, a teaching material, by this society. This society should be the official organ to produce the status papers. To those status papers guide of what we know today, so that, the other medical colleges, medical schools, which are not so strong actually, they join in. They start teaching by those ready reckoners actually. And then these become the policy briefs for the policy makers who have to invest money in it. I'm sure that money will come now. Earlier it was all the time used to be driven by the high courts or Supreme Court, but this will now can be a people driven. So our society has I think, huge role to play actually. And we look forward, that we are missing this time, but hope to be there the next time when we meet in India. And we continue to support the movement. I have been only a supporter, I must tell that part actually. Professor Sudhir has given me affection and perhaps a much larger credit, but I think a supporter. I continue to be a supporter, and I'm very, very happy supporter actually. So where I see the success right on the ground actually, is nothing theoretical, nothing is a big movement. So let this grow year by year, time by time, but we have to now show that concrete action. With these words, I'm very happy to formally inaugurated this, going so strong, such a large number of people participating. And so I greet everybody, and I wish you all the best, not for today, but in the coming time as we move on. Best wishes, Jai Hind. I'll invite, some of the CGD patients to light the lamp to show that we are relevant to all sections of the people who suffer from the PIDs. Thank you very much. Jai Hind.
We are joined with us today 3 patients of CGD, one of whom is Hardik Agarwal, who is also suffering from CGD but was successfully transplanted into 2022 and is now a proud class 8 going student. Then we have Rudronil Haldar and Ritika Haldar, who are also CGD patients who are awaiting their transplant. Please come on stage. We'll go ahead with the lighting of the lamp. They are our warriors right now, whom we have to take care of.
वक्रतुण्ड महाकाय सूर्यकोटि समप्रभा निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा गणपतये नमः। ॐ गणपतये नमः। ॐ गणपतये नमः। ॐ गणपतये नमो नमः। श्री सिद्धिविनायक नमो नमः।
We will move ahead with the lunch, and during the lunch time, we'll also have the poster sessions. Slot 3, 4, and 5. The judges, we request you to have your lunch and then we can move ahead with the poster sessions. Sir? Or it's, you may prefer to judge earlier and eat, or eat and then judge.
Finished, we are going to start the session.
Good afternoon, everyone. So after lunch, we'll be starting now. Our next session is on ALPS and ALPS-like disorders. So I would like to call upon stage Dr. Reena Gulati and Dr. Ajitesh Roy to chair the session. And the speaker will be Dr. Umair Bagel. Thank you.
Hello. Good afternoon, everyone. I hope hypoglycemia of the prior session is over now. Everyone is happy after lunch, and I hope more people come in quickly so that we can start the session. I am very pleased to introduce Dr. Umair Ahmed Bagher. Not that he needs introduction. I think he is known to most of the people around here. He is medical doctor, MBBS, DCP, and then he went in for his PhD. Currently, he works as Scientist C at the ICMR, National Institute of Hematology, Immunohematology, Mumbai. So, Dr. Umair, hand over to you, please.
Thank you, ma'am. So, at the outset, I would like to thank the organizers. Since this is a focus group meeting, we all know that what ALPS or autoimmune lymphoproliferative syndrome is. Not this beautiful mountain, but a shared puzzle of chronic lymphoproliferation with lymphadenopathy, splenomegaly, some autoimmune manifestations like Evans syndrome, ITP, and these may have some additional characteristic features. So comes a triangle, and we have to think about ALPS-like disorders along with autoimmune lymphoproliferative syndrome. So it's a diagnostic classification which is challenging but important for deciding the therapy of these patients. We all know that it is because of defect in the apoptotic pathway, and over the years around 5 different genes have been described to cause autoimmune lymphoproliferative syndrome, and one of the first Fas ligand deficiency, we were happy to be a part of that study. The diagnostic journey of ALPS begins early in the 1950s, where the Evans syndrome was first described, and then in the 1967 the clinicians identified chronic lymphadenopathy that mimicked malignant lymphoma, but it was non-cancerous, and it was labeled as the Cannell-Smith syndrome. In the early 1990s, a genetic basis for this lymphoproliferation was identified because of a defect in Fas gene. In the early 2000s, when Oliveira et al. From the NIH group gave a classification for autoimmune lymphoproliferative syndrome, that became the gold standard. And in the 2030s now, after many years of experience with ALPS and the exposure to next generation sequencing, we have a big umbrella of ALPD. ALPD is the autoimmune lymphoproliferative immunodeficiencies, and these are the mimics of ALPS, where the clinical phenotype is the same, but they need differentiation because targeted therapies can be used. So in the 2000, this is the classification for autoimmune lymphoproliferative syndrome and chronic lymphadenopathy with elevated double negative T cells. If these two are present, then it was diagnosed as ALPS. But elevated double negative T cells is a non-specific marker, and it can be elevated in other autoimmune diseases like SLE. Also, the cutoff of more than 2.5% TC alpha beta positive is not sensitive. Apart from that, there are different functional assays like the apoptosis assay, the deep somatic sequencing for these genes, and the serum markers like the serum Fas ligand levels, IL-10 levels, or with vitamin B12 levels that do help. Apart from that, there are classical immunohistologic findings like the expansion of the paracortical zone that can give a clue towards the underlying diagnosis. But all these points had some of the other limitation, and because of this, in 2023-24, the French group, German group, and the NIH, they came together to expand the umbrella from the autoimmune lymphoproliferative syndrome to include autoimmune lymphoproliferative immunodeficiencies. Now, that is contrast. We always say that primary immunodeficiencies became inborn errors of immunity because of lymphoproliferation, and here we are talking about lymphoproliferation with dysregulation leading to immunodeficiencies. Why is it so? So, a big 14-year-old retrospective cohort was published last year from the French group. We at our center tried to study the link between the autoimmune cytopenias and benign lymphoproliferation, and Dr. Priyanka Setia, who just finished her PhD and has moved on to study the ALPS further in France at Frederick's group. So what we did was, we evaluated all the patients who were fulfilling the characteristic Oliveira et al. Definition, and today we will be comparing the findings from our cohort and the big 14-year-old prospective study that was published in Lancet Hematology. So you can appreciate that this was a 14-year-old cohort with around 400 patients. In our cohort, we had 31 patients, and 50% of these patients had consanguinity. The median age of presentation was around 4 years of age, but the age of diagnosis was 8 years. As expected, lymphoproliferation, that is lymphadenopathy and splenomegaly, was the common clinical manifestation. But as you can see here, the pointer is not working. But as you can see here, apart from lymphoproliferation, the other manifestations were recurrent infections like diarrhea, failure to thrive, lower respiratory tract infection, and inflammatory bowel disease. The same was also reported in the ALPID cohort, where they have other infectious disease, malignancy, and other syndromic features apart from the classic manifestations. So the classic manifestations include lymphoproliferation with cytopenias, without cytopenias, only cytopenias in one or multiple lineages. In our cohort, apart from the lymphoproliferation, the infection was also a major manifestation. But as you can see here, it is a common manifestation in the ALPS-like cohort more than the ALPS category. So basically what we did was we thoroughly evaluated these patients. We did their complete blood count, flow cytometry to look at the double negative T cells, the TB and the NK cells. Then there are many functional studies that we will see that we have done to support the diagnosis, and of course the biomarker evaluation using these different cytokines. So basically what are double negative T cells? They are called as double negative because conventionally you expect our T cells to be either CD3, either CD4, that is the helper T cell, or the cytotoxic T cells. But these cells, they are not CD4 positive, not CD8 positive, but they are TCR alpha beta positive cells, and they are generated because of the defect during the T cell development. So here you can appreciate that apart from the classical markers used, we have used special markers like V220, CD57, that is the different CD57, CD27. These are the different markers evaluated, and an additional immunophenotype of double negative T cells was characterized as has been explained. So what are these fast controlled double negative T cells? So these are the defective double negative T cells that have a defect in their apoptosis pathway, and these are characterized by the positive expression of CD38 and CD45RA. So these cells are sensitive for the diseases caused due to a genetic defect in the classical ALPS related gene. Apart from that, we also do a functional assay where, as we all know, that the apoptosis pathway is defective and it can easily be evaluated using flow cytometry. So coming on to the spectrum, these are the genes that are commonly seen in autoimmune lymphoproliferative syndrome, the FAS ligand, FADD, caspase-8 and caspase-10. Apart from that, the two very common mimics that you might have encountered is the CTLA4 deficiency or the LRBA deficiency. And here you can appreciate that B220 is a marker that is sensitive to detect the FAS and the FAS ligand type of autoimmune lymphoproliferative syndrome. The FCDNT, that is the fast controlled double negative T cell, is a very simple flow cytometry based assay that can be performed at almost all the centers that perform flow cytometry, and a marked increase of these cells is very sensitive to diagnose all these three types of ALPS, the FAS ligand and the FADD. We were unable to perform in caspase-8 and 10, but in the patients in whom CTLA4 or LRBA deficiency was there, their levels were not abnormally high. So basically the big ALPID cohort and in our cohort we have evaluated these patients for different surface based flow cytometry markers, and the fast controlled double negative T cells characterized by CD45RA and CD38 positive, they are very sensitive to pick up the classical autoimmune lymphoproliferative syndrome. And even if the double negative T cells are not normal, there is a possibility that there can be caspase-8 or caspase-10 defect. The other immunological abnormalities in our cohort was that of the ALPS-like cohort, around 25% of these patients had hypogammaglobulinemia, low class switch memory B cells. Some of them also had lymphopenia, which was more common in the ALPS-like cohort, whereas in the ALPS patient it is less likely to have. Some of them also did have low naive TH cells. Coming to the genetic architecture, the challenge with our patient is that consanguinity is predominant, and in our cohort around 50% of the patients were born out of consanguineous marriage. So in the molecular spectrum is different for the FAST for the Indian cohort versus the Western cohort. Here you can appreciate that the FAST mutations is a single common category, and the other mutations that cause ALPS-like cohort, they form around 50% of these patients. Whereas in our cohort, it is a distribution of FAS ligand, FADD and caspase-8 and caspase-10. So these are the other inborn errors of immunity that have been reported by the ALPID cohort, where you can see a big number of genes that are responsible for conventional other dysregulation or immunodeficiencies, including HLH, combined immunodeficiencies, autoimmune lymphoproliferative syndrome, and even CGD. ALPS-FAS is one gene that has multiple inheritance pathway, and because of the involvement of this gene, there is a defect in the apoptosis. It can have a dominant negative effect where there is no apoptosis, or it can have a haploinsufficient effect where the apoptosis can be deranged. So this gene involves these different domains, and the dominant, if the mutation is in the dominant negative domain in the intracellular domain, then it will mostly cause dominant negative type of phenotype, and if it is in the transmembrane domain, it is most likely to cause somatic mutations. So looking at the location of the variant in the gene also is helpful to know which type of autoimmune lymphoproliferative syndrome it can cause. In our spectrum, we identified most of the mutations in either the intracellular domain or the death domain. One of the patients recently, Suma ma'am had referred, so this patient had characteristically elevated FC controlled DNT cells. The patient had already undergone an exome sequencing and no variant was identified. But because the FC DNT cells were elevated, then we went ahead and did a deep sequencing and a somatic fast variant was identified in the patient. In the ALPS-like disorder, a big list of other genes is present, like in the Western cohort, and in our cohort, the LRB and the CTLA4 deficiencies predominate. Apart from that, there are few with classical hypergammaglobulinemia that cause monogenic common variable immunodeficiency disorder, and some of these HLH patients also do present with autoimmune manifestations. So, in a wide varied cohort of autoimmune lymphoproliferative syndrome like diseases, it is very important to use optimum functional assays so that we differentiate and we validate the variants of uncertain significance. So of these defects for autoinflammatory diseases, since morning we have been hearing as to what all functional assays can be performed. For regulatory T cell defects, it can either be an LRBA deficiency, CTLA4 deficiency or STAT3 deficiency, which can easily be performed using flow cytometry. For classical ALPS based assays, apoptosis assay can be performed. For cytotoxic T cell defects that cause that cause hemophagocytic lymphohistiocytosis, we have a very robust functional assay, that is granule release assay. And for other innate immune defects, one patient had Mendelian susceptibility to mycobacterial disease with autoimmunity. There are ready flow cytometry based assays. So just to give you an example, LRBA is a very big gene. The problem in this is the antibody that is developed against LRBA, it binds only to select exons. So this functional assay cannot be performed in all LRBA deficient cases, but only in a select cohort depending on where the variant is. CTLA4 haploinsufficiency can be easily picked up using flow cytometry. But the practical challenge here is many of these CTLA4 expression is observed on the regulatory T cells, and many of these patients have very low number of regulatory T cells, and that becomes a practical challenge in terms of interpretation. Apart from that, CD25 deficiency is a simple assay using flow cytometry, and STAT3 can be assessed by using flow. So for other one of the other cases had MACT1 deficiency leading to X-Men syndrome, and we can easily appreciate the NKG2 expression by flow cytometry. And for PIK3 delta, we can perform apoptosis assay, and these patients also have an elevated TEMRA population, that is the effector memory population. So the diagnostic standard is, after suspicion and ruling out malignancies in patients with lymphadenopathy and cytopenias, the initial screening can be done using the biomarkers along with the molecular confirmation by either targeted sequencing when we are suspecting somatic mutation or when the whole exome sequencing is negative. The problem with our cohort is the Indian patients, they have, many of the patients have nutritional B12 deficiency, so it becomes a challenge, and the levels can be masked because of vitamin B12 deficient patient. So comparing the ALPS and the other common ALPS-like disorders, apart from the overlap in the clinical features, the other immunological findings that can be seen are elevated CD21 low B cells, and these CD21 low B cells can be expanded because of the lymphoproliferation or splenomegaly in not only CTLA4 and STAT3 gain of function, but also in other chronic common variable immunodeficiency disorders. The CTLA4 deficiencies and the STAT3 gain of function, they can also have enteropathy apart from the classic autoimmune manifestations. So the classic ALPS patient, they are usually healthy looking children with nodes, multiple nodes, can be small, can be big. The non-ALPS type of lymphoproliferation, they are usually sick, may have failure to thrive and have a very complex immunodeficiency along with the lymphoproliferation. In the ALPS cohort, apart from the caspase 8 and 10, infections are not common. But in the ALPS-like cohort, there are severe infections leading to recurrent viral and bacterial infections. Conventionally, ALPS is associated with elevated immunoglobulins, that is IgG and IgA, whereas hypergamma is commonly seen. The APDS spectrum of these patients have elevated IgM levels. And other organs are also involved in the ALPS-like cohort. So, a CTLA4 or a LRBA deficiency, it has enteropathy and hypergammaglobulinemia apart from the lymphoproliferation. STAT3 gain of function, STAT3 can have loss of function, gain of function, and these gain of function patient, they have early onset multi-organ autoimmunity, can have diabetes and eczema. The APDS group, they have a classic CVID-like presentation, can have bronchiectasis and even certain viral infections. So basically sometimes the biomarkers are consistent and we can go with targeted sequencing or increased depth sequencing, but when the biomarkers are not consistent and we are not sure whether it is ALPS or ALPS-like disorder, then broader panels and a conventional whole exome sequencing can be done. So if biomarker is suspecting, like if suppose the fast control DNT cells are elevated, then deep sequencing has to be performed, or we can sort the double negative T cells and perform a targeted sequencing. Whereas in other cases, if we don't find the mutation, we have to go ahead with a whole genome sequencing and look for structural variant. So why is it important to differentiate ALPS and ALPS-like disorders? Because there are targeted therapy available that will help the patient. So the ALPS patient with defect in the fast pathway, they can benefit from sirolimus. Whereas the CTLA4 and the LRBA deficiencies, these patients have targeted therapies like abatacept, which is not available in India, but it is a wonder drug for these patients. The APDS patients with PIK3 delta inhibitor respond well to leniolisib. Again, this drug is not available in India. And for the STAT3 gain of function, JAK inhibitors work wonders. So ALPS generally have good survival, but they are at an increased risk to develop lymphoma. Whereas the non-ALPS or the patients that fall under the ALPID category, they are at higher risk to develop permanent organ damage, severe infections, and transplantation definitely needs to be considered. So now when the patient needs to be worked up, we can't label the patient as autoimmune lymphoproliferative syndrome, but rather the autoimmune lymphoproliferative immunodeficiencies, especially when we are not sure about the diagnosis. And the diagnosis of ALPS can be used only when there is defect in the FAST pathway. That can be easily evaluated using multiple flow cytometry based assays. The serum FAST ligand levels, they can be used to triage vitamin B12 levels, though a challenge, but it is easily available to almost all of us, and it can be used for diagnosis of these patients. We need to hunt for somatic mutations when these fast control double negative T cells are high. They are very sensitive to pick them up, and we need to move from empirical steroids to targeted therapies wherever possible. So this beautiful mountain range is not only Alps, but a mixture of three. So basically you have Alps, you have the Mount Fuji, and you have the Mount Everest. So it is sometimes difficult to differentiate them, but with the help of proper investigations, we can diagnose the classical ALPS and separate them from ALPS-like disorders. So with this, I would like to thank the patients, all our clinical collaborators, and Dr. Priyanka Setia and our team, and my mentor, Dr. Manisha Matkaikar, for this work. Thank you.
Wonderful, Dr. Umair. You have given in a very concise and a clear way a topic which is otherwise a huge topic and difficult to understand, especially by the youngsters. Thank you so much. Any questions from the house, please?
One question for the resident and for like our adult physicians. So when do I suspect, particularly from my experience, we have seen one case in a corporate hospital when I'm an endocrinologist, I got a referral for diabetes management. But patient has been diagnosed with Hodgkin lymphoma initially. But finally the diagnosis came out to be a case of ALPS. I can remember only one case. Or for the juniors, what should be the most important red flag signs or sometimes something like that, that you can think that it is something else, not the only lymphadenopathy with some lymphoproliferative disorders.
Multi-organ autoimmunity is the sign where it must be evaluated for these, because it is not ALPS now. It is a big umbrella of ALPID. So basically all the cases we need to do a basic evaluation, because there might be certain underlying immunodysregulation and we can target the therapy accordingly. That is why it is important when we have multi-organ involvement. Right. So here the patient had lymphoproliferation with autoimmune, probably autoimmune thyroiditis, and these kind of patients we need to evaluate to look for the underlying ALPIDs.
Right. Thank you.
Thank you, sir.
Any other question from house?
I had one question.
Yeah.
Sir,
please.
So I'm Rajat, I'm a pediatric hematologist in Calcutta. I have a kid with a ITP with DCT positive lymphoproliferation and hepatosplenomegaly and raised double negative T cells of 4.5%, but whole exam is negative. So right. I was calling it ALPS till yesterday, so I'll call it ALPID now. Question is, how do I take it forward to make a diagnosis and put on immunosuppression? Right. Because I don't have a genetic diagnosis, but I am pretty sure this is ALPS.
So one limitation with interpreting double negative T cells is not all commercial labs, they do TCR alpha beta positive double negative T cells. What they might give you in the report, if you see the report, it is CD3 positive, CD4 negative, CD8 negative. And many of the, at least the labs who don't specialize in immunodeficiencies, they label them as double negative T cells. The normal range for these type of double negative T cells is usually around 10%. If it is a alpha beta positive population, then we can easily do FCDNT cells, and if they are classical for the ALPS, then we should do deep sequencing and look for somatic variants in FAST pathway.
So deep sequencing is the answer now.
Yeah, so flow cytometry based assay is ideally an easier first approach to look at the FCDNT cells and then followed by deep sequencing. Thank you.
Any further question?
Use vitamin B12 in diabetes. Particularly after 5 years, we get diabetes, vitamin B12 and others given. So we not find any hypervitamin B12. Any case hypervitamin B12, if any patient find that due to this prolonged daily they are taking vitamin B12. So if we see the hypervitamin 12, can we further investigate?
So your question is, if we do see increased B12 level in this type of patient, what should be our next step?
Yes, sir.
Right.
So it is not specific, like increased only with diabetes.
Yes.
It is very difficult to evaluate all diabetics patient for ALPS, but there are other clinical manifestations. You have chronic lymphoproliferation, lymphadenopathy, splenomegaly, and with other autoimmunity, then we can suspect. But only with diabetes, with hypervitaminosis, it is very difficult.
I have never seen this. Sure. Thank you, sir.
Okay.
Thank you, Dr. Thank you, sir. Our next session is on hyper IgE syndrome by Dr. Biman Saikia, and I would like to request sir and ma'am to continue to chair the session.
Good afternoon, everybody. So the next session is about hyper IgE syndrome. In our 20 days, we often do conclude that any patient presented with recurrent abscess, recurrent skin abscess, that means we have to rule out hyper IgE syndrome. But I think this is a very simplistic term. So we should know from Sir, Dr. Biman Saikia. I think he needs no introduction. So over to Sir.
Thank you, chairpersons, and a very good afternoon to everyone. And at the outset, I'd like to thank the organizing team for giving me this talk. And I've been talking on hyper IgE syndrome since a very long time. And since the time I started, in fact, when I was asked what is the topic, it was like hyper IgE syndrome. If you are not talking on anything else. So yeah, it's been almost 20 years that I've been working on this topic. And Dr. Sujit Singh was the one who, at the initial stages of my training as a faculty member, he introduced me to this subject and said that go and work on this. So hyper IgE syndrome is a rare inborn error of immunity. It's called a clinical triad of cold skin abscesses, which are because of staphylococcus, with pneumonia and pneumococcal formation, and chronic eczematoid dermatitis. So that's the classical triad that we know about. And associated eosinophilia, the patients can have mucocutaneous candidiasis together with other manifestations, like they can often have arterial aneurysm, too rare, some viral infections. And the other thing is, these are the immunological manifestations. The other thing is the skeletal and the craniofacial anomalies that these patients often have. The coarse faces, ocular hypertelorism, very prominent mandible, bushy eyebrows, prominent forehead, and dental anomalies because of retained primary teeth or non-shedding of the primary teeth, and craniosynostosis and recurrent bone fractures to trivial trauma. So it's a combination of both an immunological manifestation and non-immunological manifestation in the form of skeletal anomalies, and that talks of a link between the immunological and the skeletal system. So it was described way back in 1966. Thank you. I have a bad throat, and way back in 1966, and it was termed as hyper IgE syndrome when IgE levels were found to be raised in these patients in 1972 by Buckley et al. It was after a very long gap in 2007 that the genetic cause of this disorder was discovered by two groups simultaneously, Meinegishi and Steve Holland, in 2007. That was followed by the Dockate in 2019, and this subsequently IL-6-ST was one of the major discoveries in 2017, 10 years after STAT3 was described. So Dockate is no longer considered hyper IgE syndrome. It's considered as a combined immunodeficiency. It has been removed from the hyper IgE syndromes. So, as I said, it was in 2007 that the first, the STAT3 was first described as dominant negative mutations in the STAT3 gene, which causes hyper IgE syndrome. Dominant negative means the mutant allele suppresses the normal allele to cause the disease. So it's a dominant condition, and in a heterozygous condition, the disease will occur. So we know that now STAT3 is important for the formation of the Th17 cells, and that now becomes one of the markers for diagnosing hyper IgE syndrome at the STAT3 related. So reduced Th17 cell numbers along with reduced memory B cells and an abnormal phospho-STAT3 assay will clinch the diagnosis of a STAT3 related hyper IgE syndrome. So these are the clinical characteristics. As I said, the very typical faces with broad nose, a very prominent mandible, and bushy eyebrows, and these are the typical faces that you see in these patients. And these patients, the patients are said to be, they look different from their siblings, so because of the characteristic faces. And they can have double row of teeth because of non-shedding of the primary teeth, pneumonia with pneumococcal formation, retained primary teeth, craniosynostosis, which means the sutures they fuse prematurely, and then recurrent fractures. And they will have a low number of Th17 cells, which if you define as CD4 positive, interferon gamma positive, IL-17 positive Th17 cells, you'll see that these patients will have a low number of Th17 cells, and as well as low memory B cells and reduced phosphostat-3, phosphorylation of STAT3. But phosphostat-3 assay, maybe even in a mutation-proven STAT3 hyper-IgE syndrome, the phosphostat-3 assay may still be normal. So if it is not abnormal, it's okay. But it's normal, you cannot rule out the disease. Okay, these patients can have abscesses. This was a patient who had a sternoclavicular and a psoas abscess with retained primary teeth. So to diagnose this patient, we often use NIH scoring system, which we have the clinical features and we give the score to each of these clinical entities and features here, and a score more than 40 will say, indicates that this patient has hyper-IgE syndrome. So the diagnosis of autosomal dominant hyper-IgE syndrome basically are depending on this clinical findings, which many of these are actually complications. For example, the fractures and the new metaphyseal formation. These are complications of the disease, but milder and incomplete phenotypes can also. Now we understand that with the description of the IL-6 ST or the GP130, now we know about this axis and how it works in hyper-IgE syndrome. This biallelic mutation in IL-6 ST was described in 2017, and this patient had typical features of hyper-IgE syndrome with the skeletal manifestations, which indicates that it was related to STAT3, and we know that the GP130 or the IL-6 ST works upstream of the STAT3. So this is the pathway this actually is working in this STAT3-related hyper-IgE syndrome. We have this IL-6 ST or the GP130, which is common to the IL-6 receptor as well as the IL-11 receptor. So if there's something wrong here, both the IL-6 and the IL-11 receptors are not working. IL-11 receptor, the mutations in the IL-11 receptor are shown to lead to, craniosynostosis with dental anomalies, but the immunological features are absent with the isolated IL-11 receptor. Only the skeletal features are seen, whereas in pure IL-6 receptor deficiency, only the immunological features are seen. So that explains why when there's a mutation in the GP130, because both the IL-6 and the IL-11 are not able to function and phosphorylate the STAT3. So the combination of the skeletal as well as the immunological features in STAT3 and GP130 related hyper-IgE syndrome. So in the latest classification of the iOS classification that was published in this, I'm showing this slide to bring to your notice this journal, Journal of Human Immunity, which has recently been started. And in the first volume and the first edition, this update was published. This journal is totally dedicated to PIDs, and I encourage all of you, if you want to publish, use this journal, try to publish in this journal because it's dedicated to PIDs. So if you go to the iOS classification, the STAT3 related hyper-IgE syndrome will be more than 80% of all the hyper-IgE syndromes will be related to the STAT3 with low Th17 cell numbers, reduced memory cells, and normal or impaired phosphostat3. IL-6 receptor deficiency is autosomal recessive. Here the Th17 cells will be normal and no skeletal or facial features will be there. Only the infectious complications without the skeletal manifestations. IL-6 ST, again, both autosomal recessive and autosomal dominant forms of IL-6 ST are known. And then ZNF341 deficiency is another cause of hyper-IgE syndrome, and the lately described autosomal dominant gain of function mutations causing hyper IgE syndrome. So these are some of the publications from our center, and this was the multicentric study that we published in the Frontiers in Immunology of 104 cases of hyper IgE syndrome, and these are the spectrum of mutations that were seen. This is the what is known, already published, and this is our cohort. So we had mutations in predominantly the DNA binding domain and the SH2 domain, but we also had linker domain and coil domain mutations. Linker domain and coil domain mutations are not too common. So we had patients in this, and this patient was specifically very interesting because this was a deletion here. So because of the stop codon here, the linker, the SH2 and the transactivation domains were deleted in this patient. So we thought, because otherwise it's a dominant negative kind of a mechanism, but in this patient, since this part of the allele was absent, so we thought that it could not be a dominant negative, it should be a haploinsufficiency. So this topic of dominant negative versus haploinsufficiency has been there for some time. This article in 2021 had a huge number of cases, 100 plus cases, and all these cases, that particular patient with the deletion was included in this study, and it was shown that all were dominant negative and none of them were haploinsufficiency. Even in the patient that we had with the deletion, we thought it would be haploinsufficiency, but it was shown that there was an alternative splice variant in that particular patient because of this mutation, which was actually dominant negative. So we didn't know whether it was haploinsufficiency or dominant negative that is working. But this publication, which came in 2025 by Bodo Grimbacher's group, they irrevocably showed that dominant negative, haploinsufficiency can be a mechanism of disease in hyper IgE syndrome, STAT3 related. So these patients, the treatment of these patients more or less revolves around managing the infections using prophylactic antibiotics with or without antifungals. Corticosteroids may be prescribed to manage eczema like skin inflammation, and drugs like anti-IL-5 and anti-IL-5 alpha has been used to manage the allergic and the eosinophilic inflammation. More important part is the genetic counseling and psychosocial assistance and regular monitoring of the disease progression, the immune status, and the potential complications is the vital part, and supportive care for recurrent skin infections, pulmonary complications, skeletal anomalies, and growth delays are important. Now, my team have been interested in more in the craniofacial and the skeletal anomalies. Why these patients with the STAT3 defect would develop craniosynostosis and retain primary teeth and fractures. What is going on, and how is the skeletal system related with the STAT3 gene? So we know that the sutures, the maintenance of the sutures is a interplay between the osteoclastic and osteoblastic activity, and the very active osteoclastic activity, so that the whatever bone is laid down is removed by the osteoclasts, is very important to maintain the homeostasis and maintaining the sutures. So any abnormality or any the imbalance in this osteoblast thing could lead to craniosynostosis. Similarly, reduced osteoclastic activity where the apical tooth is not resolved would lead to a retained primary teeth. So again, indicating that the osteoclasts may be abnormal, may be responsible for retaining the retained primary tooth and the double row of tooth that we see in these patients. So when we started our work and to see what could be the reason behind the skeletal manifestations, we came across, we scanned multiple genes that were actually involved in the skeletal system, and we came across osteopontin as one of the genes that was markedly reduced in patients with hyper IgE syndrome compared to healthy controls. And we had published this. This was a PhD thesis done by Shubham. So he's sitting right in front of me. This was his PhD work. And we found that osteopontin was actually dysregulated in patients of hyper IgE syndrome. And the osteopontin gene actually had STAT3 response elements, which was a very new finding in that particular study. And osteopontin dysregulation was shown in one study even before STAT3 was actually described as a cause of hyper IgE syndrome. So there was some link between osteopontin dysregulation and hyper IgE syndrome. Then we started looking at the osteoclasts. We generated osteoclasts from the peripheral blood, mononuclear cells in patients of hyper IgE syndrome, looked at various gene expression profiles. We knocked down the STAT3 by using static and looked at the differences of the gene expression in STAT3 knockdown and compared to the healthy control. So we showed that the fold change expression of various genes like the RANK, the TRAP, MMP9, SOX3, which are very important in skeletal development, where whenever STAT3 was knocked down, these genes were kind of dysregulated. And by using these mutant plasmids in patients and compared them with patients, we showed that the fold change expression of these particular genes, RANK, MMP9, and osteopontin, STAT3, and SOX3, they were different in the mutants versus the wild type here. So there was something wrong in the osteoclast activity in these patients, and so a restricted expression of MMP9 and osteopontin, which are required for bone resorption in the hyper IgE patient-derived osteoclasts, so confirmed that the osteoclasts in the patients with hyper IgE syndrome were hypofunctional. They were not doing their resorption activity as good as their normal counterparts. But the surprise came after that in 2021, when a group from China, which were working on similar lines for a very long time, they showed that the defect in hyper IgE, STAT3-related hyper IgE syndrome does not lie in the osteoclast, but it lies in the osteoblasts. By knocking down the STAT3 gene in osteoclast purely by Kree-lock mechanism, they showed that it was the osteoblasts which were responsible and not the osteoclasts. So we shifted gears. We now wanted to look what's happening to the osteoblast and the crosstalk between osteoclast and osteoblast. We recruited 9 more patients here with craniofacial and the dental anomalies. And then we proceeded. We isolated the osteoblasts from the mesenchymal stem cells from the peripheral blood in these patients. So these are the mesenchymal stem cells that were isolated at day 21 from the peripheral blood and these MSCs were characterized, CD45 negative, 34 negative, and the MSC markers CD90, 73, 105, CD44 positive. And then these MSCs were differentiated into osteoblasts. Sorry, I'll take some more time. Excuse me, I'll have some more slides. Then these were under osteoblastic conditions. These MSCs were converted to osteoblast, and alizarin red staining, which basically stains the calcium nodules, and serous red, which stains the collagen, and osteocalcin, which showed that these MSCs were actually converted to osteoblasts here. And compared to the healthy control, the patients, the alizarin red staining, which is calcium production and collagen production, is quite low in the patients compared to the healthy controls. So this is again characterizing the mesenchymal stem cells, and we looked at the OCN expression, the ALP expression, and showed that the patients they had reduced expression of osteocalcin and alkaline phosphatase. So we did from these osteoblasts, we did the RNA-seq analysis and showed that the PCA analysis showed that the control and the patients. Now the RNA-seq analysis of the osteoblasts derived from the patients. They were clustered very differently. The controls were here. The patients' osteoblast, they were very different. And the same thing here. The control and the osteoblast derived from the patients, they segregated very well as far as the transcriptome mix is concerned. And we then tried to project this into now. Pseudotime trajectory is one thing where you can, the transcription, we can project the transcription profile of in a pseudotime to see where that biologically, where does that transcription profile, is located. So we show that the pseudotime trajectory in controls versus the patients were different. And here it shows that when we compare this data with the early pseudotime programs and the late pseudotime programs, we showed that the osteoblasts in patients with hyper IgE syndrome, they were arrested at an early stage. So the mature osteoblast formation is not happening in the patients with hyper IgE syndrome. And various genes like TBX15, BMP4, DLX5, which are involved in skeletal development, a strong inducer of osteoblast differentiation, promotes osteoblast differentiation. So all the genes, they were very significantly different between the patients of hyper IgE syndrome versus controls. We also did the validation by quantitative real-time PCR, and the same genes can see that they are different in the patients versus the controls. Then we did we knocked some gene editing. They use this, these variants. These are all hyper IgE syndrome patients' variants that we use. These are mutants. We transfected the osteoblasts with these mutants, and also did shRNA silencing, and again showed a very different expression pattern in the mutants versus the controls. Then we did some osteoblast co-culture experiments, both 2D co-cultures here and the 3D co-culture in a hanging drop. What you do in 3D co-culture is just take a drop of the media in a cover slip, put your osteoclast and osteoblast, and then invert the cover slip and let your drop hang, and let the cells grow within that droplet so it's a very beautiful way of doing 3D cultures. So we did that in these patients. And we obtained these spheroids within that. These are the spheroids. This is a section of these spheroids which are stained for FD and PI, just to look at how these cells are doing, whether they are dying or not. So this is the red is the dead cells. So minimal cell death occurring within these spheroids. So this is only the osteoclast within the spheroids. And then we stained the spheroids. Now we have better images. This was, the stain has not gone deep into the spheroids, but now we have improved upon this as well. So we have done F-actin and Rankel expression in these spheroids and looked at the bone formation score versus the bone resorption score, and both the bone resorption capacity and the formation capacity in the osteoblast derived from hyper IgE syndrome patients plus the mutants, they are different. We also did some scanning electron microscope based studies on we used the dentin discs here. This is a dentin disc. We layered those dentin discs with the osteoclasts here, dentin disc with osteoblast, osteoclast with shRNA. You can see the pits, resorption pits are reduced in this, where this is a normal osteoclast, where you can see a lot of resorption pits here. And when you use the mutants here, again you can see the resorption pits are very low. And again here, just a single resorption pit. These are just representative images. So, but the co-culture experiments show that the resorption capacity of the osteoclasts under the influence of the osteoblasts is defective. So there is a crosstalk between the osteoblasts and the osteoclasts, which ultimately influences the bone formation as well as the bone resorption capacity in these patients of hyper IgE syndrome. So Rashi is the one who is actually, she's still to complete this work, she's on it, and we have to conclude this work a little bit more. So she has shown that STAT3 loss of function affects the osteoblastogenesis, particularly in the early osteoblast, by disrupting the key pathways. And the STAT3 mutation or knockdown impairs the osteoblast-derived regulatory signaling by reduced osteoclast survival, diminished resorption markers, and decreased resorption pit formation. So so this has taken 20 years, and maybe I'll be working more on this. But yes, it's been a very slow process, but that's how science works. Slowly is the fastest speed that you can obtain. You can, reach at the stage where you want to reach. So I acknowledge all my entire team. Rashi is the one who is working this. Shubham was the one who started this work. Smriti did the osteoclast work. She's in NIH now. And my colleagues, Manisha, Mukesh, Sagar, they have contributed to patients, and we have been working very closely. Dr. Casanova, Bertrand Boyson, he had gifted us all those plasmids, 8 plasmids that we worked on. And Professor Sujit Singh, of course, he was the one who introduced me to this topic and put me to work, focused on a single disease and a single aspect for all these years. Thank you so much. If you have any questions.
Thank you very much, sir, for excellent deliberation. If there is any question from the house, please.
Sir, sometimes in my chamber, adult patient come with high IgE level. Sometimes some allergic reaction, certain parasite infection, allergic rhinitis, etc. come with IgE level. Which IgE level high with minimal criteria, clinical criteria, we suspect hyper IgE syndrome?
No, I think I showed you the NIH scoring criteria. Simple, only having the raised IgE is not enough. You have to have the other features also to kind of reach at a diagnosis or even suspecting hyper IgE syndrome. Only IgE would not qualify. IgE will be a very common finding if you're in routine practice, and every other patient will come with a raised IgE. So we cannot rely on just IgE unless we have the other features like facial anomalies and staphylococcal skin, cold abscesses, eczema, history of eczema. So those are the kind of features that should make you think of a hyper IgE syndrome.
Thank you, sir.
Okay. Yeah. Yes.
Thank you, sir. I think these patients are very challenging also clinically to manage because the phenotype is so variable, which we poorly understand, while some people or some children tend to become so sick early on and many go on to live into the 4th and 5th decade. So we had, just sharing one experience, we had one child recently who had aspergillosis in the lung with a proven hyper IgE syndrome. A lobectomy was performed, and subsequently we took this child because of the phenotype for a bone marrow transplant, and is doing fine. And the child has been transplanted, is doing fine. But the question here is whom to transplant, or what are those factors that would determine the phenotype? Are there any answers to that?
I don't think we understand the disease fully till now. The typical hyper IgE syndromes that are associated with the STAT3, the IL-6 ST and IL-6 and the ZNF, that pathway we understand, and those can be considered as a bona fide hyper IgE syndromes associated with the STAT3 axis. The rest of the genes like the CARD11 and all those, I don't think we understand the whole spectrum, and I don't think there's a unifying pathogenesis to all these. So it's just the clinical, syndromic similarities that are used to put this into a single group, but the pathogenesis will be definitely different. And each patient and each mutation is different from the other one.
Okay. So because of time constant, we have to close. So we can discuss later on. Thank you, sir. Thank you so much.
Thank you very much. So we close this session now, and over to you for the next session. Thank you.
Thank you, sir. Up next, we have a debate session, and I would like to call upon stage Dr. Madhumita Nondi and Dr. Dipanjana Datta as the chairperson, and I would like to request Dr. Mukesh Desai to moderate the session. Thank you.
Good afternoon, everyone. Since yesterday, we pediatric non-specialists or pediatric generalists are being bombarded with all weird acronyms. So, but it has been a great eye-opener for us. And at least we are getting aware of things, what's happening in the world of immune dysregulation. And so, what I feel, just as I was sitting through these lectures, I was feeling that why only CVID? A day will come when each and every disease condition will have to be renamed based on exact molecular perturbations. So, of course, our experts are there. Let's, without further ado, let's start the proceedings. I would request our able moderator, Dr. Desai, and we have two speakers. Let's start the proceedings.
So may I please call Dr. Rudropon Chatterjee to speak?
So thank you to the moderators and the organizers for having me. So I'm speaking for the motion, should CVID remain a diagnosis in the era of molecular diagnosis? So I have no financial disclosures, but I have used artificial intelligence to make some of the infographics on this talk. And all the while, because, you know, as my lovely wife says, I'm not quite intelligent in the stuff that really matters. So with that disclosure, we'll begin with this story of a 27-year-old female. She's been around the rigmarole of the Indian medical system for 20 years. In 2004, she had CSOM, cervical lymphadenopathy, biopsy showed granulomas, and she got ATT for 2 years. 2008, she develops bronchiectasis, on and off antibiotics for the next 2 years. 2010, she gets chronic small bowel diarrhea, polyarthralgia, weight loss. Gastroenterologists are alarmed. They do a biopsy from the bowel, just some non-specific inflammation. They think of a lot of things, but ultimately end up doing nothing. And then she develops an episode of pallor, oral candidiasis, again lymphadenopathy, hepatosplenomegaly, and she has pancytopenia. And the hematologists now take her up and biopsy her bone marrow, and they find that it's normal, right? 2012, she goes to gynecology for DUB. Endometrial biopsy is normal. Again, thought of as TB. 2013, she goes to medicine with bacterial meningitis. 2014, she comes to pulmonology with bronchiectasis and right pleural effusion. ADA is high, exudative. They start on ATT again. November 2014, finally, she ends up in the emergency with bacterial sepsis and shock. And immunology gets called here, and we feel this could be an immunodeficiency. We do her immunoglobulins, which are all low. Isohemagglutinin are absent, but her T cell subsets and B cell subsets look fairly normal. So we think this is CVID. She ends up in pseudomonas sepsis. We give her IVIG. She improves. And so what has she had till now? She's had recurrent bacterial infections, meningitis, and hypogammaglobulinemia. And she's been treated for stuff that she doesn't have. Granulomas, she's never had AFB. She's been treated with ATT 3 times. Chronic diarrhea that's non-infective, been treated as infective diarrhea. Seronegative arthritis, and she's been treated for that as well. We started her on IVIG. In 2 years, she's regained weight. In 2025, she continues to follow up with reduced frequency of infections. She's on regular IVIG. Autoimmune manifestations persist. And we do a whole exome. She's better now. We do a whole exome, and it's negative. So do we deny such a patient the label of common variable immunodeficiency because she doesn't have a genetic etiology? The human aspect of that is this is a person who's been around the medical system for 20 years. She deserves closure. She deserves a diagnosis. And the regulatory aspect is that if you don't call her CVID, if she was in the US, she would not get onto insurance. If she's in India, she would not get into the National Rare Disease Policy, and she would not be getting free IVIG. So is CVID in your genes? And it differs based on where you're looking. If you are in the US, where consanguinity is low, roughly 20 to 27% of CVID cases have some genetic etiology. If you're in Iran, where consanguinity is high, that might go up to 54%. But you look at the negative space there, and if you stratify by infections or complications, anywhere from 63 to 75% of patients may not have any gene associated with their disease. So is the clinical context consistent? And if you look at the largest CVID cohorts from Mount Sinai in New York, a majority of them get recurrent sinopulmonary infections. 40% of them have at least one episode of pneumonia, and it's all due to these similar organisms, Streptococcus species, Haemophilus influenza, and other bacterial infections. They also have some viral infections, so it's not just your B cells, some amount of intracellular bacteria, some amount of pneumocystis jirovecii, giardia, shingles, herpes zoster. So it's also the T cells that are involved. And they have a consistent immunophenotype. So if you look at their immunoglobulins, they're all low, whether they're in men or females. They have low immunoglobulins, and they have low switched memory B cells. This is the key mature B cell that produces your antibodies, and these cells are low uniformly in men as well as women. So the clinical phenotype is consistent, the immunophenotype is consistent. What's inconsistent is the genetics. Okay. So CVID is a spectrum. It swings between autoimmunity and infection, and this really depends on your germinal center. If you have small hypoplastic germinal centers, you have reduced switched memory B cells, you have more infections. If you have large hyperplastic germinal centers, you also have, in the periphery, a lot of T follicular helper cells, CXCR3 positive T follicular helper cells, and they're predisposed to autoimmunity. And patients swing between these two conditions. So, does the genotype matter to the phenotype? And at first glance, this is again a very large single center cohort from France, Dr. Casanova's group. If you look at the difference between the blue and the red bar, which is where a gene has been identified and whether it's not been identified, there seems to be a difference, right? There seems to be a marked difference in the burden of autoimmunity and other clinical manifestations. But then you do something intelligent and you take away TACI. TACI is a gene that's a ligand for APRIL and BAF. So it basically helps your B cells mature. If you take away TACI, that variability markedly reduces. Suddenly, you see the burden of disease, clinical disease, is similar. The blue and the green is similar between those with a gene identified and without a gene identified. So the bulk of the variability is being driven by only one single gene. So if it's not in the genes, what is it? And I love a pair of contrarians. This is a pair of contrarian twins that came along. One of them developed CVID. One of them was absolutely normal. And you would think that twins are identical genetically. So what's happening here? So back in 2015, they looked at epigenetics, and they found that as these twins diverged in their clinical phenotype, in some of the genes associated with B cell maturation, the CVID twin has hypermethylation. So hypermethylation means your DNA is less active. So these genes are not being expressed due to epigenetic changes. And 7 years later, when functional genomics catches up with these trailblazers, these contrarian twins, you find out that this bears out with single-cell transcriptomics. You see these tiny blue dots. These are cells, memory B cells from the CVID twin, and they're markedly divergent from his normal twin. So is it still demethylation and methylation that's different? So each number here is an individual cell. So number 1 to number 23, individual memory B cell. And you see the twin with CVID has marked hypermethylation of his genes compared to the normal twin. So is this just a oddity, this pair of twins alone? Well, no, if you take these same hypermethylation changes and look for them in general CVID patients compared to healthy controls, the same genes bear out. These are all genes downregulated that are involved in B cell maturation, and the cause is the same. They have a lot of hypermethylation in these genes. So what are epigenetics? Epigenetics is where you stay. It's what you eat. Even whether you kiss your girlfriend on the mouth or on the cheek, that matters for your epigenetics. What does not matter is your germline. So these twins were identical, but they had the same genes. It does not matter. Where they are matters. So taking CVID from a monogenic to more of a polygenic phenotype, right? Lastly, TNFR SF13B, it's a poster boy of monogenic CVID. Any immunologist at some point of their career would have looked at a genetic report and said, aha, I have solved CVID. This is TNFR SF13B deficiency. And this was the case of this family, multi-generation family, all with TNFR SF13B mutations. All of them had a CVID phenotype until another contrarian comes along, this little boy who also has CVID, but lo and behold, no TNFR SF13B, right? So the immunologists are confused. They go back to the mother and they sequence the mother again. This is 5-6 years down the line. They have new genes that they know of. And the mother now has another mutation. This is called TCF3. This is another gene that is distant from TNFR SF13B. And this is known as epistasis. So when distant genes interact with each other, this is a phenomenon known as epistasis. And so they decide to look at the phenotype of the mother again. And so pay attention to this small dot here, 2-2, which is the mother. When she's compared with her relatives, it seems that she has lower memory B cells, she has lower total B cells, and even if you stimulate her cells with CD40 ligand and a lot of other inflammatory cytokines, she still does not produce as many antibody secreting cells. So she's still more immunodeficient than her own relatives. So that tells you it's not a single gene. TNFR13SFB has fallen. It is being influenced by multiple other genes in creating this phenotype. So where are we at the end in terms of our knowledge of monogenic CVID? This is known as the Dunning-Kruger effect. 10 years ago when NGS came into the picture, we were all excited. We thought we knew a lot. Confidence was at its peak. It was the mount of stupid, and we thought we knew everything. We knew nothing. 10 years down the line, now NGS is cheap. Everyone is coming with variants of uncertain significance, and we are drowning in them. So we are in the valley of despair. But we're moving towards a stable plateau of sustainability along the slope of enlightenment as functional genomics progressively takes over, and we know that there are multiple things happening in every patient. So I'm back to the city of joy after a long time. So I'll end with this. So 11 years ago, you take a boy out of Bengal and put him in Mumbai, and sometime later you find that he's an immunologist, but he's also a damned capitalist. And so do you think then that makes all the Bengali moms fall out of love with Karl Marx and move into the arms of Adam Smith? No, it does not. That is an outlier. You should not take single genes that are outliers and generalize to the norm, which is CVID. Thank you. And if anyone is offended by that, I'm a boy who feels he looks like that in his head. So I'm obviously delusional. So you can please ignore whatever I said.
Thank you, Rudrapan, for a very, very aggressive defense of your proposition, taking us to the mount of stupid, to the valley of despair, and then now some enlightenment. So I think we will have another speaker, Dr. Vijaya Gauri, who will want to very aggressively defend her proposition. And I request both of Vijaya to finish in less than 10 minutes, if whatever your time schedule permits, so we can have some rebuttal from both of you.
Yeah, thank you, Dr. Rudrapan. That was wonderful. This was more difficult to say CVID is monogenic and nothing else. But then the leaflets that we plucked from the chai bagan, it identifies whether this is going to be a good tea or not. So unless I have my monogenic defect in hand, I don't know whether this CVID, which is a multisystemic disease, which has got a heterogeneous symptom complex of infections, autoimmunity, autoinflammation, malignancy, lymphoproliferation, is an umbrella definition of recurrent infections, hypogamma, without clinical manifestations, symptomatic before 4 years, but I can label it as CVID only after 4 years. Why heterogeneous? Because the infections are not restricted to encapsulated bacteria. 30 to 40% have underlying T cell dysfunction as well. Autoimmunity in around 30 to 40% of so-called CVID cases. Granulomatous disease, 54% of lymphocytic ILD, 30 to 40% granulomatous hepatitis, and malignancy risk is 5 to 12 times more, incidences 15 to 22% more in so-called CVID cases. Why heterogeneous? The onset of varied clinical manifestations and lab abnormalities do not necessarily coincide. My experience here is predominantly from Vijayawada Hospital for Children, and we had 6 patients of PI3 kinase delta syndrome, all of them carrying the same most common mutation, and you can see here that they presented with varied clinical manifestations. There was an SLE phenotype in one boy, I'll discuss him. There was Evans phenotype in 2 children, IBD in 3, bronchiectasis as a presentation in 2 patients, EBV lymphoproliferation in 1 patient, and EBER-positive malignancy in 2 of our patients, one being angioimmunoblastic T-cell lymphoma and other a non-Hodgkin's lymphoma with a Richter's transformation. Hypogamma was seen in only 2 of them, but low class switch memory B cells, even with the normal B cell numbers, were seen in 4 of them. Normal LSSA and immunoglobulin profile was seen in 2 of them. And look at the organisms, it's not just restricted to B cell encapsulated organisms. In one of our patients, there was a family history of IBD in the father who died at 35 years of age, and this girl presented with lymphoproliferation, which was EBER positive. This was our first patient of APDS syndrome, which you might have seen yesterday, today also probably you will be seeing. She presented with an inflammatory bowel disease, went on to develop bronchiectasis, and our first diagnosed case of APDS syndrome. This was the boy I was talking about, had an autoimmune thrombocytopenia, hemolytic anemia, went on to develop an SLE phenotype, diagnosed and managed as SLE, developed an alopecia areata, keratoconjunctivitis sicca, and finally landed up at immunology department, diagnosed as APDS, no hypogamma in him. Who are they? This was another case of PI3 kinase receptor mutation who was referred to Tata Memorial Hospital because this was a second malignancy which he was coming about with. This was EBER-positive Hodgkin's lymphoma, previously treated as splenic marginal zone lymphoma. He had pan-hypogammaglobulinemia before the chemotherapy was given to him. It is not pan-hypogamma, it is hyper-IgM phenotype. IgM is normal here. The most interesting thing is every time he would have a varicella, he had an EBV titer high also. Both of them, Herpiviridae family, went hand in hand. And he had panlymphopenia as well. Turned out to be PI3 kinase receptor mutation. Treated with sirolimus, we gave him rituximab, we referred him for BMT, but we lost him. So what do we infer? All these things, manifestations can take longer. Autoimmunity with infection, lymphoproliferation, autoimmunity, and hypogamma, we should always look for monogenic defect. That's our experience. Another boy, 15-year-old, this I saw in Coimbatore, who was diagnosed as CVID by Madras Medical College Hospital in view of bronchiectasis and recurrent respiratory tract infection, was managed on IVIG. And then in Coimbatore, he got admitted in Coimbatore Medical College Hospital, admits them beyond 12 years of age, they go to medical ICU, admitted with classical HLH, and he was being treated, thinking that this was sepsis. Somehow I landed up there. His EBV PCR copies were 1.42 and as we all, 42 lakhs. And as we all know, male child, EBV HLH, history of CVID, and lo and behold, turned out to be X-linked lymphoproliferative syndrome, SH2D1A mutation, but we lost the child. Where were we? Why couldn't we diagnose this child? One size does not fit all. The second thing, pathophysiology is varied, B cell dysfunction to defect in BT interaction with hypogammaglobulinemia. The disease evolves over a period of time. Again, reference from Tata Memorial Hospital. This boy is from Calcutta. History of cervical adenopathy and tonsillopharyngitis recurrently from early childhood. He had failure to thrive, recurrent RTI after 12 years of age. He had a pan-hypogammaglobulinemia. He had skin lesions which were like vasculitis. He had psoriatic lesions at other parts of the body. And he also had viral warts when he came to us at Vadia. He had history of varicella zoster twice. At around 18 years, he was in Hyderabad where they thought that he had a jaundice. He was diagnosed to have an ampullary tumor. They ruled out neuroendocrine tumor. And then he reached Tata and somehow landed up with us. He had pan-hypogammaglobulinemia, B and T lymphopenia, turned out to be STK4 homozygous likely pathogenic combined immune deficiency. We have lost so much time to diagnose this boy. Another boy, 9-year-old, referred for viral warts, ichthyosis, scarring varicella zoster, CMV pneumonia. Why was he referred? Because he had a EBV-driven Hodgkin's lymphoma. Turns out to be RAC2 hypomorphic SCID. He had lymphopenia and eosinophilia as well. We lost 9 years. What do we infer? Phenotype is broad, ranges from bacterial to viral to multi-organ involvement. Sometimes patient may also have distinct initial presentations like autoimmunity, granulomas. Term CVID cannot be generalized and applied universally. Possibility of monogenic etiology needs probing. Why heterogeneous variable age of manifestation? Again, a 33-year-old who had lymphadenopathy from 26 years of age and also IBD and was not picked up, was managed with AKT. Finally, he developed an Hodgkin's lymphoma and landed at Tata. We were happy about it because he finally reached at Wadia Hospital, turned out to be a pan-lymphopenic, pan-hypogammaglobulinemia, and his EBV serology was positive, PCR was 1,000 only. We have quite a few people with EBV coming because we keep looking for it probably, and turned out to be a CTLA4 mutation. The index patient's sister also had an IBD and she has hypogammaglobulinemia as well. Again, we waited till 26 years to diagnose this man. A small young boy, symptom onset at 10 years, and then he had Evan syndrome, IBD, and the scopy showed pancolitis, biopsy was early IBD, he had pan-hypogamma as well, and stool calprotectin was high, polyautoimmunity, sent an exome sequencing, turns out to be CTLA4, lo and behold, mother carries the same mutation. Till date, I called her up yesterday, asymptomatic. Child is transplanted, doing well. Another 41-year-old male, 38 kgs at present, was diagnosed as a CVID and was managed at Madras Medical College Hospital with IVIG. No medicine prophylaxis was given to him. Multiple admissions in last 11 years. Look at his infectology, Pseudomonas, strep pneumonia, H1N1, pylori-induced gastritis, chronic diarrhea, Giardia, Campylobacter jejuni. And finally, he landed at GKNM Hospital with Dr. Ajita, who sent an exome sequencing. Turns out to be IKBKB, heterozygous likely pathogenic mutation. He had a huge spleen and portal hypertension, and this is my literature review, which shows that gain-of-function mutations of NF-kappa B pathway causes immune dysregulation, functional B T cell defects, and also causes nodular regenerative hyperplasia with or without immune hepatic fibrosis responsible for hepatosplenomegaly and portal hypertension. We cannot transplant him now. CVID phenotype, again with an IBD, turns out to be ICOS-L mutation. He had an IBD for 4 years, developed a sepsis, went into an ICU, and then referred for an immunological diagnosis. We are missing out on many of these patients. All of that which is there within this green block has been seen in Wadia Children's Hospital. I have not included TASI here. I have not included BAF. I have not included April here. Vaccine responses in this set of patients was pretty variable. They were already treated with IVIG, or they were too sick for us to wait for not giving them an IVIG. What is the consensus? International consensus statement says those with the classical feature of infection, autoimmunity, lymphoproliferation, along with hypogamma, IgA, IgM low, T-cell dependent or T-cell independent antigen responses should be considered as CVID. But we should always remember that not everybody would present with an infection alone, and anybody who has got this latter phenotype of infection, autoimmunity, lymphoproliferation, and malignancy should definitely be offered a genetic diagnosis or genetic test. Because we need to target them, we have to take them for stem cell transplantation. So these are the two literatures. Maximum work on CVID has been done by Amaratunga et al. From New Zealand, and I saw his papers from, say, 2009 till date, and he ultimately has agreed that it is the monogenic cause which needs to be ruled out before calling somebody a CVID. And this is ECID versus ICON. What is the common factor between the two of them? This is something which, so this is, ultimately the Th1, Th2 fate would depend upon what is the interferon milieu, what is the cytokine milieu. If it is interferon gamma or IL-12, it is Th1 differentiation. If it is IL-4, it is Th2 differentiation. IL-4 reacts with B cell, we have antibody production. When we have all the infections are under control, the Treg stops B cell activation, also stops T cell activation. We have more of Treg formation. But if there is a genetic predisposition anywhere, or if there is a Treg defect, we have autoimmunity. Unless we look at it, we will never be able to identify this. This is a paradigm of Th1, Th2 by Mossman and Kaufman. Another flow cytometric analysis of CREC shows the possible subgroups or defects in B cells can be in B cell production. It can be early B cell maturation or survival, B cell activation and proliferation, germinal center or post-germinal center. Unless we do a monogenic array defect, how are we supposed to identify all this? With subsequent effect, all this will have on BT interaction, and that will determine the patient phenotype. So disease evolves over a period of time. Monogenic common variable deficiency is a very important journal article which I saw. This is more CVID score of gene positive patients and gene negative patients, and they derived a more CVID score 7. There are 8 categories. These are the thing which is within the box, and they gave a cutoff of monogenic CVID score at 2. If the score is 2, it is non-monogenic CVID. Sensitivity is 100%. If the cutoff is 7 or the score is 7 or more, it's likely monogenic. And I extrapolated it in our CVIT cohort. I could not present it because of certain personal reasons. So I saw that almost 48% of our patients in our cohort had more CVID score of 7 when they were monogenic CVID positive. So that goes ahead. We can say the same thing is seen here also. International consensus state mono CVID score is 25% in consanguineous, non-consanguineous population, but with consanguinity it is as high as 54%. So what is my conclusion? With genome first approach, IUIS genes that are diagnosed have doubled with shifting from one table to other. Precision medicine for managing monogenic disease based on pathophysiology has revolutionized survival. We need to do the NGS. CVID-like disorders are excellent example of locus heterogeneity, where mutations of over 50 genes can lead to similar phenotypes. CVID may vary in presentation, but underlying commonality is hypogammaglobulinemia. As CVID phenotype evolves over a period of time, approach should be flexible to reinvestigate and redesignate them. Vaccine response is difficult, as I told you. Cases not discussed here are STAT3 gain of function where there was no hypogamma, and TASI presenting with AITP infection phenotype and hypogamma. We are treating her, it's more a potentiator, and I have not included her. I acknowledge Dr. Zanet Karim Boy, my mentor Dr. Mukesh Desai, my entire team at Wadia Children's Hospital, who have supported me and who are sort of enlightening team for me. Coimbatore, GKNM Hospital, KG Hospital, Masonic Children, and PSG, where I am working right now, our patients and parents, and PIDCON organizing team for having me here. Thank you very much.
Thank you, Dr. Vijaya Gauri. Now, I would invite Dr. Rudrapan, if he has any rebuttal to make. Only 2 minutes for time.
I have a very short and sweet rebuttal. She's beautifully picked up outliers that I spoke of. Those are all the damned capitalists up there. I'd ask you just a simple question. You know, what's the proportion of genetic positivity in the average middle-aged male who comes to your OPD with diarrhea and bronchiectasis? If it's more than 2%, I yield the debate to you. Thank you.
And even if it is 2%, and I can offer a treatment for him, a hematopoietic stem cell transplant before he goes on to develop a lymphoma, a malignancy, or bronchiectasis, I think it makes a deal. It's okay. Absolutely okay.
So friends, you all heard both the debaters argue and very vigorously put their point in favor of what they believed in. I think we need the term CVID because there are so many disorders which we can only categorize them as CVID. So I'm with Rudrapan on that, because from there we can start to what Vijay is saying, that you need to find a monogenic cause and find out for every patient whether they have a disease which is identifiable and treatable. And if 2% is enough for Vijaya, I think in the monogenic cohort of CVIDs, you will find that now increasingly we can recognize a monogenic defect in almost 20% of these patients. So I think the debate is not yet settled, but we need both of you, and very well argued. And if there are any questions or anything you want, some comments, we would really appreciate.
Yeah, one thing as a geneticist I would actually mention that, genetics is not a magic tool. So we need the clinical part to even come to the genetic part. And very rightly, as you said, you showed this course and you pointed out, and sir, like we all know that when we test, it is not that every report comes positive. Most of it is inconclusive, and if we don't have the strong genetic, the clinical background of it, it is impossible to actually give a diagnosis. So I am on with sir that yes, a clinical diagnosis is important. Genetics definitely redefines the personal, personal genomics part of it, the personalized treatment part of it, and I guess both are needed in the clinical setup. Otherwise, care to the, as you showed, that patients waiting for 26 years, 9 years, that is something that we really see, that diagnosis is late, and at times that management intervention, that time is very crucial. So yes, we need both.
So all those in favor of both, raise your hands. So, madam, they're both.
So here it seems in this war there are no winners or losers. Everyone is on the same platform, and things are evolving. And so thank you very much. Both the speakers have done very well, and our moderator, sir, Dr. Desai also, and my co-chairperson, Dr. Dipanjan. Thank you, everyone.
We'll go into the last two presentations. Before that, after the presentations are over, the last two slots of poster presentations will start. We'll have Dr. Sagar, Dr. Suprit, Dr. Vijaya and Dr. Sanjeev in the tea break for the poster presentations. So
now we will have the paper presentations. The first one will be by Dr. Sachi Chawla from PGI Chandigarh. I would request Dr. Surjit Singh sir and Lisa ma'am to judge the papers. Thank you.
Good afternoon, everyone. I am presenting my research on deciphering the immunological and genetic landscape of patients with CVID, insights from North India. CVID, as we know, is the commonest primary antibody deficiency defined by marked reduction of IgG along with IgA, along with evidence of B-cell dysfunction and exclusion of other causes of hypogam. The B-cell defect is central, but patients can have normal peripheral numbers, implying there is defect in terminal differentiation into memory subsets. Clinical presentation is diverse. Etiology of this disease, as we just now also heard, that it is very heterogeneous. Only 25 to 30% of the patients have genetic diagnosis, while 70 to 75% do not possess any genetic etiology. In these patients, other evidences like epigenetic dysfunction is the growing one. The knowledge addressing the genetic and epigenetic landscape was lacking from the Indian cohort. Therefore, this study was designed, in which we enrolled the patients depending upon the ECIT 2014 criteria. A total of 59 patients were evaluated for the immune cell dysfunction in terms of B and T cell immunophenotyping. We also carried the whole exome sequencing, and in the patients who were non-monogenic, we tried to see the role of DNA methylation by sorting the CD19 B cells and subjecting the DNA to targeted human methylome panel. We identified the differentially methylated regions with a difference of more than 25% in patients compared to the controls. Here we identified that most of our patients belong to the northern region of our country, and socio-demographically there was male preponderance and early onset with less than 20 years of age. The clinical phenotype was complex for most of the patients, representing the infectious as well as the non-infectious phenotypes. The diagnostic delay here we witnessed was higher than that of the Iranian as well as the European cohort. Here we also witnessed that celiac disease was higher as an autoimmune manifestation. Coming to the immune cell dysfunction, we observed that the patients lacked the differentiation to the memory subsets, which led to the expansion of naive B cells in the patients, and there was also reduction of the plasma blast, implying the antibody secreting cells were diminished in the patients. There was expansion of the potentially autoreactive CD21 low B cells in our patients. Along with B cell dysfunction, there was evidence of T cell dysfunction. There was reversal of CD4 to CD8 ratio. There was expansion of CD4 TEMRA cells, and also there was reduction in the CD8 central naive as well as the central memory T cells. So this means there was hallmark B cell dysregulation with disrupted B and T cell immunological crosstalk. Next, we move on to the immune cell architecture across different clinical conditions in CVID. We observed when there are conditions of viral infections and bronchiectasis, there is expansion of CD21 low B cells in our cohort. And among T cells, we observed a much varied pattern where there was diversion from the naive towards the effector memory population in conditions of bacterial infections and bronchiectasis. We also observed there was expansion of the CD8 TEMRA population with potential cytotoxic capacity in conditions of bronchiectasis. These cell types can serve as immunological biomarkers in predicting the clinical heterogeneity in CVID. Then we come to the genetic spectrum of our cohort. We observed that a large proportion of our cohort was formed of non-monogenic patients. Genetic etiology was identified in 21 patients, where there were 16 different type of genes identified. These were the novel variants in these 16 genes. Out of these, 6 were novel variants. We also observed the phenomena of hypomorphic variant in RAG1 gene, along with the skewed lionization in IL2RG variant. Then we tried to compare our monogenic cohort with the non-monogenic cohort. We observed that our monogenic patients had significant viral association with the viral infections as well as underlying consanguinity. The non-monogenic patient cohort showed little bit higher of autoimmunity. The B cell dysfunction was common to both, but it was more profound in the monogenic group compared to the non-monogenic ones. Here the transition, the differential was that the monogenic group showed arrest at transition B cell stage, while the non-monogenic group showed the arrest at the CD21 low B cell stage. We also observed when the clinical manifestations were growing, the chances of finding monogenic etiology were significant. Then we come to the cause in the non-monogenic group of our patient. We saw that a subgroup of our patients showed 66,079 differentially methylated regions, which separated the patient group entirely from the control group. Out of these DMRs, 87% were hypermethylated and 12% were hypomethylated. We also found these DMRs to be closely associated near the transcriptional start site. So from here I can form that there is significant hypermethylation which is leading to the gene silencing in non-monogenic patient group in our cohort, and these DMRs are closely associated to the proximal regulatory elements which can thereby silence the gene expression. So we also tried to look if there are any B cell specific genes which were affected. We found a set of genes which are responsible, which can act as cell surface receptors as well as downstream signaling molecules, as well as the transcription factors that can control the B cell positioning within the germinal center during the germinal center reaction to be hypermethylated in patients, non-monogenic patients as compared to the healthy controls. Then these set of DMRs were belonging to biologically important relevant pathways like MAPK cascade, leukocyte migration and immune effector processes. And among these pathways also, more than 75% of our genes were hypermethylated and less than 25% of our genes were hypomethylated. So with this, I come to the conclusion of my study that our cohort of CVID patient is formed of both the non-monogenic and the monogenic patients, with monogenic patients having defect in these genes and non-monogenic patients giving us a pilot scale data that there is hypermethylation underlying the B cells, which is leading to the gene silencing. Both of these type of defects are leading to the arrest in the B cell development and development at different stages, like transitional B cell stages in monogenic cases and CD21 low B cells in non-monogenic cases. These are culminating to the impaired B cell differentiation and reduced plasma cell formation, giving rise to the greater susceptibility to a number of clinical manifestations. Thank you.
Sanchi, based on the B cell phenotypes, can you predict whether a given patient will turn out to be monogenic or a non-monogenic variant? And how do your studies on hypermethylation compare with studies on other cohorts of CVID from other centers?
Sir, to your first question, we observed here with the existing monogenic patients that there was arrest at the transitional cell stages in the monogenic cases. This data suggests that if there is defect earlier to the CD21 low cell stages, or there are more number of clinical manifestations in the patients, this can hint us to the underlying monogenic etiology. And coming to the hypermethylation part, there are various studies as previously have been shown. There were 4 genes which came in my data also, the calcium channel regulating genes, which came out to be hypermethylated, and these have already been reported in the monozygotic twins affected with CVID. But I have also identified a set of new genes which are regulating the B cell positioning in the germinal center, which were hypermethylated in our non-monogenic patients.
Very interesting talk. What I want to ask you, can you use a demethylating agent to treat your patients who got hypermethylation?
Sir, like in other diseases, yes, this approach is used, but here I would say if I can add the patients with monogenic etiology and I can evaluate the DMRs in those patients also, I'll be able to identify the true target sites which I want to, for which I want to reverse the methylation. So yes, sir, this data can provide us evidence of the epigenetic dysfunction underlying the non-monogenic patients and can pave a way to focus at the new targeting approach, therapeutic approaches which can actually downregulate the hypermethylation.
What about the enzymes which are important for methylation? Any mutation
DNA
in
methyl
that?
transfer. Sir, we did whole exome sequencing. We did not identify as such any mutation in DNMT genes.
Mutation.
Sir, we have not carried out the, we have carried out whole exome sequencing only. We have not evaluated.
Okay.
Sachi, one clarification. You mentioned cases and controls in the non-monogenic CVID.
Yes,
are these, controls the monogenic ones,
No,
or
no.
they are healthy controls?
This is the healthy controls.
Why did you not compare the two instead of comparing it to healthy controls? Obviously, they'll be different.
This is the future perspective. That time it was limited resource availability, so I was.
But then you cannot draw the conclusions that you are drawing. That the two groups are different in their methylation status.
So
that's
this
not
is
the
healthy,
conclusion.
healthy control compared
From
to
healthy controls, agreed.
Yes, ma'am.
But between the two, we are not sure.
That is what, ma'am, I am saying. If we identify the monogenic and non-monogenic group in terms of the differential DNA methylation, we can identify the true methylation sites and target those. So that can be the future perspective.
Okay. And maybe you will discover more monogenic if you do deep sequencing?
Yes, ma'am, that can. Thank you.
Our next presentation is by Dr. Umair Ahmed Bagir.
Good afternoon, everyone. This is part of my 7-year-long PhD journey where we tried to look at the comprehensive transcriptomic profiling in CVID patients. So CVID is a heterogeneous disease and multiple, as Rudrapan has mentioned, multiple genetic, epigenetic, polygenic and transcriptomic studies have been done to trying to decode this disease. So over the years, different transcriptomics studies have shown the cytotoxic effector profile, the upregulation of the interferon responsive genes, the conservation of the B cell defects where only the early B cell, the genes involved in the early B cell, they were downregulated like the PAX5 gene, and clustering using select few genes was done. But the bulk RNA sequencing can dilute and obscure the cell intrinsic findings. A mechanistic link between the clinical presentation and the transcriptome, is it possible? And can CVID like disorders share a common pattern with the idiopathic CVID? With this thing in mind, the objective was to do the transcriptome profiling from T cells and B cells in CVID patients to understand the etiopathogenesis for hypergammaglobulinemia. So we took patient blood, sorted them magnetically into T cells, B cells, checked for the purity, and because they are sorted cells, no commercial lab was ready to do the downstream transcriptome profiling. So in-house, I standardized the extraction, did the quality control, and used a single cell sequencing kit for performing the bulk RNA sequencing on sorted T cells and B cells using NovaSeq X Plus machine. After the bulk RNA sequencing, I debulked the media using a cybersort method where the bulk sequencing was deconvoluted to single cell sequencing to identify the cell type. A custom bioinformatic pipeline was used to perform differential expression, enrichment pathway, and WCGNA analysis, the weighted gene cluster analysis. Heterogeneous group was present in this cohort where a mix of recurrent infections, autoimmunity was there, a mix of monogenic and idiopathic patients was used. So this is the debulked debulked data from the T cell transcriptomics. You can appreciate majority of them are subsets of the T cells, and this is the debulked data where the patients and the control, majority of these patients, majority of this data has arisen from the B cells. So this is the T cell transcriptomics where 301 differentially expressed genes were identified. Of these, the hyperactivation and cytotoxic genes were observed to be upregulated in CVID patients. The checkpoint in exhaustion markers like the TIM3 and the LAG3, they were also upregulated, but there was a downregulation of the IL-7 receptor and the 6R, which is critical for the survival. Along with that, there was a reduced expression of mitochondrial genes indicating a metabolic compromise. Then this was validated using RT-PCR, and the enrichment pathway also showed significant changes in the pathway involved in the IL-17 signaling pathway and the T-cell receptor signaling. The WCGNA network clustering blinded analysis showed upregulation of the cytotoxic genes and downregulation of the metabolic genes. The B cell transcriptomics, however, had only 18 differentially expressed genes, and they were also validated using RT-PCR, and you can appreciate the immunoglobulin rearrangement genes in them. And this pathway also highlighted the B cell dysregulation, and the pathways related to chromatin condensation was also involved. Here IL-17 signaling pathway was affected in this patient when a random blinded WCG analysis was performed. But because this is primarily a B cell defect, I isolated all the immunoglobulin genes, though it's a transcriptome profiling, isolated all the immunoglobulin genes that are mentioned, and then performed a separate exclusive immunoglobulin gene rearrangement pattern, and surprisingly IGHV 434 upregulation was seen in CVID patients, and this is a gene that is reported to be seen in patients with autoimmunity, especially SLE. So T cell in the transcriptomic studies show a T cell hyperexhaustion pattern whereas there is a B cell developmental arrest in these patients. There is a tug of war where the T cells are in overdrive, they are activated, and there is a upregulation of those genes along with the immune exhaustion that is present in this patient, and the debulking of the bulk RNA sequencing also showed the expansion of activated CD8 T cell component in these patients. There was a metabolic drain driven by the T cell exhaustion, and which may be a reason responsible for that, and the activated T cell compartment is metabolically exhausted. Coming on to the B cell component, there is a block at the switched stage where these immunoglobulin rearrangement genes have been shown previously are responsible, and the cybersort also confirms the presence of this pattern. the important finding is that the A2 and the H2 down-regulated genes, we all know that these patients are susceptible to respiratory infection. But the G2 and G4, they are down-regulated and they can be responsible for failed polysaccharide capsule response. And now we are further conducting a study to correlate the same. The important finding is this IGHV 434 in 2026. There was a patent filed by Bodo Grimpasher, a US patent, where they have shown that how you can use repertoire sequencing to manage hypergammaglobulinemia patients, and this is one of the important clones that they have reported in these patients. So to summarize, there is a chronic activation and mitochondrial failure in T cells and a developmental block, and some patients have IGHV4 autoreactivity, and CVID is a collision of these two distinct failures. Thank you.
So, Umair, do you think we are anywhere near having a biomarker for diagnosis of CVID?
So IgG is the best one, easily available one, but metabolically, like for vaccine responses, now we are doing RT-PCR and correlating it with the vaccine response for the G2 and G4 to see whether it can easily be used instead of the complicated vaccine response. Yes, ma'am.
The switch between the hyperactive and the exhausted cells, is this, I mean, you saw both, right?
Yeah.
But did you do a clinical correlation to see whether there was any difference in the state of the patients in which time point in which you collected the samples?
Like it was just one single time point study, and almost all of them had active infection, so it is difficult to comment on that.
Okay.
Dr. Umair, that's a wonderful work. So I have one technical question, and so it's that. So you said that in the B cell you could see only 8 or 18 differential expression. Is it due to that when you did the sorting, was it that there is low B cell number, or is it the B cell number was normal?
Only one patient had low B cells, but in the rest of them the number was normal.
Okay. And my second question is that in your patient cohort for the CVID, all were like genetically confirmed, or you, because I see that you have recruited the clinically.
So I had both the genetic and non-genetic. I compared the both, did a separate DEGs for both. There was no significant difference. Only one gene, IL-32, that was different. The rest, there was no difference.
Irrespective of genetic etiology,
yes.
the findings were same.
Yeah.
Okay. Thank you.
Thank you.
Thank you. Now we will take a short tea break, and the next session will start at 4:30 p.m.
Can you hear me? So thank you so much for your invitation. I'm sorry I cannot be in India now. I really wish I could go. And I'm sorry for my voice is not at its best. I'll try my best. Let me know if you cannot hear me properly. So let's start. Today I'm going to talk about the autoantibodies that neutralizing type 1 interferon in a variety of viral infections that we studied. So I think everyone in this meeting probably already know type 1 interferon immunity pretty well. Just a simple summary. This is kind of what we think type 1 interferon immunity is, almost like a first line alarm that can ring in the body when there is a viral infection. What happens is that usually if there's a cell, for example, a cell in the lungs that get infected by virus, there are detectors inside the cells that can detect viral components. Then it activates a pathway, induce the production of type 1 interferons. So in human, there are 17 type 1 interferons subtypes. All of them all binds to the same receptor, which is IFNAR1 and IFNAR2, activates the pathway that eventually induce the production of interferon-stimulated genes, ISGs. These ISGs can actually kill the virus or stop viral proliferation. They either bind the viral RNA, bind the viral DNA, degrade the viral protein, stop the viral protein from translation. There are hundreds or thousands of them. Then they attack the virus, and then the cell get into this type 1 interferon status, meaning that they now are resistant to the viral infection. There's a cell type called plasmacytoid dendritic cells in the blood. They're very rare, less than 1% of your white blood cells. They're called professional type 1 interferon producers because they can make tons of type 1 interferon at a very early time point into the bloodstream. And because they have constitutively expressed IRF7, one of the key transcription factors that's important for this pathway. Now, before the COVID pandemic, there are many of these genes on this pathway have already been shown to be critical to antiviral functions. And the viral diseases our lab and other labs have studied were, for example, there are herpes simplex encephalitis caused by HSV, live attenuated vaccines, RSV or rhinovirus pneumonia, and importantly, influenza pneumonia. So since 2015, our lab has identified TOR3, IRF7, IRF9, three inbound errors in the type 1 interferon pathway that the defect of them leads to severe influenza pneumonia infection. And so this happened, between 2015 and 2019. So when actually the pandemic happened, we immediately have the hypothesis that the same genetic defect that leads to influenza pneumonia can also underlie severe COVID pneumonia. And our lab, led by Jean-Laurent Casanova, and actually my former postdoc mentor, Helen Su, they organized this international consortium called the COVID Human Genetic Effort. Many of you probably also part of the consortium. We recruited patients from all over the world, and the first hypothesis we tested was actually indeed are the same genetic defect also leads to COVID-19 pneumonia, and we were very lucky to be correct. So the highlighted red genes on the type 1 interferon pathway, which have been shown to cause other viral infections, especially influenza pneumonia, can also cause severe COVID-19 pneumonia. And this is what we found between 2020 and 2022 with the entire consortium and many, many collaborators. Why I talk about these inbound errors first is because these cases, although very rare, all these inbound errors are extremely rare, but what they showed is that a genetic defect in the type 1 interferon pathway leads to severe COVID-19 pneumonia. And the causality is clear here because these are inbound errors. These patients, no matter if they are pediatric patients or adult patients, they're born with these genetic defects. And multiple genes on the same pathway leads to the same phenotype also is another evidence that the entire pathway is important to control COVID-19 infection. So based on this discovery, at the same time in 2020, our lab identified autoantibodies neutralizing type O interferon that also underlie severe COVID-19 pneumonia. These discovery is not made from imagination, because before the COVID-19, before 2020, we already know that different autoantibodies neutralizing cytokines can lead to the same phenotype as the defect of the same cytokine pathway. For example, there are autoantibodies neutralizing Th17, IL-17, there are autoantibodies neutralizing interferon gamma, there are autoantibodies neutralizing IL-6. So the autoantibody neutralizing a particular cytokine leads to the same infections as the patients who have defect in the cytokine itself or their receptors. So we already knew that. This has been known for 30 years before the pandemic. So when we discovered that the defect in the type I interferon pathway can lead to severe COVID-19, it's only reasonable for us to test whether there are autoantibodies neutralizing type I interferon, and indeed there are. You're going to see these similar figures again in my talk. So essentially what it does is that we take the patient's serum or plasma, mix them with the cytokines. Here is different subtypes of type I interferon. There are type interferon alpha, omega, or beta. And then we use it to stimulate a cell that has the receptor and a reporter. So if the interferon is not neutralized, the cell will respond to the interferon. We can read it. And if the patient serum has autoantibody that neutralizes the interferon, then the cell will not respond. And that's what you see these dots under the line, under the threshold are. So each dot is a patient, showing that this patient has autoantibody neutralized the type I interferon, therefore the reporter cell cannot respond. So these are called the neutralizing. So you can see that we tested thousands of patients with critical COVID, severe COVID. Critical here are patients intubated in ICU or died. Severe COVID are patients have pneumonia, hospitalized, and the control mild COVID are the patients who essentially just had like a flu-like symptoms and they're fine. So you can see that the patients with critical or severe COVID-19 are much more likely have autoantibodies neutralizing either high titer or low titer of type I interferon. We find that totally in our cohort about 15% of the critical patients have autoantibodies. And this slide is probably the most important slide, the take-home message you're going to see today for my entire talk. That is, these autoantibodies are actually common in the general population. So let's break it down. On the figure on the left are autoantibodies in patients with critical pneumonia. And we plot the percentage of patients who are autoantibody positive along the age. And you'll see that it's overall about 15%, but in the patients who are older, over 70 years older, there's a higher percentage, goes to almost 20%. There is a slight difference between men and women, but overall, this is about 10 to 20% of the patients with critical COVID pneumonia has these autoantibodies. Now, if you look at the general population, on the plot on the right, these are blood bank donors we collected before 2019. So these are just normal healthy donors, donated their blood. We collected them all over France and other countries, and they are considered just representative of the general population even before the pandemic happened. And you see that they have about 0.5 to 1% of them have autoantibodies neutralizing type 1 interferon. Depends on what titer you're talking about. So this is the most sensitive, the lowest titer we can detect. You get to about 1%. If you use a more stringent titer, a higher titer, it goes to about 0.4%. So this is less than 1% in the general population. But you see, they're relatively stable before the age of 70. As soon as the population reached the age of 70, this is a log scale increase of the autoantibody positive rate and reach almost 7% when the patient's over 85 years old. This is relatively common. This is, we're talking about 1% to almost 7% in elderly people. This is not a rare phenomenon. And this is very surprising to us because most of these are healthy donors and they're relatively otherwise healthy in the general population. Now, after we tested the patients with severe COVID-19 pneumonia, we actually went back to the influenza pneumonia that we studied before 2019. So the cohort was collected between 2015 and 2019. And again, in the patients with critical influenza pneumonia, here where I'm talking about the normal seasonal flu that happens every year, we actually again find 5% of the patients with critical influenza pneumonia also have these autoantibodies, and you don't find them in people with mild influenza infection, which is probably I'm having now. And this again is very convincing to us because now we have several independent evidence showing that the type 1 interferon immunity is critical to control viral pneumonia. We know there are rare inbound errors in the type 1 interferon pathway that leads to severe critical, severe influenza and COVID-19 pneumonia. We show that the autoantibodies neutralizing the cytokine itself leads to the same disease. The inbound errors really showed that these it's causal, not a consequence of the infection, but also in several cases that we were able to get blood samples from the patients who have autoantibody that's collected before their illness for totally random reasons. For example, a patient blood was collected for surgery before he got the COVID infection, and we found even before the infection, the patient already have these autoantibodies. So again, the autoantibody is the cause, not the consequence of the viral infection. And our study really shows that the studies in inbound error is critical here because it started. The study actually started in 2015 when we first identified the same genes can cause severe influenza, seasonal flu. Because of these inbound errors found in flu, we found the same inbound errors in COVID patients, and we found the autoantibodies in the COVID patients because really honestly it's the sheer number of patients that helped us to identify these autoantibodies. And then we went back to our relatively small influenza cohort and again find the same autoantibodies. So this is a full circle, shows how studying the different diseases with the same disease mechanism and inbound errors and autoantibodies together shows how powerful this approach is. And now these autoantibodies, we already talked about, it leads to severe COVID-19 and severe influenza pneumonia. And these autoantibodies are one of the most important determinants right after age in determining whether a patient will get a severe infection or not. So if we calculate the odds ratio, which means the frequency of autoantibody positivity in the severe cases versus the general population, or sometimes if we have the cohort to calculate the mild cases, we got the odds ratio between 10 to 100. depends on the threshold, the autoantibody titer we use, and this is very huge. This is not the less than twofold difference of odds ratio you find in, let's say, a GWAS study. Whether having this autoantibody or not is indeed a critical determinant of whether the patient will get severe disease or not. In addition, among the most severe cases, that patient died of COVID-19, 20% of them have these autoantibodies, meaning that 1 in 5 patients died of COVID. That's millions of them worldwide are autoantibody positive. Now, since 2020, we tested these autoantibodies in a variety of severe viral infections, and the results are stunning. I already talked about we have 5% of seasonal flu pneumonia, 10% of tick-borne encephalitis, that we found later, 15% of severe COVID, 20% of the death. The COVID study was replicated by 42 independent cohorts worldwide. And all of them find essentially the same percentage of autoantibody positivity in their cohort, varies mostly because of the age of the cohort and also the threshold they use. This is not discovered by us, but by a Saudi group, that 20% of the MERS patients also had these autoantibodies. We also find 30% of patients with severe infections of live attenuated yellow fever vaccine, usually in South America, 40% of West Nile virus encephalitis, other more rare encephalitis, another live attenuated vaccine that's just on the market, 68% of them have these autoantibodies. To our surprise, 30% of fulminant hepatitis caused by HSV, not the regular HIV, HBV hepatitis. And I'm going to focus on another one, is the most recent finding, a fatal case of avian flu. So at the end of 2024, we saw on the news there is a first severe case of avian flu confirmed in America. Avian flu, mostly H5N1 or H7N9, the most prevalent ones, have been discovered since 1997 that can lead to severe human diseases. These are avian flu that they are zoonotic viruses, so normally they don't infect human. However, there are thousands of cases reported to WHO and claimed it for about 50% of case fatality rate. Now, this number is very misleading because the true infection fatality rate is very low. A meta-analysis and large surveys on people who are poultry workers who are in contact with sick birds all the time show that only 1 to 2% of people who are exposed to avian flu can actually even ever turn seropositive, meaning that the virus ever were able to infect a human. 99% of people who constantly are exposed to this virus never really get infected at all because these are birds' flu. They don't bind to the same receptor in human. So they're really not supposed to infect human. However, there are still thousands of cases of death cases reported to WHO caused by avian flu. Now, one of them appeared in the US. We got in contact with CDC and eventually got connected to the physicians who was taking care of the patient. At that time, the patient was still in ICU, but unfortunately, he died in 2 weeks. We got the patient blood, and what we showed is that this patient indeed have very high titers of autoantibodies against interferon alpha, relatively low titers to omega, and no to beta. This combination is very common. It's the most common combination among patients who are autoantibody positive. And this case is very, very alarming to us, again, because these viruses are not supposed to infect human. Human, we have type 1 interferon response almost function like the wall, the barrier between zoonotic viruses and the human, because these viruses evolved and adapted in birds, which is MX1 incompetent, cannot cross the barrier. MX1 is one of the major ISGs in human that has been shown to be essential to control influenza. And birds don't have functional MX1, so the bird virus, as soon as they get into a host, for example, a normal human, which is MX1 competent, it cannot proliferate. But in patients who have autoantibodies to type 1 interferon, the entire type 1 interferon response is downregulated, and the patients probably don't make ISGs as well as normal people, and now the virus can cross the barrier and infect that person and even leads to severe infections. Now I'm gonna go back to the inbound arrows for a minute because there is a new defect in the type 1 interferon that we discovered recently. I think it's important to talk about. So this is another new defect in the type 1 interferon receptor, if not one. We and other labs has discovered a lot of these different variants, but I want to point to one particular mutation. That's the proline 335, one amino acid deletion, which has the minor allele frequency about 6 in 1,000 in East Asian population, and 0.0008 in Indian population. This mutation is dominant negative, meaning that the patient cells with only one copy of the mutant and another copy of the wild type can still have the same phenotype as a homozygous cell, and they are disease causing. We found that patients with this mutation are susceptible to COVID-19 pneumonia and JEV, Japanese encephalitis virus. But these are the cohort we study. Theoretically, they are supposed to be susceptible to other viral infections just as the other patients. So these are huge numbers of people carrying this mutation. And these patients are not responsive to interferon alpha and omega, but are still responsive to interferon beta. So it's a partial dominant negative defect. This is very important because the number of patients are almost getting closer to the prevalence of autoantibody. So if you add these two together, there are a huge number of people, either with the autoantibody or with this particular mutation, are susceptible to viral infections. And we also wonder, based on our discovery in a patient with all avian flu and autoantibody, and this particular mutation that has the highest prevalence in East Asian countries, in China, particularly in the Guangdong, Fujian area, whether the first case of avian flu death in 1997 in Hong Kong is exactly at the heart of that region in China. It's a 3-year-old boy, so unlikely to have autoantibody, very, very rare to have such a young patient with autoantibody, indeed might have this mutation. And the likelihood, is very high in our hypothesis. So we're actually trying to look back into the samples at that time was sent to the Netherlands, whether we can find this mutation in this patient. So coming back to the alarming disease that we studied in the past 5 years since 2020, we found that both autoantibodies with very high prevalence in the general population and a disease that the inbound arrows in type 1 interferon we thought was extremely rare, but now we start to find mutations that's relatively common in some populations. Maybe we'll find new ones. These two adds together are millions of people who have defects in the type 1 interferon response, and they are almost, we consider them as like weakness on the barriers that as we against the as we human against the zoonotic viruses. We already know the avian flu can cross the barrier in patients with autoantibodies. Now we wonder whether the early case of MERS from camel or even the early case from COVID will get into infect human, adapt human because of these people. And one most scary thing we found is that in that one case of patient with avian flu and autoantibody, the American patient died in Louisiana. The virus, the avian flu, isolated from that particular patient already showed signs of adaptation to human. So they already have mutants that help them to probably bind to human receptor better. And if that really happens in this one particular patient, and if there's a cluster of patients, you can imagine now the virus has a chance to adapt from a zoonotic virus into a human pathogen. And we really need to be aware of these millions of people who have either autoantibody or the inborn errors are actually going to be the patient zero for the next pandemic. We already had 3 coronaviruses, SARS-1, MERS, SARS-2 in 20 years. I would not be surprised if there's another one became a human pathogen in the next 10 years. And we really need to get ready for that. Now I would like to stop my talk here. I would like to thank many, many collaborators from the consortium. There's just no way I can list them all here. I just want to point out only a few most important people who are important for our studies. Jean-Laurent and Laurent Abel as our head of the lab. Paul here is the one who first identified autoantibodies in patients with COVID-19. Shen Ying actually conducted most of the studies on other viruses except for the viral pneumonias. Oili and Emmanuel here helped us to collect the COVID and influenza cohort and also did the math on the autoantibodies. Charlie Rice here helped us for the viral studies, and the 2 physicians who help us to collect the patient with avian flu. And it's courageous for them to collect the samples and treat this one patient. So thank you very much, and I would like to answer if you have any questions. Thank you.
Thank you very much, Dr. Cheng. That was a really beautiful journey through what is a nice novel concept and needs to be shared widely across intensive care units as well, so that they are the people who see these patients the most, so the right questions need to be asked at the right time. Out of interest, I mean, I will be inviting a lot of questions from the audience, and I think there'll be a lot of people with questions, but how old were the majority of your patients whom you tested here? And treatment-wise, what did the testing make you do differently for them?
So we tested the patients from a month old to over 100 years old. I show the preference is mostly the autoantibodies are mostly in patients who are 70 years or older. But that's not to say there are not patients here. The first cohort, I think the average age is something like 55 something, because at the beginning we specifically tested patients who are younger, because the older patients who have other comorbidities, so it's hard to differentiate whether their disease caused by autoantibody or not. So we particularly choose otherwise healthy patients relatively younger. But overall, this could happen in patients with any age. There are more in the elderly patients, but even if a patient who's in their 20s, 30s, you should still test them. The patients with avian flu from Louisiana is 70 years old, but we now have a new case from the 2003 Netherlands avian flu outbreak. The patient is only 50 years old, so could be young and old, any age. And could you repeat your second question?
Thank you, Dr. Chian. I have a small question. That was a wonderful exposition. I was just wondering whether the long-term effects, for example, following COVID, now we are seeing cardiovascular morbidity following severe COVID-19 and also cases which were initially not so severe. So, do you think there is the possibility of long-term problems also being associated with these anti-interferon antibodies, anti-IF1?
It is possible, although we have not found them. So these patients that we found either with influenza or COVID, if they're autoantibody, all of them had a severe acute infection in the lung, or if it's encephalitis in the brain. The long-term effect might be in combination with the underlying cause of the autoantibody. What I didn't touch upon is that we still don't know why these people have autoantibody. Majority of them don't have any other problems, but there is a subset of patients who also have other autoimmune diseases. So they don't only have autoantibody to type interferon, they might also have autoantibodies to other things. And you can imagine these patients could have other phenotypes that differentiate from the patients who just have acute infections. And I think this could be an interesting field to study.
Thank you so much. We now, Dr. Vignesh, please.
Yeah, Dr. Zhang, hope I'm audible to you. Am I audible?
Yes.
Yeah, okay. So, what do you think is the determinant for developing these autoantibodies to type 1 interferons? Age is there, but do you think any other specific determinant for these development? Thank you.
Yeah, so that's one of the key, that's half of the lab is now devoted to study this. What I can tell you is that, first is we don't know. But what I can tell you is that these autoantibodies are not new. We already know they exist long before the COVID pandemic because patients with AIR mutations, the AIR gene in the thymus that leads to APS1 syndrome, these patients from birth have sky-high autoantibodies to type 1 interferon and other cytokines. So we already know if the thymus cannot present self-antigens to delete T cells in the thymus, can lead to autoantibodies to type 1 interferon, although these are extremely rare cases. We also identified 5 new genetic defects. All of them affect either the AIR expression or AIR function in the thymus. Or, again, these are rare cases, but these patients are, most of them have autoantibodies to type 1 interferon. On top of that, we discovered that patients who have thymoma, so these are very rare tumor in the thymus, that also leads to autoantibodies to type 1 interferon. So put all these together, essentially, if there is a defect in thymic function, or even just a tumor inside the thymus, can lead to these autoantibodies. So our focus is on the T cell tolerance. When it's broken, it will lead to these autoantibodies. Now, these cases definitely do not explain why the general population have these autoantibodies. This is something we are trying to study, but it might make sense why elderly people who are more likely to have type 1 interferon autoantibodies, because the thymus essentially decreased the size and went into like a proportion of the function after age 70 to 75, which is similar age when the autoantibodies start to increase in the general population. Again, this itself does not explain why only a proportion of the elderlies develop this autoantibody, and this is what we're trying to study in the lab.
Yes, Dr. Amit.
So my question is regarding the IFNAR variants. So IFNAR variants, there are different populations of different variants. For example, one of the variants in the Polynesian population also have a very high frequency. And similarly, I've showed that one of the variants is very common in East Asia. So why do you think that these variants have a selective advantage? Why were they selected in the population? I mean, why would there be such a high frequency? And the other question is regarding the selectivity for the type 1 interferons. I mean, why is this selective between alpha, beta, and omega? What would be the reason behind that?
Yeah, so both are very important questions. So the Polynesian variants you talk about is on IFNAR2. That's actually we found in patients who died of MMR vaccine. And the percentage is even higher. And also there's population in the Inuits, in the very cold Denmark region, also find another variant in IFNAR2, very high percentage in that population. Now the two IFNAR2 variants, they are probably because these populations are isolated, either in Polynesia, in the island, or in or almost at the North Pole. So these are isolation. The IFNAR1 is not. These are in China, in India, in Vietnam. These populations are highly mixed, not isolated. We don't know whether this IFNAR1 variants have any advantage, for example, why they still exist. We really don't. But more and more we find variants like that. If you look at NOMAD database, the variants that are extremely rare in overall different populations, often they are high in a particular population. This is a very common phenomenon in all genes, in all variants. And honestly, we don't know why. The second question is the different subsets. Why humans have 17 different type of interferons? Some of them are expressed in the limited tissues, but it doesn't make sense for alpha and omega, which is essentially expressed everywhere, and why we have 13 different alpha who are essentially the same cytokine. And this is not only limited to human. Fish, some fish, I think salmon has like 21 different ones, 25 different ones. Mouse has one that human doesn't have. This has been a question since the discovery of type 1 interferon 50 years ago. A short answer is still we don't know why, but we have unpublished data I can tell you is that now we have 5 patients with a defect inbound arrows in interferon omega only, 5 of them, and they have severe COVID-19. So it shows that just the lack of one subtype, omega only, is sufficient to cause the disease. We still don't know why. We've been working on these patients for over a year now. We don't know why these patients have perfectly fine alpha and perfectly fine beta. Just lacking omega is sufficient to cause severe COVID-19, and we're still working on that. But that's the first genetic data to show that indeed each subtype is probably have a different function. What's the mechanism? We still don't know.
Thank you. Do we have any more questions? Yes, go ahead. Can someone get him a mic right at the back? Yeah.
Ma'am, Ma'am, in general population, autoantibodies are present for interferons only or are for other cytokine also?
You mean these people who have autoantibodies to type 1 interferon, do they have autoantibodies to other cytokines? Is that your question?
Yes, ma'am.
Yeah, so we now have a assay that can test 46 different cytokines, autoantibodies to 46 different cytokines, and there are large array that tests hundreds of cytokines for their autoantibody binding at the same time. So in some cases, yes, we can find other antibodies binding to other cytokines. We yet don't know whether they're functional, meaning if they are neutralizing or not. And also, we have not observed any pattern. So let's say they always have an autoantibody to, let's say, IL-12. We don't see these patterns. My answer will be, this is probably going to be a complicated result. That is, there's not going to be clearly, these people are they don't just have autoantibodies to all cytokines, or even a pattern of combination of cytokines. It's probably case by case, they're different. Some of the patients, they do have autoimmune diseases. They do have autoantibody. Even some of them could have SLE, lupus. So those are probably have autoantibody to a bunch of different antigens, but there's not a clear pattern, or there's not a universal autoantibody to all cytokines or all antigens that we can observe.
Shall we close the session if there are no more questions? So, on behalf of the organizing team, we wish to profusely thank Professor Chian for this wonderful presentation that has been so thought-provoking and has generated a lot of discussion. Thank you so very much for agreeing to be with us and giving this great lecture. Thank you. The next speaker will be Dr. Dharmagat Bhattarai, who's going to speak on ARPC1B defect, his experience from Nepal. Dr. Dharmagat is a good friend. He's an associate professor in the Department of Immunology, Rheumatology and Allergy in the College of Medical Sciences, Bharatpur in Nepal, and Norvik International Hospital, Kathmandu. He's also the scientific representative from Nepal for the European Society of Infectious Diseases, the Asia Pacific Society for Infectious Diseases, the IPOPI, and HAE India, and Emerge Press. Please carry on.
Thank you, ma'am. Good evening, all of you. I am really thankful, and I have sincere gratitude to ISPID for inviting me and giving this opportunity in this August gathering. Today I am going to present a story of an adventure. This is the story about ARPC1B deficiency, the experience from Nepal. I have nothing to disclose. As you all know, Nepal is a very kind neighbor country in the lap of Himalayas. And since this is the story of adventure, my journey is just like the journey of Edmund Hillary, going in the mountainous trek without any track till then, and with so many confusions, without any guidance, and this journey started in the cloud. But when I reached at the top of the mountain, the view was spectacular, and I am going to describe one of the spectacular view of this journey of adventure. So I have diagnosed these many genetic defects in Nepal, but this is the story of a special love story for me with the cytoskeletal rearrangement defect, specifically in the ARPC1B deficiency. As you all know, the cytological rearrangement results into the different genetic defect, especially dominated by the blood and immune phenotypes, and out of them ARPC1B deficiency is one of the important addition, first described in 2016 and having a exciting story. And this story in Nepal started in 2020 when I was very depressed with the corona, with the jobless period. And one patient came with the very confusing presentations of growth failure, recurrent infections, multiple allergic episodes, recurrent pneumonias, boils, otitis media, joint pain. Probably there were many manifestations, and child was already admitted 80 different times in different hospital and treated with almost all kinds of labelings like eosinophilic colitis, IBD, ITP, tuberculosis, and many more. And when I saw the reports and when I investigated, it revealed some thrombocytopenia, anemia, high inflammatory markers, high Ig values and high IgA value and reduced IgM and IgG. ANA was co-speckle positive and raised value of the NK cells but adequate B cells and reduced CD4 T cells. So it was quite confusing. But when I started to explore further, because I was also in the cloud, I was just like a crow in the cloud, and I started to explore the history of any other children in the family, and I found that they were not consulting for this child, but that child still had some bleeding manifestation in the form of blood in stool and skin rash and eruptions, and they were treating conservatively. And I investigated myself for these tests like IgE, IgA and CD19 B cells and everything on my own interest, and I found the similar kind of presentations. So that led to further confusion, but I was quite sure from my teachings, from the great teachings in the PGI Chandigarh under my teachers, that when this kind of presentation, this is 100% primary immunodeficiency, but which one? I did not know. So I thought about combined immunodeficiency, I thought about immune actinopathy also, because the presentation was quite similar to Wiskard-Daltrich syndrome, but it was not fitting on it. So with this presentation, I started to ponder in the jobless period, but at the same time, Volpi et al. From Italy had published this interesting paper, and I had this thing in my mind that 3 patients from Nepalese origin had similar kind of presentation. This was a very fortunate coincidence for me, and I jumped into the whole exome sequencing, and I found that this genetic defect on the ARPC1B gene was happening on these 2 children. So that was the beginning. But this story did not stop there, and there were some more patients, and it came to the news also, and there were some other patients. I am not going into the detail because the presentation is similar, and it is quite severe phenotypic presentations of this disease in comparison to the presentations described in the past from the Volpi et al.'s paper. And there were even more presentations which are quite new and novel kind of presentations that were found in my cohort. So we published this paper, and today I am going to deep down into this these presentations of ARPC1B deficiency. If you see the patients from the in the picture of Nepal, most of the patients are from the one of the province, and most of these patients have origin of this province, and this thing stimulated me about the common origin or founder effect of the gene. And when I further explored about the endogamous marriages, I found that there is the quite overlap in the clan from the father's clan, mother's clan, and paternal and maternal grandmother's maiden clan. So there was the quite overlap, and this proved that though there was no consanguinity in the patients, there is quite significant endogamous marriage resulting into the autosomal recessive kind of disease. And after the publication also, it was published last year, I have diagnosed further 10 patients, previously 14 patients were there, and further 10 patients are diagnosed with this disease. If you, probably you are aware of it, only probably 64 to 65 patients are reported from around the world, and almost 1/3 patients are diagnosed in Nepal. So what is this ARPC1B? So this ARPC1B is responsible for this ARP23 complex, which has 7 polypeptides. And out of these 7 polypeptides, the main actin-related protein 2 subunits are ARP2 and 3, and the gene deletion in ARP2 and 3 results in embryonic lethality, which is proven in mouse. But the other subunits, which are regulatory subunits, these are ARPC1 to 5. Out of these, ARPC1 has 2 isoforms, ARPC1A and ARPC1B, and these are encoded by different genes, and other ARPC subunits do not have the common sequence motif. And if the defect is in ARPC1B, it causes myriads of symptoms ranging from infections, allergy, autoinflammations, and different kinds of other autoimmune and autoinflammatory manifestation and allergic manifestations. So most of the patients reported around the world, they have presentations of eosinophilia, elevated IgE and IgA, and thrombocytopenia. These are the very usual kinds of presentations described, and most common variants C.64 plus 2 T2A, this is the founder variant found in Nepal, and other than that there are other 2 variants which are reported quite commonly from Slovenia and Mexican descent. And if we compare the phenotypic presentations in this kind of disease, the first column represents the total number of patients with other variants and our variants, and in the another column you can see the comparative presentations of the different laboratory parameters and the clinical features that are present in other variants other than Nepalese founder variant and in our variant, and you can see significant differences in the multiple different laboratory and clinical parameters. So this proves that there is the distinct phenotypic variation and distinct phenotypic difference in comparison to other variants around the world. Similarly, these are the clinical and laboratory parameters differences from our variant, patients with our variant and patient with other variants. One significant finding that was found in our patients that is not quite described in the literature is arthritis in ARPC1B deficiency, because 5 of our patients out of 14, this data does not include the new patients which are not published. Out of 14 patients these are published, 5 of our patients has positive rheumatoid factor, and these patients were diagnosed as the case of RF positive rheumatoid factor, GI, RF positive polyarticular GIA. But in these patients, ACPA was also positive. So this is probably a lesson for the pediatrician, rheumatologist and immunologist. Even if you diagnose RF positive GIA, probably the journey has not ended there. We should think further, and it is proven now. And in the new patients, 10 patients which I have diagnosed, 3 more patients have RF and ACPA positivity. So this is the very confusing things for the pediatrician, but probably we should think about the disease when there is the founder variant like that in Nepal. So out of the nearly 3 dozen patients till that time last year with the ARPC1B deficiency reported, arthritis has been described only in 3 patients till that time, probably maybe 36 patients till that time. And similar to our report, RF positivity has been observed in the absence of arthritis in 1 patient of ARPC1B deficiency. And in a large cohort of IEI, as per IUI's categories, RF positivity was noted in only 15 patients. So you can see the difference. So in the use cohort of 300 patients with different kinds of inverter of immunity, it was reported only in 15 patients. And out of only 14 patients, we have 5 patients have RF positivity. Probably this is very significant. So there is the prudence of considering monogenic defects in patients with RF positive GIA who have other clinical immunodeficiency signs or allergy. And this is the another representations with the reduced normal or elevated levels of different vaccine response, immunoglobulin level and the cell counts. You can see the difference in the presentations among the patients. Another interesting addition is the celiac complex. So notably, significant anti-TTGA titers were found in 5 patients out of 14 in my cohort. And prior to diagnosis of ARPC1B, these patients were labeled as celiac disease.
with the elevated ATGA, but there was no benefit with the gluten-free diet. And CD-like features are well described in common and rare inborn error of immunity if you go through the literature. But the reports like CD-like illness in the immune actinopathy like Whisker-Eldridge syndrome are quite infrequent. And whether the apparently low frequency of CD-like illness in the immune actinopathy is because of the anti-actin antibodies remains speculative. And as per the ANA pictures, these are the real pictures of my patients, and we do not see any kinds of anti-actin antibodies in this picture. And regarding the novel features, these features are not quite described in the literature out of these 36 to 40 patients still last year. So we found the prominent frontal bony hypertrophy, hemihypertrophy, pigmentary skin changes, and typical kind of hyperkeratosis described, found in the patients. And we also found the difference in the skin pigmentations in our patients from the time of presentation to us and over the time when the child is being treated with the different modalities of treatment. And we also found the enlarged distal phalangeal tips when the child is living until the late childhood. And another interesting finding, as I described, is the founder variant effect of this variant C.64 plus 2 T2A in Nepal. And we did the homozygosity mapping and haplotype analysis, and this proved that there is this region, this variant allele is located in the only shared region of homozygosity of size more than 1 MB, and the probands analyzed have the similar homozygous haplotype. We had one patient of compound heterozygotes, but even in that patient, the heterozygote polymorphism was in trans with the polymorphism selected for the haplotype analysis, and this also further strengthened that the proband with the compound heterozygous variant has also the similar heterozygous haplotype, and many heterozygous polymorphism around the area of this split site, this is the split site mutation in this patient, was absent in other probands and located in trans with the polymorphism. This further strengthens the proposition of the founder variant. So this was the founder variant. Now this thing is further reinforced by another paper that is published 5 days before from Australia. I have not included in the slide. But interestingly, when I am talking about the region of C.64 plus T2A, that is the founder variant, there are other reported variants in the same region of exon 2 to intron 2 junction in the ARPC1B. You can see from the different countries. And only one report from Volpi, the patient is from Australia, but that patient is again from the Nepalese origin. But interestingly, there are other variants which is not C.64 plus 2, but this genomic location is significantly, conspicuously we can see that this is devoid of various nucleotide sequence associated with the mutational hotspot like palindromes or polypyrimidine sequences. This further strengthens the proposition of the founder variant. And regarding transplantation, I want to touch a little bit about it. 100% patients have recurrent infection, 60% have severe vasculitis in gross, and 80% responded very poorly with the conservative treatment that were tried in the other cohorts described from Mexico, Slovenia and Italy, and 20%, like one patient out of these 5 patients who undergone the bone marrow transplantation, one did HSCT without trial of immunomodulator. If we compare the world data and Nepal's patients who underwent the hematopoietic stem cell transplantation, this is a small number but significant number for the country, resource limited country like Nepal. And we had all kinds of donors like match related, match unrelated, and haploidentical parents were also used for the source as a donor. And there were not much post-transplant GVHD, but there were incidences of the vasculitis, sepsis and uveoretinitis prior to transplant in one of the patients. And there are history of microangiopathy and JC virus-related encephalitis in the other patients described from other part of the world. And transplant outcome till now, 1 patient died out of 5 patients transplanted, underwent the process of transplantation, and other 4 patients are doing well and they are in the follow-up. Regarding the outcome or survival, the overall survival at 2 years of follow-up was 73%, and this is significantly exciting news for the immunologist working in the resource limited setting. To conclude, ARPC1B deficiency is definitely emerging as a serious immunoactinopathy in the world, with the large volume in the Himalayan belt for some founder effect, and notable clinical features which are noted in our study, like RF positive chronic arthritis, celiac like phenotype, hyperkeratosis, broad fingertips. These are important addition to the phenotypic description of this disease in the world of immunoactinopathy, and greater proportion of otitis, gastroenteritis, IBD like features, and high IgA values, just like that observed in the other immunoactinopathy like viscodilatitis in the patients of this particular variant, because these are not very much described in the cohorts described from Greece, which are described in the patients of Afghan origin. And homozygosity mapping revealed that the gene and variant is contained within the overlapping region of homozygosity. This is a very exciting news for me, as well as the pediatricians working in the Nepalese or South Asian region. Probably they are migrating to different regions, as I showed in the picture. And this variant, C.64 plus 2T2A, is the founder variant in Nepalese population with the severe and diverse clinical phenotype. This ends my presentation, and I have sincere gratitude to everyone who helped me for the genetic adventure and the clinical adventure for the diagnosis and treatment. And I am also thankful to ISPID, Priyankar sir, and all teams from Kolkata to invite me to this August gathering. Thank you.
Congratulations, Dr. Bhattarai. They usually say that fortune favors the brave, but I think fortune favors the brave more up in the mountains. Anyway, I have a small question to ask. Did you find any bleeding manifestations in these patients with normal platelet counts, and were you able to look at platelet function in your patients?
Never. Bleeding, almost all the patients, not 100%, but almost all the patients were having bleeding manifestation, but thrombocytopenia was present in all of them. And specific differences is mean platelet volume was not less, it was normal. And secondly, we did not study the platelet functions, but only we saw the differences in the platelet size and platelet numbers, and over the how is the platelet count over the course of treatment, like conservative modalities of treatment. We treated most of the children with IVIG and sirolimus, and some of the patients with mycophenolate also, because it was tried in the different patients of Slovenia and Italian reports. So during that time, the platelets significantly improved, but we did not study the platelet function.
Thank you. Thanks once again for your wonderful work, and we now take questions. A couple of questions, if there are any. No. Okay. So we'll move on then. Thank you.
Thank you.
Thank you once again. We now have another interesting session by Dr. Peter Maguire, who is head Metabolism, Infection and Immunity section at NIH Bethesda at the USA. His major research interests are an interplay between metabolism and the immune system through a translational research program involving patients with inborn errors of mitochondrial metabolism. So we used to think that inborn errors of metabolism were on the one hand separate and inborn errors of immunity on the other hand. But I think, sir, after your presentation, all that is going to change.
Thank you so much for that introduction. Can you hear me okay?
Yes.
Okay, I just wanted to make sure. So first, I would just like to thank the organizers for the opportunity to present our work. I'm sorry I couldn't be there in person, but I wish everyone a good evening, and I hope you're enjoying the meeting. So I am actually a biochemical geneticist. I take care of kids with different types of inborn errors of metabolism. And the work I'm going to talk about today is really kind of the culmination of about 20 years of studying these patients, especially during episodes of infection, where patients can experience something called metabolic decompensation. So today I'm going to talk about infection and immunity in mitochondrial disorders. I have no conflicts of interest to declare. So I'm sure many people in the audience are familiar with mitochondria and thinking of it as essentially the powerhouse of the cell. That's kind of the euphemism that everyone uses to describe mitochondria. But clearly, mitochondria have a lot more functions besides providing energy, or I should say, extracting energy from our food. So it is involved in things like OXPHOS and the TCA cycle, beta oxidation of fatty acids, synthesis, as well as synthesis of heme, cardiolipin, and steroids. But it can serve other important functions such as calcium buffering. Free calcium should not be running around in the cell because it can activate certain cell death programs as well as other cell stress programs. The mitochondria is also involved in stress responses. Of course, apoptosis, as everyone's familiar with. But more importantly, what's emerging recently is it also serves as a signaling molecule with reactive oxygen species, as well as mitochondrial DNA as a damage-associated molecular pattern, as well as various metabolites that are produced either from the Krebs cycle or from other aspects of metabolism, which can actually affect things like gene expression and contributes to this crosstalk between the mitochondria and the nucleus. So mitochondrial disease is the most common inborn error of metabolism. It's a group of diseases, essentially. So when I say mitochondrial disease, it's kind of like saying cancer. It's a group of diseases, genetically heterogeneous, but share this common feature of oxidative defects in oxidative phosphorylation. So it affects about 1 in 4,600 individuals. So you've likely bumped into someone on the street who's affected by mitochondrial disease. And mitochondrial dysfunction is actually a good model, and these patients can serve as a good model for understanding mitochondrial dysfunction in common disease, especially things like immunometabolism. So in terms of studying these patients, yes, our primary objective is to help them, in terms of their disease and how to improve their quality of life. But they also serve as a good biologic model for understanding immunometabolism and the importance of the mitochondria in immunometabolism. With regards to the genetics, these disorders are due to pathogenic variants in either mitochondrial DNA or nuclear DNA. And as I mentioned, this results in OXPHOS deficiency, which results in decreased ATP production, increased in lactate production, because they have to be able to produce ATP by another source, so they burn a lot of glucose, and lactose is produced as a result, as well as an increased production of reactive oxygen species. Mitochondrial disease is a multi-system disease affecting multiple organ systems, especially organ systems that have high energy requirements. And those organ systems with high energy requirements tend to be kind of the hallmarks or the pathogenic hallmarks of the disease or the clinical hallmarks of the disease. However, all tissues can be affected, even tissues that we may not recognize as being part of the clinical phenotype of mitochondrial disease, which I'll talk about a little bit later. So there's high morbidity and mortality in these patients and decreased quality of life. And although there are newer treatments coming out for various mitochondrial disorders, in general, treatments are very limited. We don't have a magic bullet at this point. So we do a lot of treatment of the various complications that result from mitochondrial disease. So an important question about host pathogen interactions in mitochondrial disease, as I mentioned, this was I started my research program over 20 years ago at Mount Sinai in New York City. And so it started with the basic question of why do children with mitochondrial disease actually develop disease progression during viral infection? So viral infections can be very devastating for these kids. This is not one of my patients, by the way. This is just an example from Google. But what it does is it gives kind of an idea of the level of care that these kids may actually require with what are considered garden variety viral infections. And what can happen to these kids actually is they can develop something called metabolic strokes. So stroke is probably the improper term, but we call them metabolic strokes because they're not vascular in nature. They're probably related to failure of bioenergetics. And these strokes can actually be targeted. So they can affect things like the basal ganglia as well as the brainstem, which I'm showing you here in the red arrows. And so this is a patient that we actually follow in the NIH Clinical Center who has Leigh syndrome due to homozygous pathogenic variants in SIRF1, which is a complex 4 deficiency. And you can see that he actually has bilateral areas affected in his brainstem. As a result, he actually has a tracheostomy because this is where the respiratory drive center actually sits. So he actually receives artificial ventilation at night. So these infections can be particularly devastating and actually cause disease progression in these individuals. And so what we normally see is kind of this stepwise regression that the patients will experience. So as I mentioned, intercurrent viral infection is a leading cause of metabolic decompensation in these patients. These can be life-threatening episodes, but if they actually survive these episodes, they have a decline in their level of functioning. So in this graph on the right, what I'm showing you, normal development, of course, is in the purple line, whereas kids with mitochondrial disease kind of experience, they will have they do not have normal development, obviously, but with that, these infections, they will have a stepwise decline in age-appropriate skills. And so when I came to NIH over 15 years ago, I started this program to understand infection and immunity in kids with mitochondrial disease. So we have 2 studies that we actually conduct out of the NIH Clinical Center, one of which is the NIH mini study, and the other one is acute infection and mitochondrial disease. Through these 2 protocols, we actually study infection and immune function in these patients with mitochondrial disease. We do it at the NIH Clinical Center, but we also have these home monitoring protocols that we can actually do to get blood samples and do things while the kids are actually sick at home. And we want to understand infection history as well as immune function, and then any disability that may have resulted from having viral infections. So what have we found so far? So there are a number of factors that actually modulate host pathogen interactions in children with mitochondrial disease, and there are a number of positive factors and a number of negative factors. So on the positive side, our patients are highly adherent to what are called risk mitigation behaviors. So in other words, those non-pharmacologic behaviors that patients may engage in, such as hand washing and social distancing. And so during the pandemic, these families were already used to engaging in these behaviors because there was this inherent fear during flu season that their child will decline. The other thing they're highly adherent to is the pediatric guidelines that are recommended in terms of long-term care. So these patients are normally cared for not only by either a geneticist or a neurologist, but also their pediatricians, and we work with them very closely to ensure adherence and regular follow-ups. However, this is outweighed by a fair number of negative factors which we have reported on. The first of which is viral infections is inevitable. So regardless of their adherence to these various measures, they actually get a number of viral infections. Vaccine titers in this population are actually lower as well. They've impaired humoral immunity. They've impaired T-cell immunity. And this is in patients as well as mouse models, which we've worked with to study this disease. They also have increased inflammatory tone. So there's this kind of push and pull, essentially, of immune activation in terms of having high inflammatory tone and impaired adaptive immunity. One thing that's important, especially with this increased inflammatory tone, is that they have increased cytokine toxicity. So tissues that are affected by mitochondrial disease, when exposed to inflammatory cytokines, even at levels which would be considered normal for an infection or not due to mitochondrial dysfunction, those tissues actually become dysfunctional themselves. They do not tolerate inflammatory cytokines. Also, there are various aspects of viral pathogenesis that are also involved in mitochondrial disease, which I'm going to talk to you about in a little bit later in this talk, which is very important for understanding their disease. And what's most important too is as geneticists, we are continuing to recognize that individuals oftentimes have more than one disease. And so we are currently working on a study which involves secondary variants in antiviral or immune genes, which may alter the course of individuals' response to infections. So one of the first risk factors I mentioned for enhanced morbidity is viral infection is essentially inevitable. So this was a household study that we actually did where we were able to sample on a platform that we standardized, sample capillary blood from individuals, which they would then return to us by post, and we could look at their viral exposures over time. And so this was a study of 15 households during 2020 and 2021 during the pandemic, involving 17 children with mitochondrial disease. And during that time, despite the fact that they were engaging in all these various risk mitigation behaviors, they were exposed to more than 38 respiratory and GI viruses, which was about on average 5 viral infections a year per child. This resulted in 34 episodes of illness with 6 hospitalizations. And in those 6 hospitalizations, there were 5 neurologic events that occurred. So what I mean by that is there was disease progression where their CNS lesions either got worse, or they developed CNS lesions for the first time, or their seizure frequency actually increased. So an exacerbation essentially of their underlying neurologic condition. With regards to innate immunity, as I mentioned before, they tend to have this underlying increased inflammatory tone. So this was a paper that we published back in 2022 where we looked at inflammatory and interferon gene markers in patients with mitochondrial disease. So this was kind of whole blood RNA-seq. And the part that makes this interesting is that this was a genetically and age-diverse cohort of individuals with mitochondrial disease. At the same time, we also looked at various mouse models of mitochondrial disease to see whether the immune signatures persisted across different species. And so we compared these mitochondrial disease patients to, as I mentioned, other cohorts and animal models, and looking at gene set enrichment analysis. So looking on kind of the pathway level to see what was actually enriched, we saw increased inflammatory signaling, type 1 interferons, increased interleukin beta-1 response, antiviral responses, as well as enhanced signatures for monocytes and dendritic cells. On the adaptive side, we saw a decrease in signatures for T cells and B cells, so kind of further substantiating this kind of difference between innate and adaptive immunity in kids with mitochondrial disease. Another risk factor, now turning to adaptive immunity, actually has to do with impaired adaptive immunity in these patients. So in this particular paper, we looked at T cells and T cell function. And this paper first captured looking at memory immune cell populations in patients with various types of mitochondrial disease, which you can see here on the left, looking at and the different designations for the different types of complex deficiencies that they had, or mitochondrial DNA depletion. And while you can see they have a decent number, sorry, these hash lines are essentially the normal range for these values. And so you can see that they have kind of within normal range for naive cells, but they have very low levels of memory T cells. We actually were able to capture a number of patients during infection. This is 3 patients with various types of mitochondrial disease, and we're able to capture a number of different laboratory parameters. And what was interesting is that when they actually developed a spike in lactate, which is consistent with metabolic decompensation, in other words, this kind of increased impairment of mitochondrial activity where you have this increased reliance on glycolysis and you have this spike in lactate. And when that happened, you had a decrease in their immunoglobulin levels as well as a decrease in their white blood cell counts. And so this is consistent with mitochondrial disease in general, where mitochondrial disease is a disease of stress. So if you sent these individuals to be evaluated by an immunologist, they may or may not have certain features that may define them as primary immunodeficiency. However, if you look during infection, in other words, this kind of incompatibility of the organism with its environment, you will find these things you are more likely to find these things, these aspects of immune dysfunction. Also, in terms of adaptive immunity, not only do we have issues with T cells, but we also have issues with B cells. So this was a paper where we actually looked at the ability of B cells or plasma cells, or especially long-lived plasma cells, in patients with mitochondrial disease to be able to produce antiviral antibodies. And this was using a phage display immunoprecipitation library, which contained all these different viral epitopes for all viruses, essentially, that are known to infect humans. And what we found were essentially limitations in the antiviral repertoire, where we found reduced titers reduced titers as well as reduced polyclonality. So not only were they not robustly producing antibodies as much from their long-lived plasma cells, but also their ability to recognize different epitopes within various antigens was also reduced as well. So as I mentioned before, having immune reactions is actually problematic in terms of patients with mitochondrial disease, in that their tissues experience enhanced cytotoxicity with inflammation. So this was work that was actually done in a mouse model of mitochondrial hepatopathy. So in this mouse model, we selectively knocked out complex 4 in the livers of these animals and actually infected them with PR8 influenza, which is a common kind of, this is a laboratory strain, by the way, of influenza that you can use to infect mice. And our patients are infected with influenza. And so we looked at essentially how the liver responds to this kind of systemic inflammation. And what we found is that in the liver, there was enlarged hepatic mitochondria, which you can see here on the right lower corner, and also these large light-colored droplets, essentially, are lipid droplets. So they developed steatosis, so disease progression. And so what was interesting is that by blocking TNF-alpha, which was kind of a central node within the various cytokine profile that we were actually seeing in these mice, we could actually abrogate all of these findings, and the mitochondria actually returned to normal, and the steatosis actually resolved. So this is one of the first pieces of evidence that the immune system actually can be a major driver of disease progression in a mouse model of mitochondrial disease. So overall, kind of as a summary, just to give you an idea of what we see in these patients with mitochondrial disease. This is just from a review that I recently published with my colleague Philip West, who's at the JAX lab in Bar Harbor, Maine, where you have essentially this increased innate immune function, or this increased inflammatory tone that exists in the patients with mitochondrial disease, and that is due to a number of factors, including metabolic reprogramming, including the release of DAMPs from damaged mitochondria, where you have these pro-inflammatory cytokines, chemokines, and interferons. And then, as I mentioned, on the adaptive immune side, you can have things like T cell dysfunction due to purifying selection, as well as decreased antibody responses. But what I want to talk about for the remainder of my talk, and really to help you think about maybe mitochondrial disease in a different way, beyond what we kind of canonically know as far as the clinical features of this disease, is are there any additional factors that lead to increased morbidity during infection in patients with mitochondrial disease? And so it's difficult to study infection in mitochondrial disease. These patients, as I mentioned, are in extremis at some of these points, and clinical care takes priority. So it's important to use, and it's also unethical, obviously, to give infections to children with mitochondrial disease or withhold care during these episodes. So it's important to move beyond what we have seen as clinical observations, understand mechanisms, and we can do that using mouse models of mitochondrial disease. And so we have this system which we use in the lab where we have used seasonal influenza A virus to infect mouse models of mitochondrial disease. And this is work that was done by a former postdoc in the lab, Amanda Fuchs, looking at viral pathogenesis in the setting of mitochondrial disease to identify if there are any other factors that may actually be involved in mitochondrial disease. So here we have the NDUFS4 mouse, and this is a model of complex 1 dysfunction. So these mice have essentially respiratory chain complex 1 dysfunction and therefore OXPHOS deficiency. Now I should point out, OXPHOS deficiency is never 0, right? That's incompatible with viability in any organism, but they do have reduced OXPHOS dysfunction, excuse me, reduce OXPHOS function. So these mice are actually infected with a mouse-adapted strain of influenza, and this is known as the X31 influenza A virus. And this is a laboratory-produced virus, which is kind of based on PR8 a little bit. And so this virus is more representative, actually, of seasonal flu. So PR8 is another form of mouse-adapted virus, which is more kind of pandemic. This is more considered seasonal, so a little bit of a lighter kind of flu. So these animals are actually infected and followed over a series of days post-infection, where we look at various time points of various aspects of infection. And you can see here on the left, one of the best ways to determine how sick a mouse is to follow their weight over time. And you can see here that mice that are infected, NUFS4 mice, which is in pink, which are infected with influenza, actually lose more weight than the wild type. So not only do they lose more weight, but their clinical scores are actually worse as well. So the clinical severity of this infection is actually greater in these mice. So the reason for that is it turns out that the mice that are the NUFS4 knockout mice actually have higher levels of influenza in their lungs. So here we are looking at viral load, and this is by qPCR, but we've also validated by other methods. And looking at the time course of viral loads across this infection. By the way, the mice do recover from this infection, so it is more representative of seasonal influenza. Some do die, but in general they do recover. And you can see over time that they have a spike essentially in viral loads at about day 4, and then while the wild type is cleared by day 7, they still have persistence of virus. It's still detectable in the lungs of these mice. And then of course both mice are actually negative by day 10. And of course, with this comes an inflammatory response, as you would expect, and a higher inflammatory response seen in the knockout as measured by interferon gamma, IL-6, and TNF alpha. The other thing that we notice is that our mice actually have a decreased lifespan by about a week or so. And a lot of this is due to actually about a 20% drop in mice within the first couple of days of infection, but also we do have some mice who also die shortly after the infection. So since we work on immunometabolism, one of the first questions we asked is immune dysfunction in the immune cells themselves responsible for kind of these higher viral loads? And so there are many, I know this is an audience of immunologists, and so I fully realize that there are many different cell types that are involved in being able to combat this viral infection. We just looked at 2 particular ones, one of which are macrophages and one of which are T cells. And so what we did is knocked out essentially NUFS4 in either macrophages or in T cells. And you can see here that you do not get these elevated viral loads if you knock out in those 2 specific tissues. But however, in the knockout, as I'm showing you here, still has these elevated viral loads. So it started to force us to ask the question of, yes, it could be in other immune cell types, but more importantly and more interestingly is, could it be in the lung itself that is actually facilitates viral infection in these animals? And so does mitochondrial dysfunction in lung tissue contribute to the higher viral loads and increased morbidity? And the reason I bring this up is because we don't think of the lung at all in terms of mitochondrial disease. It's not necessarily considered an organ system that has high energy requirements. You don't normally see it in terms of clinical phenotyping. So this may be a condition that's brought about by stress or incompatibility with the environment, in other words, viral infection. And so what's interesting is that the metabolic reprogramming that goes on during influenza infection includes things like increased use of glycolysis, increased production of lactate, increased lipid synthesis, increased glutamine incorporation into the TCA cycle. This increased branch off from glycolysis of these accessory pathways, which I'll talk about in a minute. And then a depression of OXPHOS. Well, this is the exact same metabolic profile that mitochondrial disease actually develops in the first place because of mitochondrial dysfunction, this kind of overuse of glycolysis, essentially. And so the question is, are these cells or these tissues within the lung already primed to be able to receive the virus and help facilitate the viral life cycle? And so first thing we want to do is actually show that in the lung tissue of these mice, they actually had, complex 1 deficiency and oxphos deficiency to begin with. So to answer that question, the first thing that we did was look at complex 1 activity by enzyme activity in the lungs of these mice. And this is looking at the lung tissue. So these lungs were perfused. And you can see here that complex activity in the NDFS4 knockout mouse is low, or essentially 0, as we would expect for these mice. And then what we did is, this is a technique that was actually developed by a talented postdoc in the lab, Jose Marin Franco, where we were able to use, this is essentially a flow cytometry technique to look at bioenergetics in lung tissues. So you can use cell surface markers to determine the different components of the lung, but then you can use various inhibitory drugs to target certain metabolic pathways, and then use ATP as a readout, essentially, to determine the bioenergetic profile of these various tissues or cells within the lung. So it's single cell bioenergetics. It's kind of like CNIF, but the problem with CNIF is, for those of you who are familiar with CNIF, the problem is CNIF is based on protein synthesis as a proxy for bioenergetics, and CNIF actually falls apart when you actually have true oxidative phosphorylation dysfunction or any perturbations in glycolysis. So CNIF is good for more kind of wild type situations, but not really good for true oxphos dysfunction. And so what we saw in our these are respiratory epithelial cells that were identified by surface markers. They have a lower oxphos dependence, so in other words, lower oxidative phosphorylation, and increased use of glycolysis. So the reason that this is important is because of those accessory pathways that I mentioned before that branch off of glycolysis. And so one of those accessory pathways actually is the hexosamine biosynthesis pathway, as well as the de novo sialic acid biosynthesis pathway. And so our hypothesis was that the partitioning of glycolytic intermediates into the de novo sialic acid biosynthesis pathway. The reason this is important is because sialic acid is the self-surface receptor for influenza. So our hypothesis is there's more spilling into these pathways, more expression of sialic acid, more viral binding, more infection, mice get sicker. And you can see here in green that the hexosamine biosynthesis pathway is a branch off from glycolysis at fructose 6-phosphate. And that feeds into the purple down here, which is the de novo sialic acid biosynthesis pathway, which then leads to sialic acid on the surface of cells. So we looked in the NGFS4 knockout mouse, as well as a mouse lung epithelial cell line that we CRISPR edited for NGFS4, and detected the expression of sialic acid using lectins, MAA and SNA. And so in the mouse lung, as well as in these LET1 epithelial cells, we saw increased expression or increased binding of the lectins, which basically indicated that there was increased expression of sialic acid in the lungs, as well as this knockout cell line. So to confirm that, so sialic acid on the surface of cells can be produced in two ways, one of which is the de novo pathway, and one of which is the recycling pathway. So to prove, and so the de novo pathway is that branch off from glycolysis, essentially. So to prove that this increased glycolysis that cells with mitochondrial disease were actually having spilled off into this accessory pathway, we actually blocked that pathway, that branch point, right here, GF8, it's also known as GFAT, by azacitidine. So by blocking that, we were actually able to decrease the expression of sialic acid in our knockouts, as well as the wild type, as you would expect, but to a greater degree in the knockouts, suggesting that, or indicating that, the de novo pathway is the pathway that is being used to actually synthesize this sialic acid. And so it turns out that it's not only complex 1 deficiency that does this, but could this be more generalizable to other types of mitochondrial disease? So this is a picture of the respiratory chain showing the different complexes. And so we tried to chemically simulate mitochondrial disease using various mitochondrial inhibitors. In this particular case, I'm just showing you data for rotenone. So this is simulating our genetic model, where we're actually knocking out complex 1 function using rotenone. And then we also knocked out complex 5 using oligomycin. And you can see that when you do that in these lung epithelial cells, these wild-type lung epithelial cells, you have an increased expression of sialic acid on the surface. So it's not just complex 1 deficiency that does this, but it's oxphos deficiency probably in general, where you have this overuse of glycolysis and this increased expression of sialic acid on the surface. And so what does this actually lead to? Well, this actually leads to enhanced binding of influenza virus in these knockout cells. So you can see here, this is looking at essentially 20 minutes post-infection. So this is the period during which the virus actually binds, but does not yet enter the cell or get to the nucleus at that point. So we were looking at the expression or the viral nucleic acid that we can detect based on that, and that is actually elevated, as well as looking at HA staining or hemagglutinin staining, and that's actually elevated as well. So there's enhanced viral binding that's actually going on during these early stages of infection in our mouse knockout cell line. So, of course, the obvious question is, can we target this in some way to actually alleviate the enhanced viral load during viral infection? And so you can do that in 2 ways. One of which is 2DG, right, which is an inhibitor of hexokinase as well as other enzymes early in glycolysis. So you can block it early, essentially, and prevent this branch-off point. Or you can use a second agent, which is known as DCA. So instead of blocking glycolysis, what you are doing is you are forcing flux through glycolysis into the TCA cycle. So I'm not showing the TCA cycle here, but what I'm suggesting is that pyruvate is the entry point into the TCA cycle. So you're kind of pushing things along and stealing away, essentially, from this branch point pathway. And so when you do that in the LET1 cells, you can see here on the right, whether you apply 2DG or you apply dichloroacetate, you actually have a decrease in the expression of sialic acid in this cell line. Now, it's a bad idea to use 2DG or deoxyglucose in patients with mitochondrial disease, because this is their only way that they can generate ATP. So you can't shut off substrate level phosphorylation to make ATP. It's their only way of making it. And it would be lethal, essentially, in these animals, or even in patients if you tried to treat patients. So what we did is we tried to use this DCA idea. So in other words, forcing the glucose carbons away from this branch point pathway. And so when we do that and look in the mice during infection, and so the mice treated with DCA are shown here in purple, you see that we are actually able, by lectin staining as well as NP staining for the virus, are able to decrease essentially the sialic acid as well as the binding of the virus. And then when we look at the lung viral loads in these animals, we're actually able to significantly decrease the lung viral loads in these animals. Now, one of the reasons that DCA is important is because DCA has been used as a treatment in mitochondrial disease before to help decrease lactic acidosis. So it may have another use in patients with mitochondrial disease. So clinically, what do these mice look like? The purple is the knockout treated with DCA. You can see that various cytokines or the cytokine profile is actually changed in these animals. But also, too, their weight, while they do continue to lose some weight, it actually is an improvement over what we see in the knockout animals that are untreated, and their clinical scores actually improve as well. So I think it's important to also point out that we offered no additional intervention in these mice. So in other words, we didn't try and enhance their caloric intake to help them deal with infection. We did nothing, essentially. So just by targeting sialic acid alone, we were able to see some treatments. So we think in terms of adding on those additional treatments where in the clinic or in the ICU, we try to nutritionally support these patients or support them calorically, this may have an added benefit, essentially. So in summary, this is a paper that came out in Science Advances actually a couple of months ago. And please have a look if you're interested in more of the details. But the MDFS4 mice become more ill during a viral infection due to a tissue-intrinsic mechanism where the respiratory epithelia is actually responsible. In other words, the lung, a tissue we never think about in mitochondrial disease. And this is really due to what's called maladaptive allostasis. So allostasis versus homeostasis. Allostasis is when there is stress applied to a system and the system has to adjust to that stress. And so when that happens, because of this decreased OX-phos, you have increased glycolysis and this spill off into this branch off pathway of de novo sialic acid biosynthesis. The other part that's relevant is that sialic acid is also used by other viruses like parainfluenza and mumps and coronaviruses, et cetera. Targeting influenza metabolically is becoming a real hot topic at this point because the variable efficacy of influenza vaccination every year. But bringing this up to more common disease, lung mitochondrial dysfunction is actually a common feature in various chronic respiratory diseases, such as asthma and COPD. As a side note, the sialic acid is increased everywhere, right? So not only in the lung tissue, but in other tissues affected by mitochondrial disease, including the immune cells. So this is just looking at a knockout cell line, an MDCK cell line, where we knocked out NGFS4, and you do see increased sialic acid. But then we took our patients, a number of our patients who had a complex 1 deficiency, and also stained by these lectins to look at sialic acid expression. So there is increased sialic acid there as well, which has important implications. It can be involved in SIGLEC interactions. You can have dampened immune responses. It's involved in immune cell trafficking, platelet lifespan, neurodevelopment, endocrine feedback, as well as renal function. So I'd like to thank you for your attention. I just wanted to point out some of the people who did a lot of the work. Eliza Gordon-Lipkin, who's my staff clinician, who helps take care of the patients. Tanya Tarasenko, who's my staff scientist. Shannon, who is my nurse in the clinic. As well as Amanda's project, which I mentioned, Jose, some of his work in developing that flow cytometry platform, as well as other fellows past and present. And thank you very much. I'll take questions.
Thank you very much. I think that was a really fascinating lecture and with many, many clinical implications and take-home points. I would like to kick off the discussion with, I'm sure there are a lot of questions coming up in the audience, but a very simple thing. I mean, you've shown the work of targeting glycolysis in rats. Is there anything equivalent in humans, actually? Has clinical work been done in our intensive care units to see whether similar interventions can improve outcomes?
Yeah, so for mitochondrial disease specifically, dichloroacetate is one of the drugs that's been used, and it's currently in trials right now. And so dichloroacetate is actually an inhibitor of the pyruvate dehydrogenase kinase. What happens when pyruvate dehydrogenase gets phosphorylated, it actually inhibits flow into the TCA cycle. So what you're doing is you're relieving that inhibition by giving DCA. You're forcing glucose carbons to go through the TCA cycle. The purpose is to decrease lactate production. And so this is being used clinically right now. And so we've been talking with clinicians, especially after ICU, neurointensive friends, and things like that, about this. But it hasn't been formally tested yet. But it does have a precedent in terms of using mitochondrial disease and may offer additional benefits in terms of infection.
Thank you, sir, for that wonderful talk, which is actually pushing the boundaries of our knowledge regarding mitochondrial diseases and host pathogen interactions. Do you think adverse events towards live viral vaccines could also be having mitochondrial dysfunction as an underlying cause?
So we don't see that in our patient population. Actually, they actually tolerate vaccines quite well. And I think part of that is because obviously a vaccine is very, or the immune reaction that occurs with a vaccine is much different than what occurs, with a viral infection. Certainly in our mouse models and in our patients, you do see things like viremia. So there's a much more widespread and more systemic response versus what you may see in vaccines. But the real evidence is that we don't actually see that on the whole in our patients with mitochondrial disease. They actually tolerate
Thank you, sir. Questions? Dr. Amit, please.
So I just wanted to know that I mean, you said that they tolerate vaccines well, but is the quality of antibody that they produce, is it high affinity and high neutralizing, or is it different? I mean, that's more important.
Yeah, so absolutely. So what I primary responses. So in other words, if you look to detect shortly after vaccination, you will find antibodies. The problem is long-term, sustaining them long-term. So we find over time that they actually lose their titers over time. We certainly have reported, we've reported on that in the past, and that's one of our publications. And so that includes for things like measles and varicella and things like that. And part of that reason is because we're working on it now in mouse model is because long-lived plasma cells in bone marrow, which are important for the maintenance of titers, are heavily reliant on OX and mitochondrial function. And so in the mouse model where we knock out the T cell mitochondrial function, long-lived plasma cells are severely reduced, and therefore the diversity of the titers are actually reduced, as are the levels of the titers. So yeah, they're able to develop this response initially, but they're not able to maintain it over time.
Another question is that there's a kind of a paradox in the sense that these patients have a very elevated type 1 interferon, at least in your mouse model, they have a elevated, have a type 1 interferon overdrive, yet they are not able to clear this viruses properly. So is it because there is a lack of effective neutralizing antibodies which are important? And why do you think it is the paradox? I mean, like, on the one hand, you have got a innate immune response which is on overdrive because of damps or whatever other reasons, yet they are not able to clear these viruses effectively.
Yeah, obviously any type of virus clearance involves a number of different aspects, and so while they do have this increased kind of overdrive due to damps, and actually also due to metabolic reprogramming itself, because inflammatory and interferon type signatures can be driven by this kind of reprogramming to glycolysis, which they tend to try and do to get box office function. So, I think the most straightforward answer is obviously the ability to protect yourself against viruses, even clear viruses, involves this kind of integrated network of innate and adaptive immunity. And so they don't, we've shown in the past that they don't really have this kind of very diverse antibody repertoire, or even robust antibody repertoire in terms of viruses, to be able to protect them in terms of subsequent exposure.
Thank you.
Thank you. Are there any more questions from the audience? If not, I think we'll close this session. Thank you very much, Dr. Peter Maguire, for a brilliant and very, very revealing session.
Thank you very much, and I wish everyone well in the rest of the conference, and have a good evening.
Have a wonderful day. Good morning.
So before we sign off for the day, just one or two announcements. So we have the GB meeting now, and please stay back for the dinner. So we'll start the dinner early because we have been having a long day, almost 10 hours, and I think things ran quite well. We are just 5 minutes late. So that was good. So, and again, tomorrow morning, things start early. We have the opening batsman is at 8:30 a.m. She'll be speaking from, again, US Novel Autoinflammatory Diseases by Professor Rafaela. So that is all, yes. 8:30 in the morning, yeah.
So I would request the ISPAD members to stay back for the AGM. Rest others can relax outside. Members, please stay back.